take this off real quick. Aloha mai kako. As the interns are kind of settling in without talking, welcome to the 2025 PIPES Intern Symposium. We're super grateful to have you guys here to see the final presentations of our 30 intern cohort for this summer, where you'll get to experience their projects, their research, and their experiences and their journeys throughout the 11-week program. I'm Nalu O'Connor, I'm the PIPES program lead. We also have our staff here with us. You guys want to introduce yourself real quick?
Aloha mai kako o wau ihono o Kiana Davis. Aheli mahana o no keia papahana ke kahi. Aloha mai kako, my name is Kiana and I'm also a program coordinator with the PIPES program, and I'll hand it over to Vai.
I'm wondering if you can hear me through the, okay. Is it okay? Okay, kalamai, I have a little scratchy throat. Aloha, my name is Vai Needham. I'm also one of the program coordinators. Just mahalo nui to everybody for being here for our symposium. Thank you.
Mahalo nui. So we have a little opening here, opening ArcGIS story map, just to share a little update with you folks on PIPES and our little journey, and then we'll start with our first presentation for the day. So I think what we really wanted to do, this is a picture at Nenewe with our PIPES cohort for one of our events that had alumni, community members, and our cohort kind of engaging all in one space together in Waipio. To give some grounding and context, I think we wanted to just introduce that oli real quick that you guys heard.
Like Nalu said, we started this morning with an oli komo. During orientation week this year, all of the interns got to learn this oli komo as a way to ground themselves and get ready to center for all of the exciting things that were going to be done this summer. This oli komo was haku'd last year in January when our team came together with the mana'o to really understand the mo'oku'auhau of PIPES, and from that came an oli komo, 3 different mele, and the mele mo'oku'auhau. From here, we can see that the purpose of an oli komo is to ground ourselves to get ready. And so we talk about some of the different wahi that are important as a piko of hilo. We have pu'umaliu and a couple of other wahi, as well as the ala mo'o'aina. You'll hear a lot of mo'o'aina conversation over the course of today. But that was really the impetus of that. And then at the end of today, you'll actually get to hear our mele mahalo. The mele mahalo is a way that we mahalo not just the people, but the places that we've gotten to interact with this summer. A really important part of the mele mahalo, and probably one of my favorite lines, is hie hie halau lani ika hayo muo na manu, ika a mui mui ika wau ma'ukele. Alaila, nu'a mai na lehua ile kahiko ula o uka. And that's really where we get to the imagery of the interns and the mentors, with the mentors being the kumulehua. They're the kahua that is super pa'a. They had to hold the line sometimes over the course of the summer. And then the interns being the ie. They're the things that grow forth from the kumulehua. And if not for the kahua, the foundation that the mentors have provided them with, the interns wouldn't be able to flourish. And over the course of today and tomorrow, hopefully you'll get to see all of the flourishing and all of the hie that the interns got to partake in and grow through this summer. And now we'll go into history of PIPES.
We did rehearse this, I promise. Yeah, just for people who haven't heard about PIPES, or just in general for the kind of new shifts and new directions that we're taking programming, we thought it'd be good to start off with some history. So 31 years ago, PIPES was founded by Sharon Ziegler Chong and other like-minded people. And this first program was created called the Micronesia American Samoa Student Internship Program, or the MASSIP program. And the goal was to, or the intention was to get local students, especially of Native ancestry in those places, into the conservation sector to represent their people in their home. 3 years later, in 1997, that same kind of model came home to Hawaii, and the University of Hawaii Hawaiian Internship Program was created. With the same goal, with the same objective, focusing on Native Hawaiian students and other local students and trying to get them engaged in education opportunities and natural resource management career pathways. Fast forward to 2002. This is, I think, PIPE's first time getting the National Science Foundation REU grant. And so this just broadened, the pathways for students to have an internship. A really important part of what PIPES has done in those last 30 years is provide paid internships, which is really important for our people, really important for them as emerging professionals. So the research experience for undergraduates is the RU, and we've been really blessed to continue to have that kind of support for programming today. So yeah, that's been the last 30 years, and for a long time, PIPES was kind of the umbrella over these 3 programs. And in a little bit, our team will share with you kind of how that has shifted and some of the changes that have been made. But we still move forward, with this deep mahalo for all of the people and partnerships and mentors and interns that it's taken to have successful 31 years of paid opportunities in the conservation sector. So really excited that you folks are here and with us as things kind of change and shift.
Yeah, mahalo nui vai. Sorry to bore you folks, but we just want to also quickly mahalo our sponsors. And can we give a round of applause for them? Mahalo. Without these folks and other organizations, PIPES would not be possible. We really would like to thank Hau'oli Mauloa Foundation, Faith, can you raise your hand? They've been a long-time supporter. Yeah, can we give them a round of applause? They've been really a long-time supporter of PIPES and other organizations at the university as well as in our community for conservation and this concept of aloha aina. So we really are grateful to have a partnership with them, as well as the Hawaii Pacific Foundation. I know I don't see Janine here yet, but she'll be coming in. They have been a huge support, especially with this federal situation, in helping PIPES continue programming throughout this summer as well as the year. And I'll just mention a couple more. Our Sea Grant Ohana with the Ahupua'a Accelerator Initiative, AAI. They are a new partner this year, as well as Kipuka has been a huge support. Shout out to Hua and the team and the Ohana over there. Without their support, I don't think this summer would be possible. And the National Park Service. So can we just give another round of applause for those organizations? And I'm gonna quickly, so that we don't take up more of your time, I really wanna get to the interns, but this is kind of our grounding mo'aina framework, which is an aloha aina system of practice that the students were, whether they know it or not, were kind of put through this year as far as capacity building, but also just programming. So this is kind of our operational framework of how we're conducting ourselves now at PIPES. As for concepts and relationships, na'au, aina kaya ulu, and ka'au. Why that's important is because that is our new pathway programs here at PIPES. So interns had experiences based around these 4 relationships and created these types of threads throughout the summer. And I'm gonna quickly move us through that. Yep. I want us to get to the interns because that's the most important thing, and we have a really cool map that we really want to show you folks. So if you want to talk more about the mo'aina framework, we can talk stories later. Yeah? Thank you.
Yes, like Nalu said, we had 30 interns this summer spread across Hawai'i Island and on Kauai. That's why our map looks a little bit funny this year. Prior to COVID, PIPES was Moana Nui a Kea wide, spread across the Pacific, but due to COVID, was brought back to Hawai'i Island, and we're slowly getting our feelers out there, expanding back to where PIPES once was. But as you can see, this year was still a focus on Hawai'i Island. And so here's a little overview of where all of our interns were this summer. We had Kapailoa Aki with KBCC all the way out in Ka'u, occasionally. She was a data science major, and so a lot of her work didn't actually have to take her all the way out to Ka'u. We had Mana Akiyama with Huimo up in Hamakua. Mr. Kamu Andrade Stout with McFerp. high up on the slopes of Mauna Kea. Seth Asano with Symbrosia out in Kona. Rosemary was also out in Volcano. Ho'oli Kui Yakman with Pohaku Pelemaka out in Ka'akepa. all the way across the Pai'aina. Leinalu was with Namako Onauna and Pelika. Eliana was in Ka'alu'u. We had Miss Kamaka Frasco down at Pack Rock with Steve. Glory went all over and had a halada hana nui with Pilina Aina. We had Mika Gallardo out at Pu'ukohala. Kahinu at Sierra Club. Mr. Kainoa Cam. He has a lot to say about those Pu'a experiences. Kyle out in Kona with Bisque. Liua doing some Ko'a stuff. Our boy from Laupahoehoe out in Laupahoehoe, Kamaha'o Matsumoto. Modi'i up at Lama Lama Kaulu. Sema with the Indigenous Cropping Systems Lab, lots of Ava stuff. Aina Perry out in Kaimu. Our Pele girl, Kamalani Poi Poi with the Pele. Wai'olu Kea Publico with Imi Pono, lots of Huaka'i over there too. Haola was all the way back home in Kahuku. All over. Kamaile with Dar. Lila down in Keaukaha with Liko Napilina. Sister was all the way in Kaimu though, Miss Ella Shapiro. Kanani made the commute every single day out to Hikiola and Kealakekua. Wailea with Kahalu'ukuahava. Back in Keaukaha with Matlin, with Kumuola. Kili Pohe at home at Maku'u. And rounding us out, Kale Kukui with Pohaku Pelemaka.
Mahalo nui, thank you. We just really wanted to introduce, I know we're at 8:43, we really wanted to introduce our intern cohort as quickly as possible, and you really see the breadth that the interns and the journey that they all kind of went on as far as their place and their internship. So without further ado, I think we're gonna jump right to it, into our symposium with the first intern, coming from American Samoa, Liwa Leato. Hopefully this is that a good height? Hi, mahalo.
Whew. Hey guys, I have some nerves on me, so don't look at me too hard, all right? All right, but Tala Falava, Tayama Nuia, and Aloha Mai Kako. My name is Liwa Levausina Leato, and here is my PIPES 2025 summer project of Mapping Mana, a cultural and climate inventory of Kalaimano. And I am part of also the Ka'au pathway, so let's get into it. So my roots trace back to American Samoa, and I really credit my Atunu Upele for really serving as the backbone to my commitment to service as a Tama'itai Samoa steward to both our vai, our water, and our whanua, our land. And so as an indigenous scientist just trying to navigate space in a Western-dominated field, the Moa'aina framework from the PIPE staff really pushed my initiative to apply to the program, and shout out to the PIPES team for seeing some potential, because so thankful to be part of this year's Hui. And it's been such an honor. I've learned so much as a guest within Hawaiian space and land. And I've just had such a pleasure growing and learning alongside my fellow oceanic cousins. And so it's truly been a journey, and I'm just excited to take back my new skills and the new perspectives that I've learned over the summer back to my own home of American Samoa. But let me go ahead and hype up my host site for this summer. I've had the pleasure of working with the Multi-Scale Environmental Graphical Analysis Lab, or the Mega Lab. Shout out, we Mega in the crowd, let's go. But if you aren't too sure who they are, let me just say they are an awesome group of unconventional scientists, artists, and surfers who utilize tools like photogrammetry to create innovative solutions to protect our oceans and distribute those resources to communities that need them most. So Dr. John Burns, Dr. Hanani Kane, Makoa Pasco, Kainalu Stewart, and my fellow mega intern, Mariella, have truly been key figures in shaping my ka'au throughout this project journey. But most especially, gotta give credit where credit is due to the most hamakupuna at Kalaimano, Auntie Le'nala Leitner, who I'm sure is a familiar name to many of us in this room, who has been so gracious in sharing her mo'olelo aina of Kalaimano, and as well as her mana'o and ancestral ties to the shoreline. And she really was an integral part of putting this project together, as she truly is a living testament to the vitality of pa'akai, or salt, to the ahupua'a of Kahupulevu. Which brings me to my project of creating a baseline of cultural inventory of the poho pa'akai, or the man-made salt pans, and the kaheka regions, or the natural basins within Kalaimano, while also investigating the risks of sea level rise to these cultural sites. And here we have some examples. Here we have a picture of a natural basin, or one of the kahekas. And in this next photo, we have an image of the poho pa'akai. And as you can see, these poho pa'akais are typically made utilizing lava rock put in either a circular or rectangular formation, and are insulated with a cement-like mixture called mortar, made of limestone, water, and sand. And so all this awesome work has been done within the ahupua'a of Kahupulevu, also revered as part of the beloved lands of Kekaha. Rich in ecological diversity, this sacred landscape has long shaped and sustained traditional lifestyles. For generations, families have traveled from Mauka to Makai. And along these ancestral paths, communities heavily relied on these integral coastal shorelines for sustenance, like Kalaimano. And in alignment with deep ancestral connection to land and sea, contemporary efforts have been made to restore and protect the Kahupulevu marine resources and this has taken shape through the Kahupulevu Marine Reserve, or the Tri-Weight Program, which was essentially a 10-year no-take rest zone spanning from Kekau Point all the way to Kalaimano. And now Kalaimano was once a center of pa'akai production. You really cannot express like how vital pa'akai was to the families in the Hapua'a. It was able to sustain all the upland communities, ranging from farmers, ranchers, to coffee farmers. And pa'akai was used as a food preservation. And ultimately, across the shoreline, over 100 po'o pa'akais typically were located ishland, push production of amounts of 800 to 1,000 pounds of salt weekly. But however, through the introduction of electricity, the demand for pa'akai had decreased, and many of these po'o pa'akais remained unmaintained and unused and kind of lost away to time. But however, the practice of harvesting pa'akai is still well and alive today, primarily through harvesting from these natural basin regions. And if you ever get the chance to head out to Kalaimano, please visit the Kalaimano Visitor Center, and you will get to see Auntie Lei there. But jumping into the ha'alele and of it all, to document and analyze these poho, pa'akai pans, and kaheka regions of Kalaimano, we employed advanced spatial and modeling techniques. First, we used structure for motion photogrammetry, and essentially this method utilizes capturing overlapping images using GoPro cameras and ensuring going in a lawnmower style motion, or a zigzag motion, you create a 70% overlap for these pictures. And so ultimately, when these pictures are collected, we utilize a software called Agisoft Metashape to create our 3D mosaics. And ultimately, these photos generate point clouds, high resolution 3D models, and other products. And then next, After receiving these products, we were able to annotate high-resolution drone models of the north portion of Kalaimano to notate the Poho'opaʻakai and Kaheka regions, and this was done using ArcGIS. And then to understand the vulnerabilities of climate change due to severe sea level rise, we ran 4 sea level rise models based on the 2022 report of the Hawai'i Sea Level Rise Vulnerability and Adaptation Report. And these comprise of 2050 low and high scenarios ranging from 0.2 meters to 0.4 meter sea level rise, and 2100 low to 2100 high. High scenarios of 0.39 meters to 2.4 meters rise. And so essentially these models will help us visualize the potential inundation of impacts on the cultural landscape and inform long-term planning and conservation strategies. Delving into the huaka'i, let's check out our pa'akai inventory. So our research, as I mentioned before, was resulted in detailed inventory of pa'akai harvesting areas utilizing the structure for motion models. And so a total of 16 kaheka regions were mapped, covering a range of 1,000.78 square meters, and a total of 11 po'opa'akais covering 1979.14 square meters. And then ultimately to assess the vulnerability to sea level rise, 2 sets of low and high scenarios for the years 2050 and 2100 on the flood maps. So you can see the pink areas represent the low projections between 0.2 to 0.4 meters, and the green indicates high projections ranging from 0.46 to 2.4 meters sea level rise. And also these blue regions highlight the kaheka and the po'opa'akai, or these pa'akai harvesting areas. And so as we can see in 2050, in both the low and high scenarios, we see no overlap with potential projection floodings to these pa'akai harvesting regions. And the same can be said for the 2100 low assessment model. But as for the 2100, we see an significant increase in coastal flooding, and ultimately more overlap with some of the kaheka regions that are located near the shoreline. And so ultimately, these projections are a call to action, and to highlight the increased vulnerability of these cultural resources to climate change and underscore the importance of long-term conservation planning. These projections call us to take action and inspiring efforts to restore through traditional practices that build the resilience of these cultural sites. Auntie Lei has even expressed her hopes of restoring 1 or 2 of the po'opa'akai spaces for education and preservation. And so restoring these sites would not only hone into the ingenuity of kupuna, but also creates and protects areas for salt mapping, helping to make these areas more resilient despite the rising sea levels. And so this work only highlights the urgent need to protect and adapt these cultural landscapes through indigenous knowledge. And so ultimately, and referring to this, the graph below, this will show that in the 2100 model, a total of 20% coverage of the flooded Pa'akai region would be affected due to inundation. So ultimately, throughout my PIPES internship, I've not only gained these professional skills, but also deepened my connection to my identity as a Samoan scientist. And despite with the challenges with ArcGIS and the shifting project timelines, I found strength in mentorship and the principle of prioritizing relationship over outcomes. And so ultimately this summer at Kalaimano with the Mega Lab and with my PIPES Ohana, it's really deepened my kuleana to serve with intention, and ultimately grounding me in my values of tautua, or of service, and reaffirming my role as a Samoan scientist. And so beyond learning the technical skills like GIS and photogrammetry, I gained wisdom in slowing down, building trust, and honing in on the community-led research. And as I return to my studies, I carry a renewed purpose to serve our Blue Pacific continent with humility, integrity, and cultural grounding. And ultimately, mahalo nui loa to all the PIPES Ohana, my awesome host site, the Mega Lab, and all the funders and organizations that made this work possible. Ultimately, without your commitment to community-based research, none of this work would have been possible to protect, to document, and highlight these cultural sites. So, mahalo nui loa and whawhitai tele lava. Can I go? Questions.
Hello. Okay. Do we have any questions in the crowd? And I can run this mic to you, unless you got a loud voice. Hinano or Li'i, is there any questions online? No? I might give one to the Mega Lab. Oh, there we go.
Just regarding your interests, what sparked your interest in Pa'akai?
Pa'akai, it was, so initially entering the Mega Lab, I was put on the ongoing Opihi project, which was looking at intertidal habitat metrics, specifically looking at Opihi and limu abundance through the seasonality. So that kind of brought me to Kalaimano, and then whilst at the site, I've never seen a Po'opa'akai before, and it was honestly like, what in the world is this? And so after talking story with our team, and as well as like the kupuna from Kalaimano, they were able to inform me, and then I was just, wouldn't it be so awesome to document these cultural sites and these figures, and especially because after Auntie Leia like shared the story of Pa'akai, how it was once such a high demand commodity, now a lot of these pans are just remaining unused and unmaintained. So just wanting to highlight that journey and highlight the value in what it used to be. So mahalo.
Leo, I was just curious. It was really awesome seeing the whole journey come together in that final product. Were there any like key moments or experiences or insight that you got that is now putting you in a position to think about what you could bring back to Samoa? Did you get any ideas of ways you could help improve coastal management there?
Yes, so I was just talking about this with one of my good mentors. She works for Sea Grant, based in American Samoa. Her name is Kelly Taranjo, and so she had told me the need for the use of photogrammetry and the way they're trying to implement and look at their own sea level rise risks within American Samoa, because flooding has been a pretty significant issue back home. So she had just sent me an email just the other night saying that my skills that I've learned with you folks would be such a great help to the effort that they are now trying to push for in American Samoa. So definitely all the skills through learning with ArcGIS and photogrammetry is definitely applicable things that I could definitely take back to Samoa and implement almost immediately, depending on when I go back.
Okay, we have 3 questions online. I think we only could take one. So I'm sorry for the people that we won't be able to field your question. Do you have any advice for future Samoan scientists?
My advice is just to, oh wow. I feel like being brought up back home a lot of the times, I feel like a lot of our students kind of like stray away from science intensive backgrounds, especially because of like the prereqs that come with it. Like if you're not good at math, if you're not good at science, it's like, why be in this field? But when essentially like all these spaces are meant for everybody. And so I would just encourage to just push yourself. Don't be afraid to push boundaries and over-exceed your own expectations of yourself, because I think a lot of times students get in their head about whether or not they think that they're capable of even doing the work, and I think that stops them from making the initiative to even try to be in those fields. So I guess my advice is lead with your heart and just go for it full force.
Mahalo nui. Can we give a round of applause for Liua? Awesome. Up next, we have Kamale Schultz, who was with the Division of Aquatic Resources. His title is A Dive into Wailoa and Waiakea Ponds.
Having to go after that amazing presentation just set the bar just a little bit more high. Aloha mai kako. My name is Kamaile Schultz. I am a PIPES intern in the Aina pathway, and I was pleasured to be partnered with the Division of Aquatic Resources with Troy Sakihara, Devin Algiar, and the whole DAR team. Please join me as we take a dive into Wailoa and Waiakea River, or Waiakea Pond, through the Waiakea Restoration Project. But first, we need some ho'olauna. For 5 years of my life, I called Oahu, East Side, Ka'ava, Kailua, Kaneohe, my home. That is a picture of the first ever fish I caught by myself. Don't look too hard, it's a hammerhead shark. But I now pursue a Bachelor's of Arts in Marine Science here at University of Hilo, and I always had a connection with the kai. It was always something that would connect to me, and I would always grow up with that mentality of preserving Aina for future generations, and that's something that I want to pursue for, like I said, preserving for the future. My mentor site is the Division of Aquatic Resources in Hilo. The fun thing with them is that they like to do a lot of estuary work compared to a lot of the other DARs found throughout the islands. And I was stationed at Wailoa Station. My mentors, Troy Sakihara, an aquatic biologist with multiple amazing publications, as well as Devin Algiar, a former PIPES alum and specializing in habitat monitoring, as well as has a huge fondness for coral reef. So to jump into this Waiakea restoration project, the plan is to reintroduce aka'akai. I know some of you Olelo Hawai'i are thinking it's green onion. It is green onion, just a lot bigger. And the reason they chose aka'akai is because it allows for pua'ama growth, or pua'i'a in general, as well as it doesn't spread as fast as a certain grass I'll talk about later. But the project is also to remove that grass, which is California grass, if you guys haven't heard of that already. And it's also to study how California grass is affecting our estuary habitats. So my part of this project was to create a baseline to kind of not jumpstart it, but to jumpstart it. And to just get this project running and allow DAR to build off of what I had gained, the data that I gained. To introduce all of you who are not familiar with my location, the river Wailoa is the river I was working on. That is the river from the river mouth, our pier, all the way to the double gate bridge, right here. So this double gate bridge to here is considered Wailoa. We then have Waiakea Fish Pond. This is a historical fish pond I'll go into later in my ha'alele section. But this is from the double gate bridge over here. This whole section was considered a local i'a back in the day. And going into where I was actually working, or more so working, is Moho'uli Fish Pond. This is where we took our baseline and all our water quality samples. In our ha'alele, I had kind of 3 main goals I wanted to do this summer internship was improve my data collecting skills, learn how to cast net, and improve my kilo skills, and just to become more familiarized with the research area. So in order to get familiarized, you gotta do your research. So I was working in the ili kupono of Pi'opi'o. If it sounds familiar, there's a volleyball club named after it. The bridge by Kamehameha statue is named Pi'opi'o. And this ili kupono was known for its ali'i, I could say that. It houses a lot of the ali'i nui. Some notable names include Umia Liloa, Ke'ua, son of Kamehameha, or father of Kamehameha, as well as both Paia Kamehameha and Liho Liho. As you guys can see, the Wailo River wasn't as wide as it is, and that's because in the back of Waiakea Pond, they had to do the sugarcane mill. So that's why they had to widen the river, and a lot of that dredging did kind of clear up a lot of our mulivai outside of Wailoa for sugarcane. For fish identification, this was the first thing I saw when I was introduced, and it's our 3 mullet species. So this is an official DAR ruler of each mullet species and how to identify them. So ama-amas have the blue coloration behind the pectoral fin. Wowo have the yellow dot, and kanda, which we don't like, has no coloration at all. So this was kind of what I had to go off of, but not just how they look at when you catch them, but how to read their behavior in the water. So when it came to doing surveys only targeting certain mullet species, it's important to know how they act, because you don't want to waste time catching invasives when you're supposed to catch the native. And then here are some of the research techniques that I gotta learn. First off is cast netting. If you would believe it, I've never threw a cast net until this internship. I had some amazing teachers teach me, and he's not here, never mind. I had amazing teachers teach me how to cast net. Here are some later hands-on stuff. So this is a 1/2 meter square, which we use to collect a lot of our paragraphs, and just all kind of hand tools as such, as well as a YSI. I have used YSIs before, but just using it in an estuary habitat was kind of not different, but they had its differences. To go into our huaka'i, it's literally the same as our holiday. The data collection that we need is our water quality, fish identification, and my most favorite of all, amount of California grass. For our water quality, this looks like a lot. For those of you who are not familiar with using a YSI, these are all of the data measurements that it reads. And so this was a rough map of the data points of where we took our water quality testing. The blue is our baseline, and the red is our restoration. As of updating right now, we've lost one of our baseline, but we do have all 5 of our restoration sites. Well, actually, we lost 2 baseline sites, but. And now for my favorite part, fish identification. So if I show you this photo right there, not a lot of guys are gonna like, why is it just a picture of water? If you look really good, there's at least 50 ninui in that photo. So while doing research at the restoration area, we had a school of ninui follow us on the boat. And it was really funny to see every time we came back, they would still be there following our boat. The red thing that I outlined is a baby kaku, barracuda. This was on Wailoa Station, and it's kind of interesting that even in these estuary habitats, it houses so much different type of i'a. And this is my favorite part. In this photo, I have 2 types of mullet, and I'm really blessed that we're able to get this photo, just to show how both bad and how challenging it is to do fish identification. In the blue is our native ama. In red, I just only circled this one because that's the clearest to see, is a kanda, or Marquesian mullet. And that is the only ama that I was able to find in that photo. I'm pretty sure we only extracted two or three amas from that whole net. And that just shows how bad our invasive problem is in Wailoa. Oh yeah, and then this one, first ever cast with a cast net. Yep. You know you gotta pose. But this one too it's so much, in that net alone, there was only 2 amas. And that just shows how bad these invasives are affecting our natives. This is how you would measure. So you'd go from the head, the eyes, all the way to the tip of the tail over here. So that's how we would measure our mullet, but we normally have a half-meter PVC pipe that we use to collect. And then this is where I talk about fish behavior. In this picture right here, I am 90% sure that is all condom. just because if you can see, a lot of it is not linear, but like more formative, like they're precise. That's something that the DAR team had taught me about Kanda, is that they're more, when they flee, they jump out of the water, they jump in one direction, versus the Amas who like to sporadically jump. And that's kind of how I was able to identify these fish right here, because after I took the photo, I scared them, and they all jumped in one direction. And now, for California grass. If you're not familiar with California grass is a super invasive grass brought in for cattle, and California grass likes to grow on the top of soil and water. These photos here are from Moho'uli Fish Pond, one of the first days I did my testing, and just to show you guys how bad the California grass is over there. And so with this one, we took both an emergent and submerged height. Submergent is, or emergent, let's start with emergent first. Emergent is from where we place that 1/2 meter on the ground, and then however much high the grass went, the tallest would take emergent height. Submergent height is from wherever the 1/2 meter was all the way to the ground. And then mass was taken in grams of both the emergent and submerged. And we also wanted to take our weight. So we have a dry weight and a wet weight. Wet weight is basically how much, whatever the natural state of it is. And dry weight is considered the biomass, how much of just the grass there is in the area. And then after that, we'd use a drone to do a mapping. These are my tubs. I've seen them the whole internship. So like I said, everything on this side was emerged. Everything on this side was submerged. And after collecting it on June 10th, I let it sit for the whole internship, just to show you guys how long it took for all of this to dry. We even had an oven that we used, and it didn't even help. Well, it did help, but it was pretty tedious. So yeah, 36 days in counting. That was before I made edits. And so we would weigh all the grass that we collected with a 400-gram scale, and we'd use foil, just because foil is a lot lighter, and so it wouldn't mess up the scale. This is a graph that was created before we did our completed dry weight, and it kind of just shows how, as the water depth increases, the weight or the mass itself will increase as well. And now for our data analysis. So here are the two data sets that I was running. It was water quality, which is our YSI, and our California grass data sheet, which has both the dry and wet weight. But just to put it into perspective about how much grass there is in this area, in this little island alone, there is around 17,178 square meters of grass. That's around 2.5 football fields. In wet weight, there's around 554 metric tons of grass. That's 3 to 4 blue whales. That's 3 blue whales, guys. In dry weight, in straight biomass, there's 62.1 metric tons. That's the same as an army tank. That's an army tank, guys. But something that the statistic I don't really like, is how much estuary habitat it occupies. With 13,644 cubic meters. That's almost less than 5 Olympic swimming pools. That's how much of our estuary habitat that we're losing. And we can share that information that I just told you via some educational signing like you see at the beach, or just doing some outreach events with kids or summer programs. To begin my ho'ina, I'd like to say this olelo no eo. He lue lue ka upena e ku'oi. The fine mesh net is the one to let down into the sea. My biggest takeaway was the invasive species. California grass sucks. This was a picture taken directly after our tsunami that we had. And people had to go over there and clear the boat ramp because it was blocking our boat ramp. No one could leave. This is the second part of it. But just besides the invasives, I also found out that anything can be an estuary, even a honu. I seen her 2 times while I was at Dar, both on the high tide, and I'm pretty sure she was trying to find some limu to eat on. But you can almost find anything in an estuary. That's why estuaries are considered so important. It's because without estuaries, we wouldn't have our pool, we wouldn't have our big and even for us Kanaka, back in POPO times, having the lo'ikalo as well as fertile lands to grow on is something that we need to bring back. And so some narrow impacts from this would be, just how to take data and collecting more efficient, I guess. Or how do you build off of my work, as well as creating a future research project. And I guess the biggest, broadest one would be the start of the Aka'akai restoration, going in there and actually planting the Aka'akai and taking out California grass. So from this work, I was able to better my collection skills, better my Excel work, better my work in estuaries, and just build a better understanding on why we need restoration projects. I hope that this leaves a legacy as a building block, as something that people in future Kanaka can build off of my work, and that it taught me a lot of valuable skills that I can use in life, and just strengthening my overall goal of preserving Aina for our future generations. Mahalo nui loa for all of the amazing foundations that helped me get here today. From the amazing PIPES Ohana, from Kipuka, the Division of Aquatic Resources, I give a shout out to all you guys. You guys helped me out so much. And Ha'oli Mauloa Foundation. Mahalo nui loa. And I now open the floor to any questions.
Does anybody have any questions for Kamaile? Oh. Hold on, I'm running.
Awesome presentation, Kamaile. Because you're working with 2 invasive species, one in the water and one on land, what is your advice, or do you guys have advice of clearing more land first and then the fish, or fish and then land, or both at the same time slowly?
As of right now, we're gonna start taking out the California grass first, the land, and then hopefully we can start introducing our ama back. So we're gonna try, hopefully give them an environment, safe place first before we reintroduce fish.
Mahalo. Does anybody else have a question? Are there any questions on? Oh, okay, maybe just yell it out.
So can you explain how you lost 2 sites?
So with one of the sites, the code name was Baseline 5. So when we first originally took the baseline site, I observed that a lot of the amas was circling around the California grass. This was at this site. So we had a change in tide and we lost the California grass patch. The second one that we lost was because the banyan tree had fallen on it. And so it pushed the grass out.
Okay, I think we have time for one more question. One more? Oh, yeah.
Is there any evidence that the California grass is a nursery for any, anything invertebrates or fish or anything? Who uses that?
For my just kiloing it out, I've seen a lot of our mollies, invasive mollies and kanda using it. That's why they want to introduce a lot of our aka'akai, because aka'akai allows limu to grow on the river bottom, and that promotes our pua'amas to go in there and eat it as well.
Okay, can we get a round of applause for Kamaile Schultz? Up next, we have Rosemary Bearden, who is with the University of Hawaii Hilo Elala Lab. Can we give a round of applause for Rosemary?
Aloha mai kako. My name is Rosemary Bearden. I'm in the Ka'au pathway, and my PIPES project this summer was titled Arthropod Community eDNA as an Indicator of Forest Health. So getting into my hua, I'm from Hawai'i Island, specifically Hilo Paliku, and the first native forest that I ever visited was in Hawaii Volcanoes National Park. And I remember it really clearly because I was just immediately so taken with our native species, and it was, I mention it because it was a really big hua for me that sparked my interest in our native species and their protection for future generations. And now, all these years later, having just received my degree in biology for ecology, evolution, and conservation from UH Hilo, my PIPES project took me back to those same forests, and this time I got to collect data that will help us understand how ROD is impacting those forests, and therefore how best we can malama those forests in the face of these impacts. Oh, and this picture is an EO that I came and visited us, me and my team, while we were collecting data in Hawaii Volcanoes National Park. So that was really special. Oh, thank you. I was like, what? Okay. Quick intro to my site. I worked at Alala Biodiversity Lab, which is a molecular ecology lab on UH Hilo campus. And my mentors were Dr. Natalie Graham, who you can see here in the forest with me, and Hannah McLam, who was my mentor for all things lab. So my project uses eDNA sampling to compare the arthropod communities in fenced and unfenced forests on Hawaii Island. eDNA, or environmental DNA, is the DNA left behind by organisms in the environment. So just like how when we're walking through our environment or touching things, we leave our DNA behind, so do arthropods, and we can pick that up with eDNA sampling. So why look at arthropods? Arthropods are really useful indicators of forest health. They're one of the best measures of fine-scale habitat turnover because they're very small, very abundant, and there are a lot of indicator species, which just means that when we see certain taxa of arthropods, that indicates to us that the forest is healthy. They're also highly linked to their habitat, which means that there's a lot of turnover, and they're very easy to measure, making them useful for measuring biodiversity. So quick little slide about ROD, or rapid ohia death. It's worsened by the presence of feral ungulates in our forests, not just because they can have ROD spores on their bodies that they carry into the forest, but also because they wound our trees, which allows the fungal pathogen to enter the tree, killing it. As you can see in this figure made by the SDEV lab, parts of the forest that are unfenced have a lot more cases of ROD than fenced parts where ungulates have been removed. ROD is a major disturbance to our native ecosystems. Ohia is a huge keystone species, and so it impacts a lot of native species to be losing this tree. And ohia is also super culturally important as well as ecologically important. So what is a healthy forest? How are we quantifying forest health in this study? We have 4 different ways to measure forest health in this study to try and understand the impacts on the forest that ROD is having. The first being alpha diversity, which is the number and abundance of species in a given area. The second is beta diversity, which is how the community is changing over space and time. The third is functional groups, which refers to the ecological roles of different species. So for example, some are pollinators, some are predators, etc. And the final one is species interaction networks, which refers to the associations, like between a pollinator and its plant, that different organisms have and how often these associations are occurring. So examples include between an herbivore and a plant, or a predator and its prey. Getting into our hypotheses for this study, we have 4. The first is about alpha diversity. We expect to find that fence plots with intact canopy and reduced disturbance from ROD will exhibit greater alpha diversity, so there will be more species present. And we expect that unfenced plots will show reduced richness and dominance by only a few taxa. We also expect that community composition will differ between the fenced and unfenced plots, reflecting the changes in exposure and biotic pressure caused by ROD. And we expect that unfenced plots will be enriched more so in airborne and generalist taxa. Thirdly, we expect that fence plots will support a wider range of functional groups, whereas unfenced plots will be dominated mainly by detritivores and disturbance-tolerant species. And finally, we expect that fence plots will show higher specialization and structured species associations, whereas unfenced plots will have more generalist interactions and they'll be more diffuse. So getting into the ha'alele, this is a map of the sites that we resampled this year. This was a resampling effort, so all of these sites, data was collected last year, so we do have an understanding from last year's data of how things are changing across the landscape, but by resampling, we'll also be able to compare how things are changing through time. As you can see, for each site, there are 4 different plots, 2 fenced and 2 unfenced, which are all quite near to each other to control for any other factors that may be present. And so those were our sites. And then this is a quick run through of the methodology, simplified because there's a lot of it. But the first thing we do when we go out to our sites is we bag plants by abundance. So if there's twice as much kawa'u or ilex as there is kanavau or brucacea in a plot, we take twice as many bags of the ilex. We want the number of bags to be proportional to how much of that plant is present in our plot. So once we've done that, we take all the bags back to the lab and we do an eDNA filtration. We use a leaf wash technique, so we add water to the bag, allow it to run over the leaves, which washes off the eDNA, and then we pour this water through the filter, and the filter inside the filtration system combines, holds on to the eDNA that passed through. Then we put that in a buffer for a couple of weeks, and then we start purifying and isolating the DNA, which begins with phenol chloroform extractions. Then we do qubit to quantify how much DNA we have, and then we go to the first round of PCR, where we amplify the DNA, and then we run a gel electrophoresis to see if the correct areas are amplifying of our DNA sample. And then we go into the second round of PCR, which is dual indexing. And this is basically where we just attach a unique combination of base pairs to the DNA samples that allow us to recognize them during sequencing. And then finally, we prepare the library by pooling all the samples together and cleaning it up. And then we send it off to the USDA for sequencing. And what we get back is raw data that we can use for bioinformatics. And this is kind of what that looks like. So moving on to my huaka'i. This is an example of a fenced and unfenced plot from the study. You can see there's clear indications of rod, and there's a lot more invasive species in the unfenced site. We see a lot of ginger here. So that's that. And then these are a few pictures from my summer. This is one of our sites that was completely taken over by Himalayan raspberry. This is one of the field teams that I led this summer, collecting our eDNA samples. This is one of the lab teams I worked with, doing our eDNA filtration. This is me doing phenol chloroform extractions. This is a picture of the gel that we ran that confirmed to us that the correct regions of the DNA were being amplified by the PCR. And this is me with 3 PCR reactions running behind me. So getting back to our hypotheses from the Hua section, I don't know if you can tell, but it takes a really long time to do all of this processing to these samples. So unfortunately, none of the samples from this summer have made it all the way to the bioinformatics stage, and only a small subset of the 2024 samples from these plots is ready to be bioinformatically looked at. So unfortunately, we can't answer our bigger overarching hypotheses that I introduced in the beginning yet. However, eventually, once all that data comes back, we'll be able to get an answer for that. However, I do have a couple of things to share today, just from the small subset of data that has been processed. So yeah, this first one is just from the processed ones so far. There is a number of samples, which is just the number of bags of each plant that was collected in both the fenced and the unfenced sites. So as we can see, the canopy disturbance due to rod has altered the plant community composition. we see a lot more grass and a lot more uluhe inside of our unfenced sites as compared to our fenced sites where those species are almost non-existent. And then this here is a heat map. It looks a little scary, but here on the x-axis, it's showing the arthropods' taxonomic order. And then here on the y, it's the plant substrate. And basically, the lighter in color the box, the more sequencing reads of that arthropod order we found on the given plant. So, for example, this yellow one means that in unfenced Ohelo plants, we found a lot of Hemiptera. And this green one here is telling us that on fenced Hapu'u, we saw a lot of Diptera. And this is basically showing us that the unfenced plots have a dominance by generalist or airborne taxa, like Hemiptera and Trombidiformes, whereas the fenced plots are hosting more soil or plant-associated groups like Coleoptera and Diptera. One interesting thing we found, those unfenced sites where there was a lot of Ohelo and we saw a lot of Hemiptera in that last graph, that Hemiptera was Pealeus mori, which is a pest, and it's never been seen before on any member of the Ohelo family, so that was really interesting. Pealeus mori is in the Hemiptera order, as I mentioned, and it sucks the sap of the plant, causing damage. So some of my accomplishments this summer, I learned how to collect new types of data, like eDNA and traditional entomological sampling, which I'll get to at the end. I also learned new skills in the lab, such as how to do phenol chloroform extractions, how to use qubit, how to do bead cleanup and tape station checks. And I strengthened a lot of skills I'd been previously introduced to, such as PCR and gel electrophoresis. I also led field teams to collect my data, and I provided my plant expertise to other field teams working on other projects. One big challenge that we had with this data is that some of the DNA reads cannot be determined down to a genus or species level, which makes it harder to determine the ecology of some of the species that we detected. So for example, we found this one beetle that's in a genus that has a couple of endemic species for Hawaii, but also a lot of introduced species. So it's much harder to tell what role that beetle is actually having in the forest if we don't know if it's native or introduced, that specific species. So in order to remedy that, the lab's planning to run the samples again using a different primer to help confirm the results and get a bit more of that taxonomic resolution. Okay, and getting into the importance of this project, it's important for a few different reasons. The first being that we expect this study to be further evidence that ungulate removal and fencing helps protect forest communities and keep forests intact when it comes to rod. And another is it's really useful insights for outplanting and restoration, because if we know which of our native species, our native plant species, is holding the most abundance of our native arthropod species, then we know which species to outplant and add to our restoration initiatives. Also, it's really important to link the species interaction networks between species, because sometimes these networks and these associations disappear before those species do. So it kind of serves as an early warning system to map the interactions and see if they're still occurring in our disturbed ecosystems. And finally, this study provides us with really good baseline data of what species are present in our forests right now that we can look back on in the future and compare over time. And then my reflection about this summer, it gave me the opportunity to learn and strengthen so many skills that I can use to serve Aina as I begin my career. I also grew immensely in a professional capacity as I led my own field teams, and my professional expertise in plant stuff was utilized several times during the summer. I also, in a personal light, I want to become someone who is always deepening my connection to Aina and caring for Aina in any way that I can. And this summer just gave me so many opportunities to do that. On top of all the research skills that I gained, I also got to do all the PIPES programming, which allowed me to deepen my connections with places that I've visited my entire life. And and it just gave me so many opportunities to do the work to help Aina thrive, which was awesome. And yeah, this is the kind of work that I want to dedicate my life to, so I'm really grateful for this opportunity from PIPES. Yeah, other projects that I worked on this summer. This was one of the field teams up in Laupahoehoe Forest for a different project that I helped out with for a few weeks. I did a lot of vegetation surveys for them, so that's just kind of what that data looks like. And they taught me some more traditional entomological sampling techniques, because eDNA is not that traditional. So I learned vegetation beating, how to set up ground malaise traps and canopy malaise traps. And yeah, with that, mahalo nui loa to my friends and family, the PIPES crew, and the rest of my cohort. I appreciate you guys so much. And mahalo nui loa to all of my sponsors.
Awesome job, Rosemary. Do we have any questions?
Do you guys have any predictions for which native species is holding like the most arthropod diversity?
Yeah, which native plant species? I think we found a lot of stuff on brown hapu'u. So I would assume probably brown hapu'u holds a lot of our arthropod diversity, and also that Ohelo had a lot of different reads on it. So maybe Ohelo as well. Yeah.
Have you found like over the summer that eDNA is effective, or more so the other traditional methods that you did with the other crew?
That's a good
collecting
question.
Species and
Yeah, since I was so this summer I did only the collection aspect pretty much. I mean, I did other parts, but as you could see, not a lot of the samples made it to the final stage where we actually get to see, you know, what's being like how things are affected and what is actually present. So it's pretty hard to answer that question. I would say that I like eDNA because it means you're just collecting leaves. You don't have to hurt the plant. You don't have to kill any bugs. Like, it's pretty non-invasive, which is really awesome. But I think that you just kind of get different information from different samplings. Yeah.
I think we have time for one more question. Is there anybody in the crowd, online? No? Okay, can we get a round of applause for Rosemary? Up next, we have Kyle Kashner, who is with BISC, or the Big Island Invasive Species Committee. Can we give a round of applause?
Aloha. How you figure this out? Oh, I see it. All right, so did I do it wrong? Oh, hello, Darcy. Yes.
You did so awesome.
Aloha again. My name is Kyle Kashner, and I'm part of the Ka'au pathway. And I hope you all like entomology because we're doing another bug presentation back to back. This time in more of the traditional sense with coconut rhinoceros beetle and Japanese beetle detection on Hawai'i Island for the Big Island Invasive Species Committee. I'll first start off by introducing my host organization and then myself at the end of the hua. But again, I work for BISC, which is one of many invasive species committees throughout the state with like MISC and OISC and KISC representing the other islands, united under the Hawaii Invasive Species Council, which is a product of the Division of Forestry and Wildlife. And specifically, I had helped them detect the coconut rhinoceros beetle, which is native to the southern Asia area and is a very serious pest of palm trees, not just coconut, but also like triangle palm, and then also other monocots such as hala. These insects are very capable of very easily killing their host plant with just a few feeding events. And then in addition to the coconut rhinoceros beetle is the Japanese beetle, which is not yet established in the state, however is intercepted frequently. It's mostly in the continental US and is a pest of more than 500 plant species. It's not yet been introduced to the tropics, so we expect it could be very detrimental to our plants here. Regarding bisque, they used to be the Melastome Action Committee in the 1990s before they expanded off of just Myconia, I mean that's Clademia, off of Myconia to a lot of other non-native plants and animals. And then from there, they've started services such as Plant Pono, which just enforces best management practices on nurseries for not harboring invasive species. And then their invasive plant control program, which my mentors focus in more on. They've successfully eradicated a handful of unwanted plants from the island. And then finally, the POEM program, or Port of Entry Monitoring, which specifically looks at trying to track non-native insects that arrive in our airports and harbors. Regarding the coconut rhinoceros beetle, which has been in the state for a few years, but has only recently reached this island in October of 2023. BISC has been employing several methods to monitor and control the insect. Their most primary method being installing and checking these big black panel traps that you've probably seen hung up on like a fence or a tree around the island. They primarily just consist of a lure that the beetle flies towards when it is active at like twilight or at night. And then trying to access it, they'll collide with the panels and then fall into a little bucket that is too narrow for them to escape from. And regarding the invasion, when I first joined the BISC this summer, there have only been 15 detections of the insect in Kona area and then 6 in Waikoloa Village. However, the insect has not been seen in Waikoloa Village since September of 2024, likely mean it's exterminated locally. However, old damage to triangle palm trees can still be seen. And then just finally about myself. So I was born on Maui Island, but I've been since raised and lived my entire life in Waikoloa Village on the west side of this island. I guess my journey with just nature and respecting the Aina first started when my parents started bringing me to just tide pools and like native forests just to observe and explore. And then from there, I started doing photography to upload to iNaturalist, which is a just community science like observation platform where I can interact with professionals and they can tell me what the species is. But from there, it was not until 2024 that I had first met my mentor prior to PIPES, when Molly Murphy had collaborated with me, assisted me, to collect a specimen of the rice flat sedge, which was a state record plant that I had found in the UH Hilo campus. And then from there, Molly requested that I join PIPES. I didn't really know what PIPES was before then, so I was like, sure, why not? I mean, even from back then, I did have an interest for working with BISC, because I didn't really want to restrict myself to just one like native space just to protect. I wanted it to be island-wide, and I specifically want this to protect this island because I was raised on this island. So I think with an insect focus, BISC is right for me, and well, I guess I'm working for them now. Regarding my hotline, most of my assignments for BISC over this summer have mostly been what I mentioned previously, just their CRB mitigation and monitoring tasks. However, there's a few at the end that I've been doing that's more unique to myself. So again, the main thing is just checking the CRB traps, but also cleaning them and determining whether the caught beetles are male or female. So there's just me checking the traps all over the west side. And then there's one male beetle. I should have brought a specimen, but I mean, they're like maybe this big, and with their wingspan, maybe like this wide. They're fairly large. I mean, the Japanese beetle's like not as, we didn't see one. And then after that, we went to Kona and then Waikoloa to look for tree damage, with that mainly being V-shaped cuts in the fronds, as you can see here. Boreholes would also exist, but that cannot really be observed easily unless you're like a tree trimmer with like one of those bucket trucks inside the tree. And in addition to the V-shaped cuts, there would also be scalloped edges. This is caused by the beetle. When it first finds the tree, it will bore into the base of the frond to feed on the sap inside of the heart for 3 to 4 days. And then it takes like 5 months until this damage is realized. So there is a bit of a delay for using this 2-track CRB. However, it's very useful for Waikoloa, where that's where the species was first found. And then it also helps where we don't have traps. After that, we also dig through host material. So the CRB, when they lay their eggs, they'll lay their eggs. If they don't find like a rotting dead palm tree, they'll find like a mulch pile or a pile of compost or even one of those drainage stalks. And then so when we find a CRB somewhere, we usually check nearby host material to see if there's larvae. And if there's a scarab beetle larvae of any kind, such as the oriental flower beetle, which, I mean, it's a little gross here, so let's do the Chinese rose beetle. If we find one of any kind, we will treat the material by putting a net over it, which does not prevent the insects from developing, but when they come out or when new ones come in as adults, they get entangled and we can collect it from there. Regarding Japanese beetle traps, we use products from Trees, which is a biotechnology company based in Oklahoma. It follows a similar design with a bucket and a panel and a lure, using a different chemical, a chiromone and a pheromone specific to the species. We have these all in our ports of entry on the island, so including the Hilo Airport and the Kona Airport, but also like Hawaii Harbor. And then moving on to some of my more personal unique tasks, I'll be making insect collection, or I forgot I already did all these. I've made 3 insect collections, one representing the bycatch from the CRB and JB traps, because that is not, BISC has not been looking at their bycatch prior, and they want to know what non-target effects they're having. And then after that, I've made 2 collections representing nocturnal species from Waikoloa Village. And because I live there, I can just attract them by putting a UV light at night, displaying it on a reflective white sheet, and I get to observe species such as the oleander hawk moth. And then just really short, I've also written some insect pest reports just for like local readers of how to identify and manage pest species here, mostly using online resources, but also my own observations. But a lot of species are not well studied, so it's difficult for some groups. Regarding my huaka'i, I've definitely tried to make this personal, and BISC has introduced me to many like landscapers and other departments and organizations. I'm very thankful for that. We've been sharing our observations back and forth. But first and foremost, I've helped them illustrate the current CRB range on this island. So this is about every detection that we've seen so far after I joined, so roughly like May 25. Their primary population remains like the airport and like Host Park Nelha area. However, they have started moving east, so towards Palamanui Community College, but also like Kona Palisades area. And then it's a little laggy because I did the circles by hand. They're in the like Kukio area, and then also there was a detection like Kiholo, and then the West Hawaii Veteran Center. However, they've not really been moving south, which is good because we don't want things going south here. In the Palamanui campus, we were able to treat 2 mulch piles with nettings. With what suspect grubs we found, we gave some of them from the first pile we gave to Natalie Graham in the Alala Biodiversity Lab. Cool connection. And then also, hopefully soon, the ones from the second mulch pile will be identified by the community college itself. once they have the means to do that. And while not directly handled by BISC, while PIPES was in HCC, the Department of Transportation had found a breeding site containing more than, at least about 110 larvae. Most of them look stage 3, so likely it was not long before they became adults. And BISC probably assisted indirectly in the sense that we kept finding adult CRB near where they dug these up, and this is a large step. I mean, I don't know how big of a step considering that these insects do lay a lot of eggs, but I think it is a meaningful step as with how early the invasion is to eradicating the species. Regarding tree damage, we've not seen any new tree damage, only old palm damage, like V-shaped cuts on Triangle Palm in Waikolo Village, which is good. We've not seen any Japanese beetles, only mango flower beetle and oriental flower beetle, but also Chinese rose beetle caught in our Japanese beetle traps. However, regarding the Japanese beetle traps, I believe I've made a significant change to how they will be managing it in the future, given that on June 20th, I had found 3 individuals of the anthracite yellow-faced bee, which is a critically endangered species, Hylaeus anthracinus. I did not think much of them at first. I just thought they were strange small bees, but after inspecting them for longer, I realized they could potentially be this species, and then after communicating with my host organization, I was able to get the species confirmed, and because this is a endangered species, we want to mitigate negative impacts to them, so we had to end up taking down most of the Japanese beetle traps. However, at the same time, the Division of Forestry and Wildlife reported that because these bees were found in the airport area, this may represent a previously unknown population of the Hylaeus anthracinus, and they will be searching the area to see if they can find that new population. So regarding the changes, all of the traps in Kona Airport area, but also all of those on Maui Island had to be removed because of just close proximity to known populations. However, all of those that remain statewide, they can stay, but they just have to be switched out with lids that are not yellow, and they have to have their floral chemical removed from the lure, as these are 2 elements that do attract native bees, but also honeybees. And BISC has seen a lot of honeybees in the past, like almost half of the bucket is just honeybees. So I can see why they've not found this in the past, if this has been the case in the past, just because there's so many honeybees, you wouldn't know that there's these very small bees aside with them. Regarding my bycatch trap collections, I've noticed a different set of species between the 2 traps because of their lures. Probably the most interesting find is a longhorn beetle that was unknown to both BISC and the state pest hotline, and we've since sent it to the state entomologist for identification. That is pending. And then for the Waikoloa Village collection, I've mostly filled it with non-native moths and scarab beetles. However, if I were to continue, I would probably start getting native species just because there's so many non-native species, but at the same time, there's such a large diversity that it's almost just nice to look at that we have so much life in a very residential and dry, like pastureland area. And then finally, I wrote 4 pest reports. They will be put on BISC's public website. After working for BISC, I realized that I had actually been pretty well prepared to handle a lot of these tasks, just because I've been pinning and collecting and identifying insects, even taking photos as early as 2020. However, I still was introduced to many unique spaces and introduced to many organizations that I would not have been exposed to otherwise, just because I'm kind of a shy person. However, regarding the community, I definitely think I've helped BISC understand the diversity of insects affected by their poem program. And aside from the one native bee whose negative impacts can be easily mitigated just by switching out the traps. Our lures do not negatively affect invasive or native species. And then I also realized that not many people have actually seen a CRB in person. I realized, again, I should have brought one to show off. But because of that, the invasion on this island is kind of invisible unless you're really actively hearing about it. Tree damage is very hard to find currently. And just by explaining the situation to them, I help them realize that this is going on and there's ways that they can help. And then just here's just a spider wasp that I found in the Japanese beetle traps. Pretty large, just a pretty nice find. If you have like cane spiders, then you probably want to have these around because they will hunt those. Regarding next steps, Bisc had originally wanted me to do a lot more plant-related tasks, such as find Crofton weed in Pu'uvava, because I'm a naturalist, I guess I was the first one to find that there. Sorry, guys. But due to time and just the urgency of the CRB invasion, we kind of had to cut that. However, when I go back to college, I will be helping them CRB again, and then after I graduate, I'll most likely be putting on some plant-related tasks. I'm still like an amateur at identifying insects, but I do hope that soon I'll become that kupuna who knows how to identify everything that we have here. Regarding like the community, I feel like there was a little bit of a lack of active communication between the biosecurity agencies we've had here, because while the DLNR did suggest the mitigation methods for protecting the bees, they've known this at least since 2019, at least with the article they've provided us, meaning that this whole incident could have been prevented if we've just been more in touch. I know it's a little bit of, authority-wise, there's a little bit of a distance. However, I believe that even if we were communicating more with like the biosecurity in other states, such as Oregon, where this practice of using the better kind traps is already used at the moment, I feel like we could have been in a better spot. But because it did happen, I believe the next intern will most likely be continuing my bycatch box as well as expanding the detection map that I had helped create. And then finally, I believe that community connection is a key component, given that on Maui, they've actually eradicated their CRB because a tree trimmer had found the first breeding site in a dying tree or in a dead decaying tree. And I guess that was the only one because they haven't seen any since. So if we continue to involve ourselves closely with like landscapers and tree trimmers and mulch processors, et cetera, I believe that we'll be heading in a similar direction where we can protect our NEO. And then there's us at HCC. I'd like to thank PIPES and my host organization as well, and my funders. And then at the end, I have just a timeline if you want to see that on your own time, of just how CRB has advanced on Hawai'i Island throughout when it first came to July. Mahalo.
Kyle, stay up there, Kyle. Hopefully we'll have some questions for you and you can answer them. Do we have any questions for Kyle? By the way, we are happy that you are a part of PIPES. Thank you, Molly and Franny, for encouraging him to join. Do we have any questions? Maybe we could take one question. One question? One question? Oh, okay, just yell it out, please, you two.
What is your most favorite insect that you caught?
That I caught? I would probably have to be that wasp because it's just so pretty, and I've been looking to find one for years, but they're pretty uncommon, and just to find one dead in the Japanese beetle trap was very lucky and very surprising.
Thank you, Kyle. Up next, our last presentation before a little break is Gabrielle Shapiro with Uncle's Kokua Center and Kaimu Kalapana Puna.
Aloha mai kako. My name is Gabrielle, but you can call me Ella, and I was born and raised in Puna, Hawaii, and am a first-year PIPES intern in the Kaiaulu pathway. I am a junior at UH Manoa and have a double major in Hawaiian Studies with a Malama Aina focus, as well as botany. And this is my presentation, I Kawa Ma Mua Kawa Ma Hope, Preserving Oiwi Ancestral Knowledge Foundational to Climate Adaptation and Resiliency in Puna. Wrong thing. So, my hua, my home in Lower Puna has shaped every part of who I am. I grew up near Kumukahi, the easternmost tip of Kapai Aina o Hawaii, and from a young age, developed a deep pilina to this Aina and its elements. The rains of Moaniani Lehua, Polohinano and Ava Ava, the oceans of Kumukahi, Kapoho, and Ha'ena, and the forests of Hi'iaka. Being raised in a rural place where community and Aina are closely intertwined, I have always seen how Aina nourishes us and how we, in turn, have kuleana to care for Aina. It was in the lush forests of Lower Puna where I first realized my deep passion for Aloha Aina, which has only grown since I've gotten older, and this journey has truly been a focal point. Now to introduce my project or my site. So I had the absolute honor of working with my incredible mentor, Kaini Pu'uwai Keli'i Ho'omalu Holtz. She is a UH Hilo grad student in the TCBS program, and her thesis is entitled Articulating Community Resilience and Climate Adaptation Through Oiwi Ancestral Knowledge in Kaimu, Hawaii. And she does this through 3 decolonized methodologies: oral histories, archival research, and community engagement. And as quoted in her proposal, the outcomes of this work aim to inform both academic and community audiences by highlighting the continued relevance and innovation of Oiwi ancestral knowledge in contemporary climate resilience, conservation, and food systems. My host site is Uncle's Kokua Center. Our mission is to support the continuing good that Uncle Robert's Ohana provides to the Puna community. Along with this, they support the farmers market, Hawaiian sovereignty and Hawaiian culture, regenerative agriculture, wellness, education, and economical development. This is all thanks to the dedicated members of the Keli'i Ho'omalu Ohana and the community who have built and sustained a foundation of resilience and cultural continuity. Their work honors the legacy of their kupuna, Robert Keli'i Ho'omalu, a man of aloha who envisioned a brighter future for his ohana and community. He was dedicated to sustaining, uplifting, and supporting community programs, cultural practices, local food systems, and overall ea. And in addition to Tuji girl Keli'i Ho'omalu, who was a renowned songwriter, singer, and musician, who is also fiercely dedicated to her kaiulu and ohana. A quote from the Keli'i Ho'omalu Ohana reads, "Robert was more than the patriarch of our ohana. He was a pillar of this aina and ahupua'a. He welcomed all who entered his home with open arms, only asking for respect and love in return. As his family, we are honored to carry forward his legacy, cherishing the values he instilled and the love that he shared." This is him and Tutu G Girl. And then these are their 10 beautiful children. The Kaimu community remains central to this work and have continued the incredible legacy of Robert Keli'i Ho'omalu. And they do this through key efforts like native plant restoration, oral history and mo'olelo documentation, and youth engagement and education. Thanks to the support of my mentor and her ohana, I was inspired to come up with my main question for this summer, which is the following. And through this question, I seek to answer or to show how Ike Kupuna has fostered community resilience, abundance, and stewardship in Kaimu. I have focused specifically on how the new Aina of Kaimu and how the Ohana and Kaiulu have adapted their agricultural practices in response to the creation of this new land that was created in the 1990 Kalapana lava flow, for those that don't know. At the heart of my commitment to this project is the kuleana I feel to help preserve Ike Kupuna, ensuring these mo'olelo and ancestral ways of knowing and living continue to thrive across the generations. I feel deeply grateful to contribute to a project rooted in honoring memory, uplifting resilience, and advancing movements for food sovereignty and Oiwi land stewardship. This summer has highlighted for me that every step guided by ancestral knowledge brings us closer to true abundance and resilience in our communities and Aina. So on to my ha'alele. This summer, we are utilizing two main methodologies to gather information for our project. The first centers on interviews and oral histories. So first, there is a waiver that we have our participants go through, which just kind of goes through what our project is and why we're doing it, and then also a brief demographic survey just to collect relevant information. We have started on what will be 10 interviews with the keiki of Robert Keli'i Ho'omalu and 5 oral histories with kupuna from the area. And supportive tools that we have been using is Otter AI, audio recorders along with personal devices, obviously our handy-dandy MacBooks, and Canyon cameras to take pictures along the way. And after each interview, we review the Otter AI transcripts and listen to the audio to make necessary edits to ensure accuracy, as we understand the ethical responsibility we have in preserving this sacred Ike. Another big part of this was familiarizing ourselves with the 11 core questions. So my mentor thoughtfully developed 11 questions and additional follow-ups to help deepen each mo'olelo while still maintaining a comfortable and talk story feel. Topics include things ranging from personal connection to kaimu, traditional land use, adaptations to lava flows, and further aspirations for what they hope the community and Aina will look like. So these are the questions. And then, I lost my mouse. Okay, sorry. And then the second methodology is pertaining to my specific focus, which is planting native species and conducting vegetative surveys. So the Ohana of Kaimu has long relied on Oiwi ancestral knowledge to guide their responses to environmental and climate shifts, especially living in Puna where Pele is an active presence in all our lives. And so with respect for these traditions, we aim to continue restoring native species along the coast and across and assess their health and distribution through surveys. So this project was inspired by our heartfelt interview with Auntie Lohelani Keli'i Ho'omalu, shout out, and where she expressed her hope in restoring the Aina from Mauka to Makai. This is a quote from our interview with her, and I really wanted to highlight the last part where she talks about how the revitalization of these native plants will help to create spaces for use culturally and also help carry on the legacy of her father. The first part of starting this project was first learning these traditional practices from both my mentor and her ohana, and honestly just talking story. And then we also spent a lot of time developing our transect methodologies. This summer, one of the main things we did were these vegetative surveys to help create a guide for the community and the ohana of what is already there, what can be utilized, and also to help aid in eradication and restoration efforts. So this is our plot. As you can see, we're doing surveys on the south side of the Kaimu Beach Trail. So starting in the 80s, the Kalapana lava flow happened, and it ended in the 90s. And as you can see, it added a lot onto the coast, all that lava. And so we're surveying 400 meters, or we surveyed 400 meters on the south side, and our transects went out 30 meters horizontally, because this is where the majority of outplanting occurred, and then we went 5 meters in between the transects so that we could easily look at the different species in between, and we're only recording the presence of native and non-native, non-vascular arborescent species because they seem to have the most impact on the ecology as well as the community as a whole. And these are some of the information that we were gathered. I especially wanted to highlight the status and presence. We came up with these because we came up with these 3 statuses because indigenous, we wanted to encompass anything from endemic plants to even canoe plants that have been utilized through generations. introduced encompasses plants that are not from here, but they're not necessarily bad and can be utilized, whether that's for food and medicine or even maybe housing materials. And then invasives, obviously, boo, we don't like invasives. And then presents naturally growing or planted just because we wanted to see what naturally grows out there, what should be taken out, and what species we should plant more of that do great there. So preparations, as I said before, major preparations, we're just learning directly from my mentor and her family. I was very fortunate to have my mentor along with me throughout the way, and she was very organized and guided me throughout this entire process. And also just having hands-on experience, talking story, we created a mala within. I think that was my first day, and that was just a really good way to just get in there. And then goals and outcomes. So a major goal of our project is to preserve, apply, and uplift Ike Kupuna to support the abundance and resilience of Kaimu's Aina and community with a focus on how environmental resilience is rooted in Hawaiian knowledge systems. My specific role is to continue the Ohana's work by restoring native coastal species, creating mala, planting trees in the lava, and conducting vegetative surveys to document how Ike Kupuna is transforming the land. Through this work, I hope to honor my mentor, her Ohana, and the Aina of Kaimu by giving back to place and people who have deeply shaped it. So, for my huaka'i, in case you didn't know, this is where Kaimu Kalapana is. It's in Lower Puna, and these were our 3 main sites. So up Mauka is where we do interviews and work on the property or help care for the baby plants in the nursery. This is where we conducted our outplanting and veg surveys, and then more educational outreach and outplanting happened out by the ocean. So for our interviews, we really made sure to structure our interviews around the schedule and availability, as long as the comfort of the people that we were interviewing. They typically lasted 1 to 3 hours with oral histories, taking anywhere from like 2 to 5 hours, spanning a few days. They were less structured and more just like, tell me about your life story. And these interviews maintained a really talk story feel. We were constantly laughing and smiling along the way, and it was really inspiring and heartwarming to hear my mentor and her ohana recall fond memories of growing up together in Kaimu. These sessions always ended with a deep sense of gratitude for both the Ike shared and the people who shared it. And these mo'olelo truly carry a wealth of Ike that is essential to cultivating and sustaining a thriving Kaiaulu. So once we were finished, we edited the transcripts and added necessary contextual notes where we thought was necessary, which helps to not only compile, but also to accurately preserve what's being shared and to understand it. So key themes that we identified during this process are the following, with special highlight on Kaimu as a gathering place. Even after the lava flow, the community and ohana continue to be there, and it continues to be a hub and a place of abundance and aloha. So these stories ultimately affirm the enduring strength of place-based Ike, offering powerful models of resilience and cultural continuity. Preserving and sharing this Ike looks like this. So we plan to create a comprehensive document or online platforms with the photos from the interviews as well as the audio and the full transcriptions. And we will have these password protected for the ohana only and for the larger community with the consent of the ohana. And along with this, we hope to recreate storyboards that actually were previously up thanks to my mentor's papa who created them. So we hope to recreate these and recarry, the vision that he had for Kaimu. And along with this, my mentor also eventually maybe hopes to write a book. Yeah. Okay, so now for the second part is the implementation of this Ike Kupuna and the continued resilience for future and present generations. So it comprised of the 4 following hana. The first was just ongoing property work and nursery care. So like I said, the little species, this was the first day making our mala. And then that's just us, in the mulch, that good puna mulch. And then there's outplanting. So outplanting is a key way that we turned our data analysis into tangible actions, contributing to the ongoing legacy of resilience and abundance in Kaimu. This work isn't just about restoring place, but the continued revitalization of these practices. It's also about stepping into our roles as the next generation that holds the kuleana to malama, these vahipana, as we are continuing the hard work of my mentors Makua and Kupuna that came before us. By continuing to re-implement these indigenous knowledge systems, we are supporting not only ecological and cultural resilience, but also long-term food sovereignty and community well-being. So going on to how we outplanted, these are our methods. So first, you're gonna wanna find a puka. So that's us. Then we added a rock barrier to make sure all the mulch and obviously the plants can stay in there, the most important part. Then we weeded in and around it to, get that leaf litter back into the environment. Then we added our mulch and planted our little plant. I believe this is a kolea, and, making sure to be gentle. Then we added some more mulch and more water, and then boom, you have a plant. Or in this case, you have three baby plants. And so the third aspect was our veg surveys, which I've talked a lot about. Overall, we covered over 12,000 square meters, which is nearly 3 acres. And this was some graphs I created with our findings. So the first there was 1,159 plants that we saw within that area, comprising of 50 species. So of these species, 20 were invasive, 19 indigenous, and the rest were introduced. And as you can see, the majority of them were planted. The ones that were naturally present are obviously invasives, because who would plant those besides? We're not going to talk about the dragon fruit. But also, it was really cool to see how all the ohia that were on the lava were naturally occurring, so that was really cool. And then also, the noni and niu were doing so good out there that some of them reproduced, which is really cool, so maybe plant more of those. And then also, eventually, I hope to keep compiling this data into a Google Sheet or like some sort of pamphlet to compile an inventory of these species, their uses, and just information about them. Lastly, oh yes, and then, sorry, and then this is another, this was like all of the 50 species. As you can see, we had so many new. And then this is another graph of the 1,000 plus species, what percentages were indigenous, invasive, or introduced. Lastly, educational outreach. So this is from one of the groups we hosted, Halau Kulau Lama, which is a summer program through Kamehameha schools, and they went down with Kumule Ilai Kamamahu. And so here is pictures of us throughout the day. So together we planted over 100 native species, and it was truly amazing to watch the students grow more engaged as the day went on. They were definitely really shy at first, but it was really inspiring to see how they lit up, and even some of them asked to come back like the next day, and we're like, sure. So it was really cool. And also many of them had been to Kaimu before, but didn't realize the deeper connection to the place. They've been for concerts and markets, but didn't realize that it's the home of the Keli'i Ho'omalu Hana and the product of generations of Hana and history. So them realizing this was also really cool to see. And just seeing them want to give back and contribute to this Vahipana was incredibly heartwarming. And the trees they planted now stand as a living legacy that will nourish future generations and maybe even their own keiki and mo'opuna one day, which is really cool to think about. Okay, so on to my ho'ina and haina. Sorry, I know we want to take a break. But as this 10 weeks pipe journey comes to a close, I leave this summer with a deep sense of gratitude, pilina, and purpose. The culmination of Hana from our individual work to the huaka'i we've been fortunate to attend has truly been life-changing. I feel incredibly honored to have contributed to the ongoing work in Kaimu. We realize the significance, importance of preserving the stories of our kupuna as the next generation rises. These experiences grounded me in kilo, place-based learning, and the ongoing relevance of Ike kupuna and climate resilience. This summer has taught me that we must always look to the past and to our kupuna to guide our way forward. And my kuleana to protect and uplift Ike Hawaii is now a lifelong commitment. This internship truly reaffirmed to me that conservation cannot be separate from culture, community, or history, and how Ike kupuna and relationships to place are essential to long-term environmental and food system resilience. Kaimu, rooted in ancestral knowledge, can inspire broader restoration efforts across Puna and beyond. In fact, I myself have been deeply moved by the care and intention the Ohana puts into their aina, something that I hope to implement in my own life because the fight for food sovereignty begins at home. Rebuilding our pilina to aina, kaiulu, and food systems is vital, especially in places like Puna, where new land from Pele invites us to imagine abundance in places that at first glance might seem desolate. This experience clarified my kuleana and helped me envision the kind of kupuna I want to become, one who knows and nourishes her aina, honors Oiwi ancestral knowledge, and helps build a thriving future for the next generation. This isn't just one summer of work, but this internship has been a continuation of generations of aloha aina and dedication, and I'm just truly honored that I got to be a part of it. So big mahalos to my mentor and her amazing ohana who really treated me as their own this summer. Also to the amazing PIPES crew, the staff, and the cohort. We had a really fun time together. And of course, all the sponsors and funders, mahalo.
Do we have any questions for Ella? There's gotta be a couple out there. So continue to make eye contact.
Awesome job, Ella.
Thank you.
And I love all the photos. My question is, it looks like there's a lot of outplanting efforts in Kaimu, but how do you see the invasive removal efforts in that space going?
Yeah, I definitely was super surprised at how much invasive species there was on the lava, especially, what was it, Plukia. Oh, it was all over. And so definitely it's a lot more is able to grow on the lava than we think. And since it's I guess easier to control than a forest, which has abundant soil and all this, I think that eradication efforts can honestly I feel not be more impactful, but once they're removed, hopefully they can be gone for good and less spread. Wait, sorry, what was the first part of the question? I got distracted by the Plukia.
Do you have any strategies or things
Oh,
you
strategies.
recommend?
Yeah. So just getting in there, we surveyed all the ones that are out there on the right side. So just getting back there and it's really hard because some of them are lodged in the lava. So I don't even know. Maybe coming with OO bars and just digging them out. Yeah.
It's a great strategy. Do we have one more question for Ella? One more. No? Okay, can we get a round of applause for... Oh. Kiana's gonna throw up the agenda. We have a 15-minute break right now. Well, now it's 10 minutes, so I'm gonna ask everybody to be back here in 10:33 so that we can get started at 10:35. Interns, stay here for now. I need to call a couple of you to the front of the stage. Check, check, check. Okay, guys, can we start making our ways back to our seats? We're going to get started in a couple of minutes. Okay, as we're kind of settling in here, I'll introduce our next presentation. Can we get a round of applause for Seth Asano, who worked with Symbrosia this year? And just a little caveat before Seth starts, we have a couple of Olelo Hawai'i, or fully immersive Hawaiian language presentations in this next couple of rounds, so please bear with us. Thank you.
Okay. Okay, aloha everybody. My name is Seth Asano. Okay, yeah. So my title of my presentation is Ho'okohu Kohu e ka Limo Kohu, Restoration Efforts of Limo Kohu. I'm a new PIPES intern. I was working at Symbrosia down by OTEC in the Kona Airport area. Just to start, this is the Ho'okohu Kohu e ka Limo Kohu is from the song Kaulu Vehi o Kekai. Talks about the limu and the collection, just how pretty it can be. And to hope everybody had a good break. So to get back into the space, we'll start with a joke. We'll see, we'll see if it hits. It is a dad joke. I'm not a dad, so we'll see if it qualifies. Okay. So what did the limu kohu say to the other limu kohu? It actually didn't say anything, because if you didn't know, limu kohu don't have voices or mouths. So if you're expecting them to say something, might need to look again. But it's important, just because they do not have a voice, we need to make a way for them to speak for them and make a way for people to know about what needs to be done for them, especially for like other things we do in conservation. And so starting with the hua, we kind of want to talk about the life history of limu kohu, just because it's very different compared to other species like land animals and plants and everything. So what's special about the limu kohu is that it has a triphasic life history. So meaning that there's 3 phases in its life, 2 different morphologies, multicellular looks. So in this bottom part of it is the tetrasporophyte stage. This is interesting just because in the early stages of identifying these limu kohu, they actually thought it was a total separate species. They called it Falkenbergii hillenbrandii, but further research was done to actually see that it's actually a part of the limu kohu that we see in the wild. So this step kind of plays into my project. And so going into the tetrasporophyte stage, they can be found in the wild, but it's very different. It's very difficult to spot. You'll be lucky enough to see patches like these out in the wild. But if you do find them, they're usually attached to different substrates, other limu, and they kind of have like this cotton ball kind of look. And what Symbrosia does is they collect these and kind of, I'll go over the processes, but they do not heavily rely on wild harvest. And from that, they focus on the bromoform growth and just creating more cultures of these and kind of go into it. So when they collect it out in the wild, this is a microscope view of the limu kohu in that stage. And what they will do is they'll collect or cut off 2 to 5 cells in length of the limu kohu, And from there, we would grow it to back into its larger size, and we'll do it step by step until we get the full. We like to see like good color, good health, and good growth. And from there, we keep multiplying them into a larger culture. And this is important for Symbrosia, especially just because it has what is called bromoform. And this bromoform is a liquid chemical that inhibits the coagulamin-dependent methyltransfer step of methane formation. So this is important because these are greenhouse gases created by the cows from their burps and farts as well, but more of their burps. And using the limukohu in this stage and using that bromoform content kind of interrupts that and helps reduce the amount of methane reduced, and that's what's special about Symbrosia. And they would use this for both dairy and beef cows. A lot of partners with like Parker Ranch, and now going into different states in the continent. And from this, transferring to more of my project is mostly getting these tetrasporophytes to spore. And this is what you kind of see, these smaller, I guess you could call them like seeds. And from developing different photoperiods, water, ambient temperatures, salinity, health, we've been able to get them to spore more often rather than relying on wild sporing. And collecting them can be difficult just because they're smaller and it's not as easy to get them to spore in a lot of different scientific research. So it's great that Symbrosia, mainly Makana Heather, was able to figure out what parameters we can use to get them to spore and get more of these spores. So from these spores, they go into the gametophyte stage, and this stage is what we mostly see out in the wild and what we collect and what we normally see as limukohu. So transferring to a different multicellular stage and finding those, and this is what we mainly want to focus, just because it is kind of hard to come by if you already don't know where the spots are. And so, yeah. So we grabbed different methods of how to get these spores onto different substrates and outplant them. Cool enough, New Zealand, Aotearoa in Wellington, a group called Love Ilimunimo is focusing on giant kelp. And they basically do the same thing of outplanting these spores, but with a different type of limu, which is more of like a brown limu compared to limukohu, which is a red one. This is mainly to reproduce or maintain the habitat for food, protection, and other resources, especially due to the decline of sea bottom trawling, overfishing, pollution, changing temperature of the air and water, and unregulated predators. As you can see, this is using natural fibers. So they use the harakeke, muku, pinal. And it's a little bit easier with brown limu just because they have flagella or these type of fins where they can orientate themselves to light, temperature, but compared to the limukohu, they don't really have anything. So that's why it makes it a little bit more difficult compared to the giant kelps and browns. And so even just in general, red limu is really hard just because of this feature that they don't have. So fortunately enough, there was one other paper that is kind of focusing on what this project is about, selecting optimal substrates for ocean-based cultivation of red seaweed, Asparagopsis taxiformis. And it uses natural substrates like nylon, propylene, sisal, and cotton, and using kind of some of her methods gave us a way to kind of pursue this project. And it was really cool just because they use a binder as well. We didn't use a binder, but a binder is a way to get these spores just because they're really difficult to transfer. It's almost acting as like a glue to kind of stick them to these substrates so that when moving them around, they just don't fall off, especially if it takes a while for them to connect. Yeah. And so this is important just because it is like our native plants, animals, and vertebrates. It holds a special place culturally, ethically, environmentally, health. It's a food source and in practice, and also working with different fibers and different native plants. We also regain some of that knowledge of braiding practices and ocean restoration practices. And yeah, so this is the main focus of it, just getting back all that knowledge and replanting the wild population. It's important just because, like in the beginning I said, they don't have voices. And looking at like some of the DART regulations, there isn't a lot talking about limu in general. This is the only limu that they kind of focus on. So it would be awesome to see more limu representation throughout our ocean systems and the ecosystems as well. And just to see like how it can be misused. This is a Facebook marketplace. A full bag of limu kohu being sold, $80 per quart size Ziploc bag. So it's important to kind of regulate these things as we do other animals and things like that we want to conserve. So it's really important to give them a voice to be able to survive, conserve themselves. And so this project is under a grant, and we can see that there's a lot of different restoration efforts. Part of this grant is like the Waianae High School, Windward Community College, Symbrosia, some people in Maui, a lot in Oahu, and in Kauai. And with that comes our different natural substrates. So our control was our gore string. This is kind of the one we've been using just to test sporing, but we want to wean off of that just because it is plastic based, and we don't want to introduce more plastic into the ocean. And now we're worrying about like microplastics and things like that. So maybe trying different substrates that don't really harm our natural reefs. So we looked into coconut core. This was this little bit thicker in diameter, so we can't fit a lot of string onto these slides. It has a darker background, and you can buy it off of Amazon, so it's a little bit easier to work with. We have our howl, which is another natural substrate. It did need to be braided. Lucky enough, PIPES was able to give us like a full gallon bag of prepped howl, and I was able to braid it into what you see, this third one. And then our Milo strand, same process as the howl, but it also has like a more yellow and dark background, as we see. It's also used for dyeing, so a lot of the leakage of the yellow you can see further in my presentation. And then we use Lisa Raymond's YouTube video, trying to see how to prep these different barks, regain that knowledge, and be able to braid these and be useful. And then the Milo was collected in Mahiula, Kona, and then again the howl was collected from PIPES, and then both our gore string and coconut cores bought from Amazon and other places. And yeah, so some of the roadblocks that came into is that there isn't a binder compared to giant kelps and things like that. Our sporing for our tetraspores wasn't that great. These are all new tested substrates. We only had like matured germlings, lack of new spores. The different thicknesses of the substrates kind of made the data kind of hard to collect. We also focused on trying to get more quantitative data rather than just qualitative, and then also creating like a pioneering or foundational project so that other people can build off of this limu kohu restoration. Yeah. And so what we did, just because of the low sporing, we wanted to use a combination of both spores and already settled germlings. We call this a germling solution. It's also a solution for our problem, but also like a water solution. And so by using that, we were able to collect the water, pipette it into the different substrates, and get them on and see how well they do. And so heading into our huaka'i, some of these are pictures of the first week inoculation. So we have our Milo on the far left, and then our coconut core, howl, and then our last is our gore string. And so when looking at this, we want to take into account the color, how much is settled onto these substrates. This is our howl. You see that bright yellow. We see good color. Our Milo actually turned really dark, which is interesting when we autoclave them or clean them at high temperatures. And then we have our coconut core. We see a lot more retention just because they're more fibrous and can catch these different germlings. And then we also have our gore string. More white. You can actually see them way better. And they actually caught a lot of it, in which we've seen in previous studies. And then we also wanted to add one more substrate, which is our pihi shells. What we noticed is that with the pihi shells, it kind of depends on how steep they are. If they're more steep in angle, the bigger germlings tend to roll off and not really have a chance to settle. So we're trying to figure out more methodology to kind of attach them, especially without a binder. And this is just another picture of just the first week of inoculation. and then we go into the full first week, we see that the gore string, there's not much change, a lot of them attached. What I do was take it out the petri dish, shake it, and see what comes off and what is attached, and I'll count how much is attached. And then we go into our Milo, we see that a lot of the yellow dye is still into the bark, so it started to leak out all the yellow, and unfortunately, the water level did drop, so a lot of mold grew on top of the Milo, and then kind of affected the health. You can see that the limau kohu is more orange and more fluorescing. And then we go into our coconut core. You also see more of that fluorescing orange, but you do see some light pink, some red. Fortunately, I was enough to find just one little strand of limau kohu that was really good in color. Take note of this one because it survives. And then we have our how. Again, we see more of that orange fluorescing, but we did see at least one or two really good limau kohu surviving. And then we have lastly our opihi. We can see that they're much more smaller in size and they're actually attached to the opihi, which is great. But then you also see like this green bacteria growing on them as well, which is kind of a note. And then we go to our last week. So we have our Gore-Tex string. We see that they're growing a little bit larger. We see more of that green bacteria. They're really good in color. And then we have our how. They also have good color, but then we also see like some of the bleaching. And then you can see this is how, and then this is like fully bleached. And then we have our coconut core. That one single strand is still alive, so it gives us hope. That's good to know just because that water level was higher or lower on the substrate, so maybe focusing on that in the next experiment and then more surviving ones, but we only had like a couple counts of them. So I have another joke. So what did the opihi say to the limau kohu? It said, hop on. You probably thought it didn't say anything from the first joke. But if you're paying attention to my presentation, you'll actually see that we're using opihi and limau kohu attaching to it. So this is an important joke just because it shows that opihi can be used as a substrate and further be used as almost like a coral plug and attaching to how they do their methodology when doing coral. And so looking at the results, we see that the gore string at the top left is doing really good, high total settlement, but comparing to the other natural substrates, they're doing okay, but not as great. So there's a statistical difference. And then our last one is our Milo, which didn't really pull through just because of the whole health. We didn't want to create more mold in our lab, so we had to take it down, unfortunately, but yeah. And then our second week, we're looking adding the opihi. So we see that the coconut core, the how, they're still surviving, but at really low rates, and then our Milo is still at 0. But then our opihi are doing really well compared to the other 3. And then we see that the gore string is still at that high level. So yeah, it's good to see that and see that there is hope in using these different substrates. We just need to tweak them a little bit better so that we get better results. So we go to our haina. And what does this mean for limau kohu restoration? Again, that there is hope in the process and selections in our natural substrates. This is more of like a foundational pioneering experiment. So it's really good so we can build off of this and see what our mistakes was and what we can tweak to get better results. So some of the clear mistakes is like using more germling solution, but more spores rather than germlings, just because when you take them off of their first substrate, they can reduce in health, and then that could result in lower attachments onto these different substrates. While as using spores, they're more new, they're more healthier in that sense. And then we're also selecting different sizes and shapes of opihi, so more flat opihi rather than more steep, and maybe more larger ones as well, which could help. And then also the prepped materials, the how was fully soaked and everything. The coconut core and the Milo wasn't, so maybe doing more prep work for those substrates can allow for higher success. And then also using larger vessels so we don't come into the problem of the water going below the substrates and exposing them to air. And then also doing more assessments with the health and the bacteria I pointed out throughout the stages, and then the overall growth rate of these limau coats so we can kind of quantify how fast they're growing or how slow. And then also just to find these parameters to increase settlement, health, and growth rates. And to close it with one last joke, what did the limu kohu say while on the pihi's back? It's not the, oh, well, it said mahalo. Just because, just because we're giving them a place for them to settle and, you know, give them that chance to get to places. You probably thought it did say nothing or something else, but yeah. But I'll tie that into saying mahalo to the Pipes crew, all the Pipes hammers, Makana Hele and the Subrosia team, and all those who supported me this summer. This is really cool, different from my normal work, aquaculture, working in labs and things like that. So it was really cool, good experience, and yeah, thank you.
Ho'omai ka'i Seth, that was amazing. Do we have any questions in the audience for Seth?
Thanks, Seth. That was great. Were the OPE autoclaved before? Yeah.
Yeah.
And is there, like, next steps wise, is Symbrosia considering moving forward with OPE or including OPE in like another round of test substrates?
I think including another test round, just so we can include like a binder and things like that as well, but also choosing different, because these are mostly germlings, so we'll want to see how they do with spores, brand new seeds, and also, yeah.
And 2 quick follow-up questions.
Yeah.
Seth, are you going to be involved in that next round of
I
testing?
hope so.
Okay. And,
yeah.
oh, go ahead.
No.
The research that you showed before, it seemed like there was less colonization with a binder.
Yeah. So comparing to like the giant kelps, these are like brand new binders. She used the same binder as the giant kelp, but just because they're different species, maybe it doesn't really work as well. So maybe finding a more specific one for red limu would be more successful than using brown limu binders. Yeah. So that'll be the next step. Yeah. Thank you.
Was there another question from another intern for Seth?
Yeah. Coming from like the ocean, what would you say would be I don't need the mic. What would be a good substrate? You'd say coming from the ocean that would help the limu kohu growth. And where would you want to do some restoration efforts here on Hawai'i Island?
I think moving forward with substrates that aren't really, in a sense, needed, because using like important substrates and maybe substrates that, like OPE, we tend to just eat them and then throw them back into the ocean. So repurposing them and kind of have methodologies that are already created, like for coral, kind of helps us out in that sense. For restoration, we were working with Mauna Loa in their bay and with their restoration. So replenishing where people have seen it before and not, and then just growing the wild population would be great because it'd be fun to just go back out into your backyard and grab some limu kohu and just come back. So yeah.
Can we get one more round of applause for Seth?
Thank you.
Next up, we have Miss Modi'irari Niles Cantero, hailing all the way from Lahaina.
This is weird. This is very strange. Aloha mai kako. Aloha. I am Modi'irari Niles Cantero, and I was a PIPES intern in the Na'au pathway this summer. So just get ready for a lot of talk about pule and being vulnerable and how I was able to use my na'au this summer. Oh, here we go. But yeah, this is my project. So coming from Lahaina, my mom always made sure that, also I would just like to apologize in advance if I start crying. Anyways, this is my family, or just a very small sliver of the village that I come from and the people that raised me. But growing up, coming from Lahaina, my mom always made sure that my siblings and I had a pilina tu aina. holy moly, it's like 2 seconds already. Oh my gosh. Okay, get it together here. Okay. My mom always made sure that we had a pilina tu aina and our kupuna. And whether that was running up and down the river or playing out on the papa and the kai or just being outside from sunup to sundown, she always made sure we had that connection. But as I got older and I got busy, and I left Lahaina to come to Hilo to continue my academic journey at UH Hilo, there was a disconnect. I didn't feel as connected as I have before with Aina and my kupuna. But thanks to PIPES this summer, I was so blessed to be able to work with Lama Lama Kaulu, and they really helped. And my mentor, Makaiva Kanui, but they really helped me be able to rebuild my pilina tu aina and my kupuna. The inoa of Lama Lama Kaulu was beautifully said by one of my mentors, Pomaikai. She says that it's based off of the Olalo no Ea Lama Lama Ka'ili, which is a reference to the glow of the skin when one is healthy. Lama Lama Ka'ulu refers to the glow of growth or inspiration. This speaks to us when we are growing on our path and living in purpose. When we are doing inner work while serving our communities, we shine in a way that cannot be dulled or duplicated. Lama Lama Ka'ulu represents lineal practices and practitioners that are committed to liberation through healing and the generational inheritance of a thriving village. He ali'i ka aina, he kaua ke kanaka. Hello? Okay. Lama Lama Ka'ulu Land Healing Center supports the emotional, physical, and spiritual health of our community and Aina through traditional holistic healing and education. Lama Lama Ka'ulu's vision is to Lama Lama Ka'ulu, to glow in growth. Their goal is to assist our Lahui Kanaka to regain a vibrant state of health in any and all phases of the life cycle, from birth to kupuna. A question that really helped me this summer and to kind of stay on track was a question that was asked during orientation week, and that question was, what kind of kupuna do you want to be, and what will you do this summer to get there? When I first answered that question, I answered it very vague. I answered it along the lines of, I just wanted to be a healer, whether that was spiritually, mentally, or physically. I just wanted to be able to help our Lahui in any way that I possibly could and help heal them. But so these are some of the things that I've done this summer to get to, or a tiny little step to get to the kind of kupuna I wanted to be. The first thing I did was I began by familiarizing myself with the aina and the kaia'ulu of this vahi of pepe'ekeo, of alalaola. And by doing that, I was able to gather coal and then process it. And then I also gathered a lot of pua and just made bouquets and put them all over the site. Another thing I did was going to kole kole. And this was very special to me because it was so nice seeing like the people of that place enjoy their time in their place, like in their vahi, and just enjoy what their vahi has to offer. And then my favorite part was being able to take what I harvested and what I made and be able to share it with my ohana. Another thing I did this summer was a lot of reading and reflecting. I did a lot of research on la'aulapa'au and the different practices and the different uses of Native Hawaiian plants, the different medicinal uses of Native Hawaiian plants. Another thing I did was I did a lot of reflecting on the huaka'i that we went on throughout this summer as a cohort. The first huaka'i we went to was Kealakekua, and some of the takeaways I took from that place was the significance of mo'olelo and the importance of nanakamaka, ho'olohe kapepeau, and pa'akawaha. Another thing I took away was how important it is to document things. Then when we went on our huaka'i to Pack Rack, I realized the importance of place names. This is also where I realized how small things can have big impacts. And this is also where I decided that I wanted my project to not only be a malala aulapa'au, but to also just bring back the practice of Hawaiian medicine back into our daily lives and instilling that in our communities today. And then some takeaways from my PO was that taking care of aina is not a chore. It shouldn't feel like a chore. It is simply a kuleana that we as Kanaka have to oamu. Another thing I realized was the importance of being able to be vulnerable as a leader in your kaiaulu. Another thing was the importance of your mo'oku auhau, not only knowing where you come from, but who you come from and the people you carry as you're continuing your journey through life. And then our final huaka'i was tupu'ua'awa'a. And here I realized that if there's a will, there's a way. And that you have to start somewhere, one step at a time. And to also just be intentional in all the work that you do. But so with taking all of what I've learned from these huaka'i and being able to build a pilina to this place, I was able to create a malala aulapa'au at Lama Lama Ku'ulu at our site. And so this is the first, right here in this little mala, is where I initially had thought where the malala aulapa'au was gonna go. And so I started off by clearing out the hole, clearing out all of the weeds and the cane grass. And then this is after I cleared it all out. And the last thing to do is just to kind of just make her pretty, you know, just weed eat all on the sides. So I went to the warehouse, I grabbed the weed eater, but the weed eater had other plans. It didn't work at all. I tried cranking it or pulling the thingy, pressing the bubble. It still wasn't going, and I was just like, okay. So I just sat under a tree nearby in defeat. I was like, hello, what is going on? So I kind of just said a little pule and spoke out loud, asking for guidance on what to do and why is it not working? But so after about 20 minutes of sitting there was a big gust of wind that came from the Makai side that blew all the way through the Mauka side of the property. And after that gust of wind, I came to the realization that maybe the weed eater is not starting because the mala doesn't belong in that puka. And so I kind of walked around and I realized that there was 3 kukui trees growing up, like already growing on the uka side of the property. And I took that as like a whole ailona of maybe like this is where it belongs, where all the la'aulapa'au is already growing. And so I voiced that to my mentors, and they're so amazing, and I'm so blessed because all they had to say was, okay, mahalo for listening to my na'au. And so they let me do as I did. But before I started clearing out the area, I took a lot of time, and I spent a lot of days, honestly, just like sleeping near the mala and just like praying and just making sure that this was the right area where the mala was supposed to go. And so after about 2 days of sitting, I was finally able to see a vision of what I wanted this area to look like. And so with the help of everybody, Uncle Maka, all the girls, and my sister, we were able to clear out the Mexican sunflower. And we were able, this is what she looks like. But then after we were done clearing it out, Uncle Maka had brought it to our attention, or reminded us that the neighbor does spray fertilizer along like their side of the fence line over here. And then again, I went thinking like, oh my gosh, now I have to restart everything. But I sat in the mall again. This tree right here, we're like besties. She was my pillow. But so after, once again, sitting and just praying and asking for guidance, I came to the realization that although, yeah, the neighbor does spray fertilizer there, it'll be okay. Like the plan is to create a boundary right here with other plants to kind of, like as a blocker, if that makes sense. But yeah, I was just like, you know what? These plants are gonna heal people and they should be able to stand whatever conditions and yeah, to be able to grow strong. But so then after clearing it out, we had lovely Sema come over and test the pH levels of the soil there all over the property. And because it was kind of already expected, because Pepe'ekeo has history of the plantation, and So the soil there's already acidic. Where this mala is, the test came back at a 5.03. So that just meant that the soil was super acidic. And so to fix that, or to help restore the nutrients into the soil again, with the help of my LKU Ohana once again, we went around the property and gathered a bunch of brown and green banana leaves. And then, so this is Uncle Maka. He placed dolomite down before we put the brown leaves on. And the dolomite is a natural lime that helps raise the pH levels in the soil. And then, so after that, we went with the shovels, and you can see over here, we kind of like mixed the dolomite into the soil, and then we started covering it with the brown leaves. And then we watered it down, making sure that the brown leaves were nice and flat and they weren't gonna fly anywhere. And then we repeated the same steps with the green leaves as well. We placed those on top and then put it with water, and then we covered it again with the weed mat. So this is a little before of what the mala looked like, and then this is the current state of, This is the current state of the mala right here. And so the plans is to plant the native Hawaiian plants on the upcoming Mahe Alani on August 8th, which just so happens to be our final day as together as a cohort. But yeah, so after this huaka'i and going on this journey, like I mentioned, I realized the importance of documentation, especially for nonprofits, but also the importance of documentation for the future to have something to source. And so what I did was I created like these little posters or cards. And these are just some of the 2 plants that I'll be planting in my mala, but I created these cards of the medicinal uses along with other uses that can be used, and then along with like mo'olelos, just as kind of like a educational, the site can use for when people come. And then back to reflecting on the question of what kind of kupuna do I want to be. Here I go again, crying. After this summer being around all of the amazing kupuna I was blessed to share space with, and then also thinking about all of the kupuna that raised me, I don't only want to be a healer, because I did come to realization that even though these kupuna have all these healing sides to them, they also have a very strong warrior side to them, and they do a lot to fight for their kaiulu and for our lahui. So now, to answer the question of what kind of kupuna do I want to be, I want to be a kupuna that does heal, whether that be physically, spiritually, and mentally, but I also want to be kind of a secret weapon, I guess, for my kaia'ulu, and be able to help them in whatever way I can, whether that be conservation, sustainability, or even politically. But yeah, so my end goal after this summer is to return back to Lahaina and share all of what I learned this summer with my kaia'ulu. I also hope to start my own mala, la'au lapa'au, in my community, ensuring that everyone in my kaia'ulu has access to these medicines, but to also help bring back traditional Hawaiian practices and reinstilling them into our daily lives. I would just like to mahalo everybody, our whole PIPES cohort, and then all of our sponsors, because but yeah, just mahalo to everybody, because without them, I wouldn't have been able to rebuild my pilina with Aina and my kupuna. So mahalo. Ooh, sorry for crying.
Amazing, amazing.
Mahalo. That's mom, by the way.
Amazing, amazing job, Modi'i, of reminding us why we need all of these super strong pule. Do we have any questions in the audience for Modi'i? Hemoni no ka'oko? We do have one question from our online audience. From Tatum, they ask, or they say, mahalo cousin, love you. I was wondering how learning and teaching la'au lapa'au connect us back to Aina and culture. I guess like how you did for the educational cards.
Sorry. Can you say that one more time?
They were wondering how learning and teaching la'au lapa'au connect us back to Aina and culture. So how the hana hana that you did this summer, kind of different from other interns and the work that they did, connects you still back to Aina.
For sure. I would say that, I don't know, I feel like it's more of like, I feel like it's a kuleana to learn because these were Hawaiian practices that our kupuna once did. And I feel like it's important to bring that back again and to revitalize these practices and the uses that Aina and our kupuna have to offer us. I don't know if that answered anything, but yeah, sorry.
We do have one more question from the online audience from Kealo Nassau. Since you plan to bring this ike back to Lahaina, how did your connection to your own kaiaulu influence your approach to the mala and this healing work?
Oh yeah, okay, that's a good one. Like I mentioned before, coming from Lahaina, my mom did really instill that pilina to Aina and our kupuna growing up. And so we were always like outside in the mala and working. But then definitely after things that happened, there was that disconnect. But I'm so blessed to be able to regain that pilina to Aina and my kupuna again. And so the plans after this is to actually, there's like a farm in Lahaina that opened up after the fires. And it's kind of volunteer work. And so I was planning to reach out to them to kind of have that space to open up the Malala Aulapa'au so that the community of Lahaina has access to and can use. I don't know if that answered the question either, sorry. I'm so sorry. But yeah.
Any more from our in-person audience? Oh yeah, heading back to Mama.
Or no.
I can also kind of answer that last question for her.
Yeah, please do.
But Modi'i, and it's harder for Kanaka to talk about themselves, right? And our cherished pilina, but you also do have Ohana that does la'au lapa'au. So second day into the internship, she was making us la'au shots and singing mele over them. So bringing her own ike and bringing us la'au for us to heal too. That being said, I think one of the grounding questions that we want to keep bringing back to Modi'i that she doesn't want to talk about, but it's also a question for all of you folks too. You know, what's different with Modi'i is she had the time to be in a healing space. So we're working with healing practitioners. So besides the project, I think our overall hope was for her, and this might be a lifetime of learning, is what is the la'au you have to offer? What is the la'au in you? Because, right, the pilina, there's one thing to have the pilina with the la'au, and the la'au is a medium, right? Our kupuna practice la'au kahea, right? Where we can draw the mana from that la'au through pilina, and even that could heal. So the la'au itself is one thing, but in some ways, we individually carry healing la'au within us, in our na'au. Maybe you'll have a good answer. Maybe it's just a reflection question for all of us. But it's an important one for all of us to think about, and how are we offering that la'o in all the spaces that we're in the research that you're doing, etc. Mahalo.
Yeah, shucks. The la'o, what kind of la'o do I have to offer? Is that the question? Okay. I would say the la'o in me is my kupuna and the people that I come from. Here I go, crying again. If it wasn't for my kupuna, I wouldn't be who I am. And I'm simply just putting out all the love that they put into me and sharing that with everybody. Yeah, so I don't know if that answered your question, but I'm just gonna leave it at that. Ooh.
Can we get one last round of applause for Miss Modi'i? Up next, we have Miss Kanani Tim Singh, Kamua ona Homana Olalo Hawai'i, no keia makahiki. Ola ko olalo.
Aloha mai kākou, o kia ka upahana o ka ulana ike, ai kia ka vela ua hana wau me hiki ola ma ke ala ke kua. Ma no pipes, aia wau ma ka hui ka iaulu. No leila, o ke kālele nui o ka upahana o ea hoi kia o lalo no ea o ke kahua mamua ma hope ke kukulu. First the foundation, and then you build. That's what I base my whole project on. So because that's what I base my project on, o keia ka'u kahua. O Hawai'i ko umoku puni, o hilo I ka ua kanile hua me ka ulua o ko uone hanau. Noho kupa ma puna paia ala I ka hala o kea au kea hupua'a ma nā malu o nā moani ani le hua. O Deo Kanani Tim Singh ke kane au no Cassie Noia Strande ka wahine noho pulawa a hanau ia o Mackenzie Kanani o Aloha Tim Singh o ka punana leo o hilo ke kahua. Kukulu a puka mai ke kula o nā wahi o ka lani o pu'u. Ai kia manawa aia wau ma ke kula nui o Kaminaka ma o ahu imi ana I ka ulai pua ma ka environmental studies. No leila e like me ka'u ua hana wau me hiki ola he oihana ku I ka auhau ole a kālele o hiki ola ma ka au ho'ona au ka au ka iaulu ke au kulehe lohe ka ike ku'una Hawai'i. A leila hana pu o hiki ola me ke kahi oihana o ke ala ke kua Mountain Reserve kapa mako o KMR. Malama o KMR I ka Ewa Kokani e Leono Haneli Iwa Kālua Kumahiku Eka o ka Aina ma ke ala ke kua uka, a oia kahi o ka hale ha'akipu a me na mikini hana I ka aila. No leila o keia ka pala aina o ke ala ke kua Mountain Reserve, ka 9,627 acres of land up ke ala ke kua mountain. So hiki ke ike I ka oko'a o keia aina mai keia aina. You can see the difference from down here from up there because this once was grazing land for cattle. And then that's the native forest. But from 2019 to 2025, Hikiola, the community, KMR, have planted almost 700,000 native plants back into this forest. So this was from a map from kind of long ago. So hopefully now down here looks a little bit more like up there. There you go. Okay. Ma hiki ola he 5 ano huaka'i hiki ai ke koho no ka pi'i ana I uka, no leila loa ka huaka'i pokole, ka huaka'i nānā, ka huaka'i hana, ka huaka'i ho'ohui, a me ka huaka'i kanu. Ma ka hana ka ike. So in the beginning of my project, we had a foundation, we had a kahua, and it wasn't until I actually went komo I ka hana did I know the ike pili I should put into these books. So eia ke kahi o na hana a ui hana I keia kauvela
Ua ohi mākou i nā ano no leila ka hapanui o nā ano ohi ia ma ke mar no ka hale hākipu, ohi ia e ke kime. And then ho'o ka'awale a wai i nā ano na mea mai kai ole. A leila ka ho'okomo ana ma nā pā mea kanu, malama a mākaukau a e ulu maka ulu la'au. So nāna ka maka ho'olohe ka pepe au a pa'a kauaha. O keia ka mea I kōkua nui ai ia'u ka hiki ke pi'i me ana ke li'i ka noi. Mana hua ka I like ole a ka hiki ke a'o I nā mea like ole mai ka hui, ka ohana, KMR, hiki ola. Ka hiki ke ho'olohe I nā mo'olelo no ka ʻaina, nā lauana mai ia ʻaina, nā mea ola, nā holoholona. Pela wau I ʻike ai I nā mea e ho'okomo ai ma nā puke. So this summer I created activity books that can be used with activities in it that can be used up in Kealakekua, but also, at school or at home for grades K through 6. So making something like this is a picture at Kealakekua, the gate right before you go into KMR, and then it has the io, and then this is for Papa Mala'o, but then this was my first rough draft, and then turning it into something like this for the kids to trace and learn how to write. And then it has the eel welcoming all the little eels into the space. And then you have like the background, so having the gate and then turning it into something like this for the books. And then even turning pictures of eels or all the manus or whatnot, and then turning it into coloring books so the kids can color. So this is some examples. It's not all the pages of the books, but it's a handful of them. And then because I didn't know what I wanted to put into the books when I first started, I first started to research, like, what do kids at this age need to learn? Like, what skills do they need to learn? So in kindergarten, they need to learn how to write, and hopefully they can write their own name, but they need to learn how to write and, drawing and, like, seeing things. So this is an example, aloha, o wau, o io, welcoming, and then pelina mai ka aina o Kealakekua, then what lacos you would need to bring up, and then asking them questions like kiloa kahaki'i, and then how does a plant grow, and then I chose a handful of native plants that I saw up Kealakekua Mountain that you wouldn't usually see where we all live, like in your backyard or like on a daily basis. So like something that I thought was important for keikis to see and learn. And then you have like asking them questions, making them think. And then some of the manus that are up there. And then the pulele hua, the ope'ape'a also. And then you have the kahulis up there. And then just like I showed you with the eel, turning that into little coloring pages. And then the background is also our queen tree, iliahi, that's somehow growing on a big rock in Kealakekua. And then also teaching them the invasives, and then also a call of action. So each book is kind of based off of this ikepili. So call of action, asking them, how do we aloha aina? How do we, malama ka aina? And then this is 3rd grade. So 3rd grade, you're a little bit older. You're learning how to read and write sentences, and then doing math and like making connections to everything, like connecting the dots in some type of way. So asking them, like, what's the mokupuni moku ahupua'a of Kealakekua Mountain Reserve? Oh, sorry. And then asking them, like, what's your mokupuni, what's your ahupua'a, and hahana hi'o no ko aina, so, like, what do you see, patterns and things of your aina, where you're from? And then doing the parts of the plants, the trees, and then what are the names of these trees? And then you have the parts of the bird, and then little games like this, and then what's their names, and then making little cartoons of the ope'ape'a, and then what's the parts of the ope'ape'a. And then 6th grade, it's a little bit harder, but 6th grade is really where we tackle down, because that's the grade where kids are starting to think like, oh, in school, why do we need this knowledge? Like, why do we need to learn this? Like, how am I going to use this in my life? So kind of trying to find a way to work it all together to make it something valuable to the haumana. So asking them, what's your name? Where are you from? What's your makani? What's your uas? And then each book also has Mele Hikiola that Anakekeli Kanoe wrote, these two, and like asking them why it's important to have these meles to us, and then writing little bit stories, and then also teaching them like topography and elevation, so having them color the different elevations of Hawaii. And then just a little bit more stories of the Anila, and then some Olelo no Eaos. And then also teaching them how elevation and rainfall works together. And then asking them questions. And then pehea ke aniela, so in kakahiaka sometimes it's like this. And like, so that they see like there's patterns that work with what's around them. And then also like little stories that I wrote, and then they, from what I just taught them in the previous pages, they have to fill out the words into the story. And then for all the birds and things that I put into the book, I made little coloring pages, and then they have Olelo no Eo up there. And then making stuff like this, so ehanai ka io, feed the io, and if you know what the io eats, you'll easily get to the bottom. And then also like challenging them, like, okay, now it's your turn to hakua Olelo no Eo, it's your turn to hakua Proverbs, so making them think like, oh, what is a apapane like? What does it eat? What does it do? To like familiarizing them with that, yeah. And then each book also has a little kilo log. It's all kind of different in each book because different grades. But yeah, getting them to see the patterns and what's around them and familiarizing them with their space. And then elave ka ike a ema lama a oi mau kana awao. So these pictures are example from my mom's classroom. She was a teacher at Punana Hilo, and now she's a teacher at Ehunui Kaimalino, and Hikiola works with Ehunui Kaimalino a lot with the keiki. So I was lucky enough to have her as a resource, asking her questions about the students, like what are they interested in? What do they like what hobbies do they have, to really know my audience to really create these books. And then she also used my clip art in her classroom. It's all over the place for free. And then Helala vo no ku'ukumu. Mahalo nui loa ya anake kili I konoi ka ohana hikiola, ka ohana KMR, ka ohana pipes. Mahalo nui loa no ka ho'i ana iau I ko umole a ko ukahua. Ihiki au ke kukulu, puka megalana kila and kokua I kahano naho. Yeah. And then the main thing of my project is really to kind of familiarize the keiki with their spaces and what native things are around them. Because, you know, sometimes we forget the name of birds or we forget the name of plants, but then there's always that connection, that pilino we have with everything around us. And like with that comes respect as well to everything in our space. Mahalo.
Ho'omai ka'i e kanani. He maunino ka oko e kia naina. Anyone get questions for kanani?
I thought you did really great. I'm not sure if you answered it already, but do you have plans to implement these books into certain schools?
I forgot to say that. Thank you for reminding me. So in the future, we're looking to also make them bilingual because makuas are the real ones who are gonna put this on the keiki. So we're thinking of giving the ones in Hawaiian away to Ehunui Kaimalino and some other Kula Kaiapuni in Kona or yeah, and then also trying to make them bilingual. And then next summer, we're looking to make a summer program for little kids up Kealakekua Mountain, so taking them up Uka pretty frequently so that they can really see these things. Yeah.
Aya kekahi ninau maka YouTube, ninau maila o pua ala, pehea ko manao no ke ka'ana like ana aku i nā puke.
Yeah. Ua manao mau ana ke e kau ma kana mea ma hikiola.org. Plug. Yeah, so ina make nana ma leila ke pau pono kia mau puke a pau pa'a aia no ana ma hikiola.org.
Mahalo nui ke kahini na o he ako pohu hopu. E lilo i ke kumu i ke kahila?
Well, he kumu ko mau kuahine, and ua manao wau na keia mau makahiki hala, I will never become a kumu, but it might be something I like to do because environmental studies hana me ke kaya ulu, and something that we really have to do nowadays is really getting the audience of the new generation because half of the people in Hawaii are not in Hawaii. So really making them interested in what's around us and what we have so that we can put more of what we have. Or what we don't have. Putting out natives and whatnot, you know, just making them interested to stay here. That's my biggest goal.
Can we get one more round of applause for Miss Kanani? Up next, we have one more paha no olelo Hawaii with Miss Kamaka Frasco under the direction of Steve Du.
Alo mai kako. O Kamaka Frasco ko ino. Ayau ma ka hui na'au. Wahana wau ma ka papahana ho eao me ka hui Pacific Aquaculture and Coastal Resource Center ma Keoka. Ea mai ka upahana he puko a kani aina. Kaulai. No laila no ka uhua. No hilo Hawaii mai au. Kele nei kula I ki kula noi a Hawaii ma hilo me ka me kia Pelicania o ia maki au I lilo I kanaka hakupuke. Nui ka o aloha I na mea o la kai, a me ke kai. O ia hiki ke a o I na mea li ke ole he wahi o pio pio ai no u mana wā paakiki I kupu ai ma ka ola. Alela o pack rack he wahi ho'ona o wau ma ke o ka Hawaii a ka lele ia I na ano mea ola kai ka mahi a me na ano pāhana like ole I pili ke kai e like me na ko'a, na limu a me na ia. Alela ia he mau ki'i kea o mua o pack rack. Alela ia he mau pahu vai e ho'ohana ai na homana na ho'okolahua e like me na wana a me na ina. A leila na pahu hopo e lua ua ho'ohana wau na ko'u pahana o ia no na ko'a. A leila ea kahi e ho'okolahua ai mako maluna o na ko'a I hiki ke malama a ho'oulu o ia ale mai ka'i ko kākou mau ko'a ma loko ke kai ke manawa. O leila make mako e ho'ola a ho'oponopono I kia pilikia. O leila ua make o ai kālele ma luna o ka kahi I ohi ai na ko'a. No leila hoi mako I na ko'a mai kākai I pack rack I hiki ke malama I na ko'a a hoi I loko o ke kai. O ke kai ona wahi a mako I ohi ai na ko'a o ia o puhi Awa kapa ia ma hope o kei lua puhi mai ka waka iko. A ka kia mola au he puhi. Ai. A leila wa hele pu mako I auwili. A ka ali I loa nui na koa I hiki ke hoi hoi ia pack rack a ho'okolahua ma luna. A ka mako hele ana I waiuli wa nui na koa I paui na nalu o iai ke hele mai na malihini hehi lako ma luna o na koa a haki mai ka po'o koa a no laila wa maki mako e lawe I ia mau koa a hoi hoi I pack rack. A leila ke kaina hana o ko mako hana ma pack rack ke lawe ke hoi hoi na koa waiho mako ma kekahi aila ma loko o na pahu ke'o I hiki a lako ke ma'a I ka wai e a'e. A leila waiho ho'ohana mako I mea ho'oki I hiki ke ho'oli'ili'i ia mau apana a waiho ma kekahi mau kile la meka I hiki ke hoi hoi I ka aina. I nā koa ma loko o kekai. A leila ma hope aku waiho mako I loko o kea pahu kai I hiki ke maka ala a malama ma mua o ka hoi hoi ana I kai. A leila ma makemake pū wau e kālele ma luna o ka hanana na mo'o'aina. O iai ua hiki au ke kokua ma ka ho'ike e ana I ko mako hana ma pack rack me ka ko'u hana me nā ko'a. A leila ua makemake wau e kapa I ko'u pahana he pū ko'o kani ʻaina. O iai ua ho'omaka au me ka li'ili'i o iai ʻaʻole wau I ʻike nui e pili ana nā ko'a a I kēia papahana huʻeaʻo. A leila eia kekahi o ko'u mau noiʻi mai Papakila database. O he new pepper kahiko kea I kākau ia e pili ana ke ʻano o nā ko'a ma Oahu. A ka ʻaʻole wau I hiki ke ʻimi nui nā ʻatikala no nā ko'a o iai ʻaʻole lākou I makemake e kākau a unuhi ma ka hoʻolelo Hawaiʻi. A leila eia kei mau ki'i hoʻāku no ko mākou pipes ʻohana I huakaʻi I pack rack. Ua hiki iā mākou ke launa a walaʻau me ʻanakala manaʻe a aʻo I nā inoa wahi a me ke kumu I kapa ia mau kākai ko lākou mau inoa. A leila eia kei mau ki'i o ko mākou o ko Steve, ko'u mentor. Hoʻike ana I ka hana o mākou ma pack rack. A leila eia mai o Hola. E hana ana I nā palapala ʻaina ma Auwili me ka Megalab. A leila eia kei mau ki'i ma Pui Bay. A leila eia ka dar hoʻike ana pehea e kiloi upena ai. A leila eia mai ko ka Sierra Club e hoʻonaʻauao ana iā mākou pehea e lilo I alakaʻi maikaʻi no nā hanauna e hiki mai ana. A leila no koʻu huakaʻi ua makemake wau e kālele ma luna o koʻu hana me koʻu me ke kaʻeo. O iai ua makemake au e kokua e hoʻonaʻauao I kēia mau hamana ma ka papa ʻelima e pili ana nā koʻa ma ko kākou kai. A leila eia ko lākou hana nei I nā koʻa me ko lākou mau lima. A leila ua makemake pū e hōʻike a lākou ka hiki ia mākou a pau ke komo hoʻokahi a kukulu I koʻa he nui. O iai I nā ʻaʻole loa ke koʻa I nā ʻāpana a pau. Alena hiki ke holopono na mea pau. No leila pono kako e komo a lau lima ma ke ia mohana. A leila ua makimaki pu e kalele maloko o ka pilina me ke kai. O iei, ale na homana I ke nui e pili ana na ko'a o iei ale lako I kilo nui. No leila ua makimaki e a o ia lako pēa e tuko I ko lako mau ko'a I mau kile la meka. Ea mai ko lako mau ko'a mana kile. La meneka. A leila ea he mau ki'i hoaku e ho'ohana ana I ke kaleka ana ko'a. I hiki ke ike I ka waiho'olu'u o ko lako mau ko'a me ka ano o ke ko'a I nā mauna ke ola nei a ole ua make. A leila no ko'u ho'ina o makimaki wau e ho'ike no ka ko'u hana mua ma ke core nursery. Nui na oko'a maika'i nui na mea ia'oai. A keia kanana ana o ke core nursery, kia mola. Nui ko mako hana wa'o nui au pēa I lilo I ala ka'i maika'i no ko'u mau pōki'i. Hoiki pū ia'u ke a'o I ko'u ike o lalo Hawai'i I ke core nursery. I hiki ke ho'ola mau I ko lalo Hawai'i. A leila, no ko'u ha'ina e ho'ohana na wau I kia mau ha'awina I kia kauwela no kia mua aku. E like me ka ho'omau ana I ka haku ana I mau ha'awina no ka pape lima o ka ome ke ka'eo. Ma ka lalo Hawai'i piha I hiki a lako ke a'o I nā ko'a a me hiki ke ho'omau I ka ho'ola I ka lalo Hawai'i. I hiki pū ke lo'a I mau hanana hoaku ma pack rack e like me he puko'a kani aina e 2 kaukani 2.5 ma ke aukaha. O iai makimaki wau e I nā kaiulu a me ko'u mau pōki'i e a'o I nā waiwai a me nā hana ku'una I ole pau kia ike. Laila ea he mau ki'i mai kia hanana e o mua. A leila ea nā kanaka I komo a kokua mai a mako I hiki ke hana I kia hanana. Ea kia I maula ana. Ua kokua o dari a mako. Na liko pilina. Papahana o moku. A leila ea mai o aina ke a'o nei I ka po'e pēa e hana I i'a me ka lauhala. A leila ea he ki'i o ko mamua o ko mako komo ana I loko o ka wai e ho'i I nā ko'a a tuko ma luna o nā ko'a e a'e. Hela ea he mau ki'i o kena hana. Me ea ko Steve kokua ana I ke kane pehea e tuko I ke ko'a me ka maika'i. A leila ea he mau mea ola kai a mako I ho'ohana ai no kia hanana o iai ua makemake e lo'a I pahu me nā mea ola I hiki nā keiki ke a'o e pili ana I kia mau mea ola a me ka I hiki a lako ke ho'opā ki kai. A leila ea ko lako hana ana I kia ka ho'i ana I nā ko'a. A leila ea mako e ma hope o kia hanana. A leila wa makimaki wau e mahalo. Ia'u ko pakaia po e kokua ana ia'u. Ma na ano like ole. Me e ole o ko koe au ka umau pauhopu. Leila makimaki wau e mahalo. Hiki kula nui o Hawai'i mahilo. Pipes ki puka ke kaiulu o ke aukaha. A me hale kua mo o na ke kāko mau ana ia'u. Māla nui.
Ho'omaika'i nui e ka maka He mauni nau ka o kou e ka lehu lehu.
Aloha mai e kamaka. He nina kau e hia mau ano koa e ihana ai I keia kawela a me like he aha kau puna hele ano koa. A me nai noa ina hiki.
Yeah, ai.
Hoi hoi loa wau.
Kola mai. Ale vo I veve. Noela no ko upana wa ho'one vau e imi ina inoa ona koa a kali hiki ke imi. A ka kia mola he e lua inoa ona koa ka pohaku puna a me ka a koa kohe. A ka make vau ina koa pau. Ale vau loa I mea puna hele. Yeah. Oh, and then ale for makimaki puina. Wa haku vau I puke color me kahi ona holoholona maloko o ke koa a noho maloko ke koa. Mahalo.
Mauni nau ho aku. Ana holo viki viki.
O ka uni nao oia no e hia mau mahina ai ole makahiki paha I pono ai ke koa e ulu ma paka ma mua o ka hiki ana ke hoi hoi I loko ke kai.
Steve? She's asking how much months. How much months does it take to take care of the coral and then take it back? Yeah. I'm so sorry.
Sorry, I don't have my. Pua ala asked, ovai ko mea punahele ma pakrak, like ko mea ola punahele. Oh yeah, ua pana ia no. Kalamai. O ka uni nau, he ni nau kau. Ma kanina wele ma mua o ka maka o ke kauwela ua olalo oe. Make oe e lilo I kanaka hakupuke, mau no ke la, ai ole he aha ko make ma hope o ke ia kauwela.
Ai, mono ko make lilo I kanaka hakupuke. Oie, oi ki kumu wa make vai hana me hale kua mo'o, oie hiki o ke haku I puke ma kololo Hawai'i, nona keiki, ai hiki ke ho'o na awao ia lako e pili ana I ke kai.
He mau minuke I koe ma mua ko ho'omaka ana o ho'omaka o Laila ma ka hola umikuma lua. So he mau ni nau hou aku.
Mahalo nui e kamaka, nui ka vaivai o kao pāhana. O ka uni nau ia oi, he ake kahi haawina vaivai au ia o ai ma ka pili ana me nā haumana ma ka umeke.
O kei haawina nui oia ka pono e ho'omanawa nui me lako. Oia nui ko lako I kehu a me ka make e holo a walao me ke kai, leila pona e ho'omanawa nui. Hawi la haawi I alo ia lako, I hiki ia lako ke holo he a mako. Mahalo.
If there's no more questions, can we get one more round of applause for Kamaka? Up next, we have Miss Lila Shapiro with Pilina. Likona Pilina.
Okay. Aloha mai kako. My name is Lila Shapiro, and I am from Puna. I just graduated with a biology degree from Lewis and Clark College in Portland, Oregon, and I am a first-year intern in the Ka'au pathway. My project this summer is E'a'ea Ulu using indigenous nematode warriors against Queensland longhorn beetles in Kukui and Ulu. Being born and raised in Puna, surrounded by nature, cultivated my curiosity and shaped my passion for conservation. Observing and engaging with my environment has deeply influenced the way I approach biology. The Aina has always been my greatest teacher, instilling in me a sense of wonder and a desire to protect it. This deep connection to the land and Kai is central to my identity. After graduating from Lewis and Clark College, PIPES and working with Likona Pilina has allowed me to reconnect to my roots and gain a clearer understanding of the type of work I want to be a part of moving forward. So, my host organization for this summer was Likona Pilina. They excited me because of the crucial work they are doing for our native forests, and it offered the opportunity to contribute to a project from its inception. Since the early 2000s, Likona Pilina has been using this hybrid model, combining native and non-invasive non-native plants to research lowland forest restoration. These methods have proved extremely successful. They've improved resistance to invasives and boosted desired plant recruitments, along with many more amazing findings that I don't have time for, unfortunately, today. Unfortunately, in 2021, the Queensland longhorn beetle became a concern at the study site, threatening two important canopy species, Kukui and Ulu. These two trees provide shade that suppress weed growth and create an ideal habitat for our native seedlings. About a year ago, the effects got too intense to ignore, so they began looking into treatment methods. Kathy Mello and Roxana Myers with USDA discovered that there was this indigenous nematode found at Hilo Bayfront that could be used to kill QLB larvae. Although our lab has been using this biocontrol method for about a year, its effects haven't been fully studied or quantified yet due to the demands of their original long-term study and limited staff available for extensive monitoring. This is supposed to be a video, so just imagine them like wiggling around. That's how they move. And so that's where I come in. It's been really exciting to dedicate my summer to this project and get to work with and add to brand new protocol for treatment for trees that are so important for the health of our ecosystems and that hold so much cultural significance. The question that has guided my past 11 weeks was, does the nematode species Heterohabditis indica serve as an effective biocontrol for Kukui and Ulu trees? So now getting into my ha'alele. Our study site is at the Keaukaha Military Reservation within the Ahupua'a of Waiakea. Zooming in a little closer, this is a map of our study site. So treatments were conducted along the Puna Trail, the Escape Road, and then within the already designated Likona Pilina plots. Here are a few pictures of what the inside of this forest looks like. So you can see it's an a'a field, and there's many different plant species within it. And then, just a warning for the next couple of pictures. They're kind of disgusting, and I really hope it doesn't put you off your lunch, but this is why we're trying to get rid of them. They're not only detrimental to our trees, but they're also horribly unpleasant to look at. So this is the Queensland longhorn beetle, which is a plant-boring beetle native to Australia and very invasive in Hawai'i. It was first discovered at a residence in Puna in 2009, but reports of individuals infesting host trees did not become frequent until 2014. The females lay their eggs on the bark of trees, which hatched into cream-colored segmented larvae. And then let's see one of our larvae. So then these larvae can grow up to the size of your pinky, probably bigger than my pinky. And they use their mandibles, their jaws, to bore into the tree, and they create these tunnels called galleries that really just wreak havoc on the inside of the tree, and it impacts the host's ability to transport water and nutrients. So you can see in this picture, up here is what the inside of a tree should look like. It shouldn't look all mushy like this. And then, this one's really gross, then they pupate within the tree, right, kind of at the beginning of the bark, and then they emerge as adult beetles. So QOB increases the risk of infections of other pathogens, weakens branches, and can ultimately kill the tree. This photo shows the effect of an infection. This is actually one tree. This branch you can see is completely healthy, whereas this one is infected. And the infection was actually all the way up here, but it affects the tree's entire system. And here are some symptoms of QOB. This picture shows frass. So these are wood chippings that get expelled from the tree as the larvae dig into the tree further and further. And so all this should be inside the tree, not outside the tree. This is an exit hole, which is created when the adult beetle emerges from the tree. And then this is sap, which is a telltale sign of infection for kukui trees. Now let's dive into our forest. So here's our kukui tree. Kukui symbolizes enlightenment and has been the official state tree since 1959. Traditionally, all parts of the trees was used. However, due to invasive species and anthropogenic changes to our forests, kukui populations have been declining across all the islands. And so that's why work like this is so important. We need to find solutions to these invasive creatures. And then ulu, as a kino law of ku and a food source, ulu holds immense cultural significance. In fact, one mo'olelo describes an extreme drought and famine where ku transformed himself into an ulu tree in order to feed his family. Because of this, ulu symbolizes resilience, perseverance, and security. And now a look at our actual nematodes. Okay, yay, that's what they look like when they're moving. So here are nematodes under a microscope. Nematodes are just like a type of worm. They are an obligate insect parasite, meaning that they need a host in order to complete their life cycle. They have a symbiotic relationship with a bacteria that's found in their gut, and this bacteria gets released into their host when they crawl inside of it, and it kills it via sepsis. The nematodes are then able to feed on the host and reproduce within it, eventually releasing hundreds of thousands of offspring that disperse in search of a new host. And so remember this picture, so this is that drop of water. So you can see all the little lines, like that's an individual worm. And so that drop of water has a whole lot of nematodes. And so because of their insect lethality, they have been widely used as an effective biocontrol against various pests and invasive insects. Oops, okay. And so here's a picture of our lab setup. In order to bring the nematodes into the field, we have to grow them ourselves. So we have our initial batch from Bayfront that the USDA gave us, and then we have to rear Galeria moth larvae in here because we're not allowed to grow more QLB. That would be kind of horrible and against the point. So we grow moths instead, and then we use these larvae that you can see in these petri dishes, and we inoculate them using our nematodes. And then we have water in these petri dishes, and we wait for the nematodes to emerge from their host, and then collect them and put them in these flasks, which we then bring into the field. So there's a whole lot of background work that has to happen before we actually get into the forest. And so right before we go into the forest, I go back to the lab, and I count how many nematodes are in a drop of water in order to take the concentration so we know how many worms we're injecting into our trees. And then we get into the forest. So once we're there, we look at our trees and we are looking for signs of infection. So these are some pretty severely infected trees. Like that's a whole handful of tree that's outside the tree that should be inside the tree. And this is all sappy and gooey. And so once we see the infections, then we use our needle to poke straight into that infected area. And because the bark is so weakened, it is unfortunately pretty easy. And so we shoot nematodes into each infected areas. We've gotten ourselves into some precarious situations trying to reach the high up infections. And so also in the field, we go through logs that have fallen to make sure that we dig out any larvae that remain, and then we bring them back to the lab. So this is another little larva that we found. This is also an example of our data sheet where we take meticulous notes so we can use this for our research and then future studies. And then beyond the research and data collection, some of my favorite field days and some of the most rewarding parts of this summer was hosting school groups to get to share my love of Aina with the next generation. Getting to reconnect Keiki to their roots and show them that a career in Aina is possible and that they can help their Kai'ulu as well was incredibly fulfilling, and their excitement and curiosity gave me so much hope for the future of conservation. And now going into my huaka'i, here is the information for how we ranked infection levels for trees. So trees were ranked on a scale from 0, no infection, to 3, severe infection. And in case you forgot how horrible these larvae look like, here's a video, hopefully it works, of Emma Oh no. Oh, I didn't know there was gonna be sound. Wait, okay, it's working. So Emma up there pulled that out of a super infected tree. We were shocked. If you can't tell, that's the biggest larva that either of us had ever seen, and to pull it straight out of a tree that was crazy. So now that you see why this is so bad, now we can look at the data, and you can see how important this is. Okay, so this is our raw data. For the kukui trees, out of the 62 infected trees that we had, 30 trees had no signs of infection after treatment. 16 had a reduction in symptoms, and 16 showed no change in infection levels. Having no change in infection is amazing on its own, because without treatment, it can be assumed that the infection will get worse and worse until the tree dies. And then having that many trees with no infection by the end of the summer was way more than we could have asked for. But to determine if this raw data was actually statistically significant, I ran a paired t-test. And my p-value was 1.316 to the power of -14. Any value below 0.05 indicates significance. And so look at all those zeros. A p-value so low effectively rules out random chance and confirms that these decreases were highly significant. So in other words, it totally works. And then for our ulu, QOB are known to prefer kukui trees over ulu, so our sample is much smaller for our ulu trees, but we're not complaining about that because of course we'd rather have our trees not be infected. So of the 11 infected ulu trees, 7 had no signs of infection after treatment, 1 had a reduction in symptoms, and 2 showed no change in infection level. I ran a paired t-test for the ulu as well, and my p-value was 0.0002, which is also highly significant. Then I wanted to see if there was a relationship between a tree's initial infection and its final infection after treatment. To do this, I organized my data in a chi-squared contingency table, and this shows the observed frequencies of 2 different variables. So here's my table for kukui. The rows represent initial infection, and the columns show final infection. So I can see in the cells how many trees had a certain combination of initial and final infection. For example, if I wanted to see how many trees had initial infection of 2 and ended at 0, that's 8 trees. So it just helps me see how their infection has shifted after treatment. But then this graphic is a little bit more pleasant to look at, and it breaks this down into percent values. And so we can use this to compare how effective treatment is depending on the initial infection level. So for example, a larger percentage of trees with an initial infection of 1 got to 0, so we have 63%, compared to the ones starting at a 3, 31%. So it looks like it's easier for trees to get better with treatment if they're starting at a lower infection, which makes sense. But what's really exciting is that no matter the starting infection levels, the largest percentage for each one got to 0. So that suggests that this biocontrol was effective across a range of infection severities. Now here's my table for ulu. No ulu had a level 3 infection, which was great, so that's why it's not in the table. And then we see a similar trend here. So it's a larger percentage starting with a less severe infection gets to 0 compared to with a mid infection. And now moving into my ho'ina. Oh, I don't really want this sound. Moving into my ho'ina, I wanted to go back to my research question, which was, does the nematode species Heterohabditis indica serve as an effective biocontrol for kukui and ulu trees? And so yes, our findings show that it is an effective biocontrol. Across all treated trees, no infection levels increased, and a paired t-test confirmed a statistically significant reduction in infection following treatment. These results are especially exciting because this is a very preliminary study, and we've been refining our protocols all summer. In fact, a couple weeks ago, we discovered that our nematodes were accompanied by another scavenger nematode that is not as good as hunting for these larvae, and so it's known to be far less effective. But the fact that this treatment still worked despite these challenges and our limited background in this subject is pretty remarkable. It's been incredibly exciting to contribute to brand new research with the potential for lasting impacts throughout our Kai'ulu, especially for my home community in Puna, where QOB has already devastated so many trees. As QOB continues to devastate forests and threaten other species, nematodes could be key to early intervention before more is lost. Unfortunately, we didn't have this treatment soon enough, and the Kukui populations at the Keaukaha Military Reservation have already suffered a significant decline. But hopefully, through our research and sharing of ike, other species can be protected from QOB before it's too late. And in the near future, we aim to get certified to distribute nematodes, empowering community members to treat trees on their own aina and help control QOB. So please keep checking your trees and let us know if you need nematodes soon. I've already begun working with several organizations and individuals in Puna. And I'm excited to equip my community with ways to fight this invasive species. Then my haina. Before working with Likona Pulina, I didn't have experience in forest ecology or biocontrols. Now I have not only gained valuable skills, but have discovered new passions. Who knew I liked nematodes? This summer was the first time nematodes have been researched as a biocontrol for QOB, and I'm incredibly honored to have contributed foundational protocols, data, and observations. It means a lot to know that the work I've done can serve as a starting point for future studies, and I'm excited to see how this project continues to grow. I leave with way more questions than answers, but that's what makes this work so exciting. This project has shown me that research becomes even more powerful when it responds to community needs. What began as a project to save the trees in Likona Pulina's long-term forest restoration site has already begun to reach the rest of our moku, supporting farmers, laymakers, and people trying to protect the trees in their own backyards. Knowing that QOB first appeared in Puna, where I am from, makes this work especially meaningful, and I'm grateful to have contributed to a project that directly benefits my community. As invasive species like QOB continue to impact our islands, effective management will require collective effort. By combining research grounded in cultural knowledge with community action, we can help build healthier, more resilient ecosystems, and in turn support the well-being of our kaiāulu. Reflecting back on the summer, I can see how much I've evolved as a scientist and as a person. What I've learned through our huaka'i and my time with Likona Pulina has set the foundation of the biologist I hope to become. I'm especially grateful to have had this experience before applying to grad school because it has given me a much clearer vision of the work I want to be doing. Oh no, I lost my train of thought. I'm especially grateful to have found this because although I'm still trying to figure out the exact area of study, I now have a much clearer vision of how I want to approach my work with intention, grounded in culture, and with a commitment to values that truly matter to me and my kaiāulu. And then huge mahalos to my mentors, Becca and Emma. It's been so much fun working with you guys to problem solve and help to save our trees, and I've learned so much from you. Mahalo nui to my family for always supporting me, and thank you to the PIPES staff for making this summer such a meaningful and enriching experience. And to my fellow interns, thank you so much for all the memories we've made the past 11 weeks. And lastly, mahalo nui to my funding sources. None of this would have been possible without your support. Thank you.
Awesome, awesome job, Lila. And I especially appreciated the warnings before the videos. Those were a little rough for me. Do we have any questions in the audience about any of the amazing work that Lila has shared with us today? Seth.
So if one of the trees are past the point of like no return in some of those 3 levels and they stay at 3 level, do you have like a protocol to like cut it off or just keep treating it, or yeah?
Well, let me see if I can find the graph. So none of our trees actually stayed, and oh, that's not true, this one did. Okay, sorry. There has been, I think that we had 11 trees die throughout this summer, but we still treated the tree even after it died to make sure that no larvae were in that tree. So that's our protocol. We don't really cut anything down at the site, but we keep treating it until there's no more signs of infection. So yeah, that's our protocol. And a future study that I am really interested in is seeing where that point of no return is, because this was just like very preliminary, does this work?
Thank you.
Other questions? Ooh, from Dad.
Great job, Lila. Yeah, you're right. This leads, it's really exciting. It leads to a lot more questions, yeah. And one thing I'm thinking is, when you inject into the tree, does it have to go right on, do you think, and I'm not even sure you'll know the answer, does it have to go right on to the larvae, or can the nematodes find the larvae if they just end up in the tree? Can they like swim up to it and seek it out?
Yeah. That is another question that we're interested in. The nematodes are so teeny tiny, so we're not thinking that they can travel all that far. And we want to give them the best opportunity to get to the infection, so that's why we go straight into the infected area. From just like my observations throughout the summer, we've had trees that have low down infections that we're able to inject right into, and then they have higher up infections, and those infections look like they continue. Like the down low ones will end. So I'm thinking that they can't actually move that far. Yes.
Follow up. Do you think later they could maybe be sprayed on the tree possibly and then find their way into the holes? Is that a possibility?
There's been studies that I've read that looks at what is the most effective way to get nematodes into a tree, and they've tried spraying it, injecting it, I think like putting it in its roots, or plugging the holes with like a sponge that has nematodes in it, and they found that the spraying wasn't nearly as effective as injecting it into the tree, because the nematodes already have to find their way to the larva, so we might as well get them in, but then it does make the process way more tedious. But yeah, that would also be interesting to see, but other studies, it looks like it's not as effective.
Any other questions for Lila?
Kiana? Oh.
Do you think this protocol would be applicable to other tree species that are like damaged by QLB?
What was the last part?
Damaged by the queen horns long beetle.
Oh yeah, this could totally be used for other tree species because it's also affecting cacao and avocado. So this could be used for anything that's infected with QLB. Emma has a question.
Wait, but then the people online can't see that.
Awesome job, Lila. You're such a star. What should we do next?
Oh,
what should I do next?
Well, I know that you want me to get my PhD in nematodes,
Yes.
but I don't think that's going to happen. I think that what I'm most excited about seeing is outreach. Like, I'm excited for this Saturday. There's an event at OK Farms where we're going to be doing a demonstration about nematodes. And I think that this is going to really take in a collective effort because, like, injecting trees is such a tedious process. I think it's really going to take the whole community. And the more people know what to look out for, then the more we can try to control this pest. I also just, of course, want to keep saving our trees at KMR and continue to treat them. And I think it would be doing more research to see like what concentration levels we need, because if we only need a few thousand, right now we're injecting like 80,000 nematodes sometimes into each tree. If we only need a few thousand, like we can keep our stock up easier, and it will also be faster, like not injecting it 8 times, you know? Yeah, I feel like there's a lot of directions that could be taken.
Mahalo.
Can we get one last round of applause for Miss Lila? And that is the conclusion of the first half of this morning's presentations. One more round of applause for all of this morning's interns. We actually have lunch next, which is ready for us outside. But before we have lunch, can I get one of our interns to pule no ka'ai? You can just be where you're at, but would anyone like to pule so that we can all eat? There's lunch outside. On the menu, we have shoyu chicken and tofu veggie stir fry. Mentors, invited guests, please go eat first. Interns, as soon as they're all done grabbing plates, you guys can go. And we will reconvene at 1 o'clock, if I'm not mistaken. well, kani kohola e kahi. There's himo ke kwana ikoi. And then right at 1 o'clock, we'll be starting with Mr. Kainoa Cam's presentation. Yeah. So if we could all settle in and get ready for the afternoon half of our PIPES Summer 2025 Symposium.
Okay, everybody, it is now 1 p.m. and I will be beginning my presentation. Okay. Ano ai mekiloha kako o Kainoa Cam ko Oinoa. Kainoa is my name, and I'm going to be telling you guys about my journey building peeling into the storied landscape of Hilea Ka'u, native plant species regeneration and invasive species control. I know we all just had lunch, so hope you guys is not knack attacking right now, and we're all nice and paying attention. So first, I'm just going to start off by introducing the ahupua'a that I was working in right here in the ahupua'a of Hilea, mainly around the pu'u of Kaiholena, which is a very important pu'u to the people of Ka'u. And then a little bit about the organization that I've been working for, the Nature Conservancy, which is a global organization that works to preserve different areas of nature. This unit, the Kaiholena unit, is part of the largest and most intact native forest in the state of Hawaii. It consists of around 2,000 actively managed and fenced areas. And very importantly, it is at the boundary of agricultural land below and the state forest reserve above. The TNC acquired this parcel in around 2002 and has been managing it ever since then, and mainly for watershed protection. And I said invasive species, but mainly a lot of work with removing ungulates. So that's kind of the focus on my presentation is ungulates, but there is plenty of other stuff that I was doing as well. And we also work with watershed partnerships such as Three Mountain Alliance, local communities, volunteers, national parks, all kinds of stuff like that. But just to reiterate, the 100,000 acre Ka'u forest is above the preserve. And so this is nice because it allows people to have access to the forest, and it is a very landlocked area with private land. And here's a quick map of the Kaiholena unit, the Nature Conservancy's preserve. So as you can see here, this green is fenced areas right here, and this is where we were working a lot, around the Hilea Kaiholena sides, around this pu'u. And then a little bit about who I am. My name is Kainoa again. I'm a recent graduate of UH Manoa in botany. Just this last May I graduated. I'm very interested in aloha aina, la'olapa'o, botany, agriculture, and now hunting and fishing. I spent 2 years working at Kapapalo'i o Kanewai, which shaped a lot of who I am as a person and has continued to influence me today. And currently I'm finishing up in the aina pathway of the PIPES program. So I'm going to start off by telling you guys a little bit about the ha'alele and a little bit about why these ungulates are such a problem and some of the things that we face in the preserve. So to start off, this is a clear example right here of a pig wallow. And so the Kaiholena unit is pretty wet already, but in some areas where it's pretty dry, it will kind of make big pools of water, which mosquitoes will come in and kind of breed and stuff like that. But more importantly, you can also see that there's no understory if you look around. It's all dirt. And this is from ungulates from their digging and burrowing and eating things like that. And this makes it difficult because native plants can be slow germinating or slow growing. And so it allows a perfect habitat for weeds to come in and kind of establish themselves. And so that's part of the pig activity, as well as here's a common weed, tibuchina, that we deal with a lot in the preserve. And this is an example of one of the weeds that could kind of take over and establish when the pigs will go clear the understory. Here's another example of some pig disturbance and pig activity. The pigs will often knock over any kinds of trees, native or not, and often dig into the trees and eat the insides. And this is an example of a hapu'u fern. If you look at the circled red, this was knocked over by a pig and the inside was eaten. And so these are just all these little disturbance events that the pigs are creating. They'll often scratch up against stuff as well and dig up roots. Here is a photo of how you can clearly see managed side versus not managed. The right side is managed with ungulate control and the left side is not. And you can see there is plenty of understory, all these little ferns and stuff, and on the left side, there isn't. So sometimes these are very clear boundaries. When we're checking the fence and walking along the fence, we can really see the difference in activity on the managed or not sides. And then also very importantly in Hawaii is rapid ohia death. And so rod or rapid ohia death can be spread very easily by pigs. And so if you look, this is just outside the fence line of our preserve. And this land right here is not actively being managed. And you can see all these brown, it's just plenty of dead ohia trees from rapid ohia death. That is likely spread by ungulates. So now this is the huaka'i section. I'm going to be talking a little bit about the methods that we were using to control ungulates. Here's a quick little video of the top of the preserve. It was a kind of a misty day, a little bit hard to see, but just so you guys get an idea of where I was working up there. So first and foremost, before we enter the forest, we always are deconning. We're always spraying with alcohol. For those of us who went to that huaka'i at Kuhuku Ranch, for example, if you guys remember when we sprayed the tires of the truck, that's a very common practice, again, in order to stop the spread of any sort of plant pathogens or anything like that. We're also checking the car for pests such as fire ants or cookie frogs, making sure we don't spread things, especially because we work at 2 different preserves, or we have 2 different preserves that TNC manages in Ko'u, the Konahema Preserve and the Kaiho'olena unit. And so we also have different cars and different shoes for these preserves to make sure that nothing is kind of spread in between. And so first and foremost, a lot of our job is ungulate control, like I mentioned. And so here's some pig photos right here, just showing regularly that we bait and check these traps and dispose of the ungulates. And so usually if an area is kind of not much activity, it's kind of dead, or we're not seeing any tracks or digging, we'll go ahead and move those tracks to a separate area and just try to close out those last pigs in the preserve. Here's an example of setting up a new trap, which we did recently. Another thing that we do at the preserve is hunting with dogs. When there are few pigs left in a certain region, we'll go ahead and run the dogs just to clear them out, make sure we get every last pig that's around the area. And a lot of what we do as well is fence maintenance. So we're working with various types of fence, including barbed wire, hog wire, deer mesh, electric. And all these things are regularly being checked, fixed, worked on. You know, it's rugged terrain, it's very wet up there. There's plenty of animals. A lot of stuff can happen to the fence. So we're constantly checking that. And part of what TNC does as well is TNC has its grazing license. And so there is active cattle ranching. That is, we're leasing the land to ranchers up there in the preserve. And so part of our job sometimes is we got to check the barbed wire as well. So this was a day I got to learn how to fix a barbed wire fence that 2 bulls had knocked over when they were fighting. That's pounding the fence post to come along, knocking it in, tying it around. This is showing a bit of how we fix some sections as well. And here's the cattle. Another very interesting thing that we do is, in order to continue the fence line, we use tarp. And so usually around areas that are prone to flooding, such as gulches and flood zones, we go ahead and run the tarp to continue the fence line. And this is so that it allows the water to pass through freely without allowing any ungulates or anything to come into the fence line. This is just a picture of tightening the tarp because it got dragged down a little bit. Here's some photos of this one day we went over to fix one of these poles in the ground for the tarp. So we used concrete just to hang it back up, pick up more slack on it. And again, another type of fence we have here is electric fence. This is mainly more for the cattle, but it's part of our job to maintain and check this out. So this was a day where we were fixing some electric fence over here. So another thing that we do, like I mentioned before, is removing invasive weeds in the preserve. So a lot of this is done through pesticide spraying as well as hand pulling and cutting. So here's a picture of 2 of my coworkers preparing to mix pesticide spray and take it onto the UTV. This is especially done in areas with like fire roads and stuff like that to make sure everything is clear, the LZ for the helicopter, just areas like that. And another thing we do is actually seed collection. So there's plenty of, again, rare native plants over here. This is the Nuku'i'iwi. This preserve is actually one of the few places on Hawai'i Island that has this plant. And so here's a bag in case the seed falls and no one's able to collect it, it'll fall into the bag. And we have partnerships with nurseries and local people who helped us grow these plants, and then we use them for outplanting. This is Maile and Olapa right here. But again, we're fortunate to have a lot of intact native forest in our preserve, and so again, yeah, this is part of our job. And then we also have these halepiha, or little kind of restoration plots. And so the goal with these halepiha is to sort of grow plants that can be harvested for the ka'u keiki and the ka'u community. So these are plants such as olona, mamaki, maile, sort of things like that. And when we go into this halepiha, we're checking the fence lines over there, cutting the grass, stuff like that. This is some kopiko right here that we had planted. This is an area where we had planted kopiko. And what we'll do is we'll use the tarp. This is a common practice, but we'll use the tarp to kind of prepare the area before we're ready to plant. So this is a little bit about kind of the whole INA section. So this is a little bit about how TNC works with the community and stuff like that. So when the Kaiholena unit was originally fenced off in 2008, the community was called upon to help. And so local hunters from Ka'u actually were the people who kind of came in and helped remove all of the pigs originally from the preserve. So 28 of the first 33 pigs were removed by local hunters and used to feed their families. And so from these initial kind of community hunts, TNC has kept relations with these people in the community, and then we have this ongoing program now where local hunters will come and use TNC to access the higher state land of the Ka'u Forest Reserve above. And again, this is important because the Ka'u Forest Reserve is landlocked by plenty of private land, and so it's difficult to get access to the higher state forest reserve. And this is what I had mentioned before. Many areas of the 64,643 acre Ka'u Forest Reserve are landlocked, but this unit allows access for community hunters. And again, a lot of what we're doing is for the watershed protection. A lot of these ungulates can sort of contaminate the water source. And this area, Hilea feeds areas such as Punalu'u and Ninole, and a lot of the water goes underground as well. So now this is my Ha'ina section, just talking about the future for me. So again, as I mentioned before, I spent 2 years working lo'i, learning how to grow kalo, learning that kind of stuff, agriculture. This is a picture of Kapapulo'i Kanewai, where I was working at on Oahu. And so Nature Conservancy in Pipes was my first time doing sort of conservation work. And I feel that I was able to get my foot in the door, get this valuable experience. And so now I feel like I have more opportunities in the future if I decide to pursue agriculture or conservation or anything like that, or maybe something combination of the two, agroforestry and stuff. And part of my personal project was learning about pigs and learning about hunting and stuff as well. So a lot of the stuff, the skills that I learned from working with TNC, I was able to take out into my personal life. For example, I was learning how to track very well at the Kaiholena unit. Because it's very wet and muddy up there, it's easy for the pigs to leave marks of where they've been as opposed to a more dry area. And so it was a perfect training ground for me, especially because there's a finite number of pigs in the fencing area, yeah. So we can kind of keep track of who's around there, how long ago the pigs were there, what's their gender, what's their size. So very interesting stuff, again, that I was able to learn through working at TNC. And I had never even seen a wild pig or smelled a wild pig, to be honest with you guys, up until this summer. But through some of this work, I was able to go out hunting on my own. And this was a big milestone for me. Through these connections that I made through Pipes and through TNC, talking story with people, I was able to take this out on my own. And yeah, I was able to produce my own meat. And a lot of this is about food sovereignty for me. Again, I learned how to grow kalo, grow crops and stuff like that. And so now part of this is learning how to make meat. And you know, it's true when people say, what are we gonna do when the boats stop coming? Yeah, how can we feed ourselves, feed our families, feed our community? And so this was just such a blessing to be able to go out and learn this sort of stuff on my own. And yeah, that's the presentation. Mahalo to my mentor, Lori Lorenzo, over there in the back. Woo! Mahalo to the TNC crew. Mahalo to the PIPE staff. Mahalo to all the interns for your guys' support. And mahalo to Bud Cobell and Kamuela Andrade for teaching me how to hunt.
Amazing presentation, Kainoa. Learned so much about food sovereignty. Aimekai a kind action. Do we have any questions from the audience for Kainoa?
After you guys retrieve the pigs, what do you guys do with the pigs?
Unfortunately, we have to dispose of the pigs.
How?
Well, we have to just kind of leave them around the preserve areas such as that.
Any other questions for Kainoa?
Hello.
Aloha, everyone. My question is about the rapid Ohia death. That picture that you showed, that's pretty dramatic, but that's pretty much around the whole island right now. So can you share with us what you've seen firsthand in terms of not only the rapid Ohia death, but the areas you guys have fenced off and maybe the results you're seeing within the enclosure where the pigs have been removed?
Yeah, so I mean, to be honest with you, I haven't been around very long. This is all sort of new stuff to me, as I mentioned with conservation. But driving up every day, and not to say that there is no rapid Ohia death in the preserve, in the managed areas, it's a little unclear sometimes how it's spread. There's like beetles, airborne perhaps, but it does make a big impact. And actually, Rosemary's paper was talking about it as well earlier with ungulates and how they will spread because they're known to scratch up against things, dig around, and so they can be a big spreader of rapid Ohia death. But as far as my personal experience, we would drive up every day and I would just look at all this out just outside the preserve. And I didn't really get a good picture of the tree line inside the preserve, but it is a very noticeable difference. I hope that answered the question.
One last question from our online audience. Kealo asks, what is the biggest pig you caught / encountered? And how did you feel when you caught your first solo pu'a?
I think the biggest pig we had caught in the preserve was maybe around 200 pounds. Pretty big pig. It took 3 of us and we could barely lift it up. And then with my own pig, what was the question about that?
How did you feel?
Oh, you know, it took me a really long time. This is a little bit about my personal experience. I went out stalking 4 days in a row because I was like, when I present, I need to show these guys that I accomplished something and I actually got the pig, you know? So instead of working on my story map, I was out hunting every night. But I went 4 days in a row, and every night I made a mistake, but I learned a lesson. And the last night, Sunday night, I pulled it together. I got my pig. Kamaha'o, my friend here, he switched with me. I was supposed to practice on Monday, and he saved me. And I was able to practice on Tuesday, and I got it all together, and I got my pig.
Amazing, amazing job. One last round of applause for Kainoa.
Mahalo, guys.
Up next, we have Miss Matlin Ua, who worked with Kumuola this summer.
Aloha mai kako. Can everyone hear me all right? My name is Matlin Ua. I'm a PIPES intern this summer working in the Aina pathway, and I was partnered up with Kumuola Marine Science Education Center. Today, I will be presenting my summer research project, Ai Aka'i'a. And I didn't even pull it up. There we go. Thanks, guys, for not letting me know. Okay, so this is my summer project. It's basically a baseline understanding of limu production and consumption in Kapola'o and Waiahole local i'a. But before we dive into the project, I kind of want to introduce you to my background and how I came to UH Hilo, how I got involved with pipes. So I was raised in Las Vegas, landlocked state, super hot, super dry. You're like, how would she get interested in the marine science? It was a blessing that I was able to be a part of this organization called Girls in Ocean Science, where a bunch of female marine scientists came together for this conference, and they would talk to middle and high school girls, which was me at the time. And I learned about what they did and the work that they contributed to, and I just felt this spark, and I was like, that's what I need to do. I know I'm wearing an SDSU shirt. I realized what tuition was. And so thankfully, I was able to get in-state tuition at UH Hilo. And this is where I am currently. I'm going into my senior year in marine science. And to be honest, I wasn't super involved with the marine science department. I would just go to classes and go home. But thankfully, Dr. Stephen Colbert and Dr. Steve Du reached out to me with an internship opportunity working at Mokupapapa Discovery Center. The girl in the middle, her name is Stella. She's a master's student, and she was studying microplastics and how microplastics affect coral metabolism rates. So I got a lot of hands-on work working with her, and Dr. Du actually encouraged me to apply for PIPES. And I was like, yeah, I've heard about them before. I just never applied. So I was like, why not? And thankfully, thanks guys, PIPES accepted me. And I was partnered up with Kumu'ola Marine Science Education Center. They're located in the Ilikupono of Honohononui and in the Moku of Hilo in the Ahupua'a of Waiakea. They're stewarded by the Kamehameha School's Bishop Estate Trust, and they facilitate activities for school groups, and they also have volunteer work days for the community. Zooming in a little bit more into Kumu'ola, they have 3 main local i'a. My project will be based on the first 2 locals. So the first one is Kapola'o. This is the one that's closest to the Makaha, which is the gate that allows pua and salt water to come into the local. This is a place where boys became men and gained kuleana to the local. The second local is Waiahole. It's the largest local on site, so it's all of that water that's in front of the house all the way until the trees in the back. It's also known for housing a hole. And then they have a third local. I believe it's around this area. It's Waiopio. It's the third local, but unfortunately it's overrun by invasive trees and California grass. It could potentially be an upcoming main local as sea levels rise. But again, just to remind you, my project will be on the first 2 locals that I mentioned. So kind of getting into my project, I wanted to bridge the gap of our understanding of our food production. So there are 2 questions that I wanted to answer. The first one being, how much limu is produced over a span of 26 days? The second one is, how does limu production and consumption compare to our i'a population counts? So how much, are we making not enough food? Are we making too much food? That's kind of what I wanted to answer. So what do we know already? Based on a previous study done by Skylar Chong, shout out, our local mainly has diatoms, and the most abundant being some variations of Melosyra, Oscillatoria, Actinocyclis, and some Pleurosyra. So we know that already. We just need to understand how much we're making and then how the interactions with the fish are occurring. So, so to get into my experiment, this is what we had. This is a continuation of Skylar Chong's preliminary study. Basically what he did is he deployed settlement plates, and they are paired up. If you look closely, one of them has a cage around it. This is to prevent the grazers from eating the limu, to let the limu grow. The other one is open to allow for grazing. So just to recap, we have 20 total sediment plates. 10 are covered by that wire, 10 are uncovered. And they were paired up, and then the groups were placed into Kapalaho Loko'i'a and Waiahole Loko'i'a. We placed them on pohaku that had visual limu growth. And we also wanted to make sure that the plates were not being exposed as there were tidal changes. So those were some of the important things that we had to take into account. And this is day one of us deploying the plates. It is a little hard to see the plates, but they're all in a single straight line. And then now we could take a look at how much growth happened visually over the span of the 3 weeks. So this is day one, the day we deployed it. Week one, oh my gosh, week one. You could see a little growth happening over there. Week 2, and then week 3 for Kapalaho. Moving on into Waiahole, day 1. Week 1, not a lot of growth on here. We're thinking that it might have been because of a really severe tidal change that happened. Week 2, we can see that growth happening. You can see the difference between the covered cages, which is the darker ones, and the ones open to grazing, the lighter ones. And then week 3. So while we were letting those plates marinate, we also had some other duties that we were able to do. So we were able to clean the makaha, the gate, check for pua, and then sample and count if applicable. We also set up site if groups were scheduled, and we facilitated activities for those groups. We also moved mud, super fun, to kind of level out the land. We also cut and removed invasive California grass, boo. And we also had some experience with some cane spiders. I had it crawl all over me. It was something. But yeah, that's just some of the things that we did while my plates were marinating. So now came day 26. This is the pulling day. So I created an SOP so everyone knew exactly what to do and how to do it. And basically what we did is we set up stations where we would have the 2 pairs were on the same table with 1 liter Nalgene's and a toothbrush. And what we did is we pulled the tiles, we cut them from the cinder blocks, we placed them into the pans. You can already see the color difference is super insane. But also the type of limu seems to be different. So this top one up here seems to be more orangey or brown than the bottom one down here, which is a little bit more green. We scrubbed the top of the plates, rinsed the top of the plates out, and then poured them into the 1 liter Nalgene's, and then we labeled them. And you can, again, see the difference between the uncovered plates and then the covered plates. And then what we had to do is we had to take it to the analytical lab to filter, dry, and combust these samples. So that's a little selfie of me. Behind me is the setup for the filtering process. So basically, it would just pull all the water that was in those Nalgene's, and so it would just be the limu that was left over on that little filter plate thing. And that's what that looked like. So this is before drying, and this is after drying. The color changed a lot. It got a lot lighter because all of that water and moisture was removed. And then what we did is we combusted them, and we wanted to do that to take the ash-free dry mass, which is basically all of the organic matter, and they just burn it off to compare the before weight and then the after weight to see how much limu was present on that filter. And then this is what we got. I hope you guys can see in the back, but the important part to look at is these 2 columns right here. So how much limu we produced in the entire local is about 5.8 pounds. It seems like quite a lot, yeah? So a main thing to be aware of is that the production rate is above the consumption rate, which means that our fish are not eating more than what we can produce, which is good. And then we were also able to calculate how much percent benthic production was consumed by the fish, and that's about 44%. So super interesting data. It was a lot of crunching numbers. Thank you to my mentors for helping me and giving me a break when I needed it. But basically, to answer question number 1, how much limu is produced over a 26-day period, we have the answer of 5.8 pounds a day, which is quite a lot. And then I also created a graph in R. And this basically shows the ash-free dry mass, so what we combusted, compared to treatment and location. So on the x-axis, we have location, and then on the y-axis, we have our ash-free dry mass numbers. I ran a ANOVA, a two-way ANOVA, and what's really stood out to us is that we had a highly statistically significant value when it came to the difference in cover. So of course, we're gonna see a lot more limu growth on the covered than the uncovered, but as well as location. The locations are extremely different, which was super interesting, and I would love to look into that more. But something else that's important to note is that Waiahole had a lot more grazing than Kapala'o. I'm not too sure why. Maybe it's just the abundance of fish is more prevalent in Waiahole. And then to get to our second question, how does limu production and consumption compare to our I'a population counts? And the answer to that is the local is producing enough food for our grazers, which is good. But we need to make sure and be aware of our consumption due to the amount of the grazers that we have in our local. So looking at this graph, which I also created in R, we can see on the x-axis the type of species, and then depending on the color, it shows the type of, I've put trophic group, the type of food that these I'a eat. So ama are herbivorous grazers, so there are also our most abundant fish with about 0.208 average per meter squared there. So it's really important to know how much food we're producing so our amas can be healthy and not be all skinny. We don't want that in the local. And I know that was a lot, and thank you for bearing with me. I know we just ate, it's all digesting. So some key takeaways that I want you guys to remember is that Kumuola produces about 5.8 pounds of limu per day, and then the I'a are not eating more than what the limu can produce. But we just gotta make sure we don't fall into that alternative state, so we should keep monitoring and doing extra studies based on that. There were a few challenges that we encountered. The first one being the samples were so thick. There was so much limu growth that originally we wanted to filter 500 milliliters, and we had to kind of alter our volume because it was so thick that the filter, it was just getting stuck. It was dripping. It wasn't fast at all. So that was our first challenge, but I was super thankful that Skylar was there to kind of help me through that because I was freaking out. And the second one is that I forgot to weigh the original metal pan sizes. So that had some issues with our calculations in the end, but thankfully I saved them and we're able to go back and weigh those. But yeah, just to get into the broader impacts, look, the local I'a is a super important place for stewardship, but also for education for the next generation. And I learned so much about the history and the culture and the places I visit and the importance of place-based knowledge through this internship. And with Kumuola, I've become more well-rounded. Although I'm in the Aina pathway, boots on the ground, fins in the water, I was able to kind of get my hands on a little bit of everything. I worked with the community, I did my own research, I listened to my na'au a lot, and I was just super thankful that I was able to learn these different skills, like using hand tools and ho'oleu pena. I'm pretty good at it now. Kamaile, we can compete later. But yeah, so after this internship is through, I would love to just stay in touch with Kumuola. Just because I have that connection to that space, I just want to give back to what they have given me. I also plan to take the skills and knowledge that I've learned and apply that into my senior thesis in the fall. Yeah, and I just hope that I'm able to be a good role model of what a steward to that Aina is. And I'm immensely blessed to be able to have this connection to Kumuola, and I hope that sufficed. But yeah, I just want to say mahalo to Kumuola, to my mentors, to all of the senior research aides that helped me out, and Skylar, and also to my cohort. Thanks, guys. And if you want to connect with Kumuola, here's a QR code that you can take to their website. Does anyone have any questions?
Do we have any questions for Matlin?
Go easy on me. Okay.
That was really great. My question is, why 26 days? Why not a month? Why not a few weeks? Like, what is special about it being 26 days?
Yeah, that's a great question. So, of course, I would love it to be longer, but due to the time that it would take to filter, dry, and combust all of my samples, I pushed it as far as I could with leaving those out to grow. But that 26 days is kind of where I had to cut it off, because then we'd be going to like Waipio and, our huaka'is and stuff. But 26 days isn't really a important number in that aspect. It was just the number that I was able to kind of push as long as possible before I had to pull them out. Yeah. Of course, I'd like to keep them out longer if possible for a longer study. Yeah.
Do you intend on doing another study?
Hey, that'd be great, honestly. Yeah, they're not gonna get rid of me that easily. I'll be around, pulling mud, fighting cane spiders, and catching fish. Yeah, that'd be great, for sure.
Do we have other questions for Miss Matlin? That way? Where? Oh, right.
Oh, no.
I had a question about, do you guys know what species of lemur that were growing on your guys' plates? And like if you guys ever noticed like the fish, or which fish in specific, like native or non-native, if you observe them eating said lemur?
Yeah, that's a great question. Okay, so to answer your first question, based on Skylar's previous study, and I also did a little bit of I looked at the microscope and tried to ID, but honestly, I'm not super confident in my lemur identification skills, but I can for sure say that we had a lot of Melocyra present in those samples. And then we also had some Pleurocyra too. So those were the two most abundant that I saw when I was looking at my samples. And then this answer to your second question about the type of fish, I know for sure that the ama-amas are definitely the ones that graze the most on the lemur. But then looking at that second graph, this one, we can also see that the condas are also herbivorous too. The grass carp are as well, but they mainly feed on the California grass. They were introduced to kind of keep the border from them closing in. But it's mainly the ama-ama that we see eating the lemur.
Yeah. Any other questions? If not, can we get one more round of applause for Miss Matlin? Whoa! And we will transition to Mr. Ho'oli Kui Yakman next, who is a returning intern with Pohaku Pelemaka.
I'll change it. Thank you, Auntie Layla, for that lay. Well, aloha mai kako. My name is Ho'oli Yakman. I am a current UH Hilo student. I'm going into my senior year, aiming to get my degree in Ha'awina, Hawai'i through Kahakaula and anthropology. I'm also a second-year PIPES intern, and I did the Mo'o'aina internship as well. and I was placed within the Kaiaulu pathway this year. And I'm here to present my project entitled Hana'a No'eau, Multidisciplinary Approaches to New Education. So before I get into it, just a little bit about me. I come from Puna here on Hawai'i Island, as well as Makaha on Oahu. So I'd just like to first honor the people and places who have shaped me into the person I am today. Oh, sorry. Anyways, friends and family. Oh, and lei. I love to make lei. Okay. This summer, I returned to my OG host site, Pohaku Pelemaka. Shout out to them in the back. They're a nonprofit organization based in Lower Puna. My mentors are Auntie Leila Kealoha, Auntie Makani Greg, and Uncle Mikey, who are all in this room. And Pohaku Pelemaka was formed in 2013, and their mission is to essentially protect the vital qualities that make Puna Puna, as well as supporting Native Hawaiian well-being while passing down intergenerational knowledge. And so allowing this to guide our goals for summer of last year, 2024, we really focused on implementing practices that safeguard our new. And so we established a partnership with New Now, and through them, we're able to learn how to conduct ethnobotanical surveys on our coconut trees. So just getting to know like the morphology and then working towards eventually establishing a coconut, the very first coconut database for Hawaii. And then, oh wait. Because of the threat of the coconut rhinoceros beetle, better known as the CRB, and in our starting of our own learning journey of everything coconut, we needed to tackle our mulch piles in the grove. And so these piles, because these piles are breeding sites for the CRB. And so one of the goals last summer was to implement that and to eventually reduce these habitats completely. And then in the future, thinning out some trees. So again, just reemphasizing that these practices are efforts towards my site's long-term goal of returning the families of Puna back into these spaces, which have sustained for many generations. And so with this in mind, Kalei Kukui and I, who is my intern partner at this site, we allowed this to be the foundation of our project, and that is community. And so we felt that there was this koleana to Awamo. And so by the end of last summer, our perspective shifted. This wasn't just any other project, but this was a part of our long-term commitment to this place we also call home. And so we did this through the Mo'oaina internship program, also under PIPES, as I mentioned, which allowed us to continue our surveying, which is super important. I'm going to repeat again in establishing that database for Hawaii. And one reoccurring theme from the start of this project all the way up until now and looking forward was community. And so we do these surveys not just to establish a database, but with the intention of later bringing that back into the community and sharing it. And so committing to this long-term project also meant continuing to establish and strengthen those connections we already have within our own community. And so Kalei Kukui and I realized just how important it is to spend time in the grove, essentially with the Aina, because when we do this, we allow the Aina to speak to us, creating a connection between us and the most prominent kumu there is in that space, the kumu niu, and eventually uncovering its various functions. And so this is why it's important for us to further our connections with our immediate community, because this is how we become a stronger Kaiaulu. And so we do this by creating those spaces through the Ho'ovehe'i Canoe Camp, held annually every summer. And it's meant to not only bring the community together, but to create a space where all three generations, from Kupuna down to Keiki, can pass that intergenerational knowledge down. And so here's a video of Auntie Leila speaking to the students of Ahalamaku Oya I'o of Pahoa High and Intermediate. If it can load, but it's okay. Oh, right there. We've also been letting the community know that we are here, actively stewarding the area by posting up signage throughout the Grove, which is also reminds our visitors and community members how to conduct themselves while in the Grove to respect the area, leaving it better than you found it. And so because we've been working towards strengthening our own kaiāulu, we also found how important it is to do the same beyond. As I briefly mentioned, we established a partnership with New Now in the summer of 2024. They're a cultural agroforestry community organization based on West Oahu. And so through these partnerships, they can help expose us in different ways and teach us different ways we can show up for our own community. And so because we're able to lu'upiha I kahana or dive deep into the work, these are some of the things that we're able to accomplish. First being establishing our survey methodologies. This provided a clear, consistent, and reliable framework for our surveying process. And then secondly, because we established these methodologies, we noticed how much we grew in our efficiency. And so we saw that we were doing like 2 pages worth of surveys in one session, whereas last summer it was maybe 20 trees a week. So this is some of us in these pictures. And then the last accomplishment is increasing our mulch management efforts, which with the intention that we're able to return the nutrients back into the sandy soil of Ka'akepa, but also serve as a safeguarding protocol in a way for the CRB. So yeah, a little bit more pictures. That's New Now and the ohana from, I believe, Onomea. Yeah, over there, working on a trench. And so let's not forget why we're doing this work. It's for the community. And so as a nonprofit, we felt like it was time to begin to reintroduce the new or the coconut back into the community. And as we began to reintroduce communities to new, we realized we weren't just communicating to one kind of community, rather many communities from various backgrounds. For example, we had a visit of teachers from Alaska who aren't surrounded by new, and so having to communicate what we do, things we've learned in our journey, and what we see made me view new in a different way. But of course, we had visits from local students, so this also meant communicating and sharing new in both cultural and non-cultural lens. And within these workshops, through informal talks, thank you. And so within these workshops, we're also using posters given to us by New Now and Dr. Rowland. Dr. Rowland is a coconut scientist from Tahiti, and his poster displayed a wide range of coconut shapes and colors, and New Now explained the reproductive process for the new. And so seeing the impact that these materials have when used at the same time as these informal talks have had, was super impactful. And moving on. And so another thing that Pohaku Pelemaka provides each camp participant at the Ho'ovehe Canoe Camp is this interactive workbook. And this workbook includes the Mo'oku Auha worksheets, which are customizable, and a bunch of resources such as Hoku charts, cloud charts, kilo logs, and countless oli and mele, which are meant to supply the community with ike kupuna. And with our newest edition of the Hepule Niu. And so feeling inspired by the work that has been done before me, I felt called to look deeper within myself to see how I can provide for my own community the same way they're doing for me. Because we were able to establish our survey methodologies, I was able to put some sort of imagery together for our survey methodologies. And so this includes 7 out of the 13 categories that make up our surveys. One thing about the images that you're currently seeing is, with an exception of a couple, is that all of these are colors, shapes, and coconuts that you can find all around Ka'akepa. And so I created this in hopes for it to be an ongoing piece of educational material to be used within these workshops, but also as something that is a direct reflection, like I mentioned before, of the varieties you will find in Ka'akepa and perhaps their unique functionality. I also envision for this to serve as a guide for learning how to conduct surveys on the coconut, which provides a detailed explanation along with a cohesive visual for learning. And so it's not about reinventing the wheel, it's simply about using the tools that we have already to engage with the community. Speaking of tools to engage the community that I have not included in the story map, but we are actively doing, is bringing life back into the social media for Pohaku Pelemaka. And so did this through the collaboration with Auntie Christy Barrow, who is a longtime Puna resident. And we were just kind of communicating and trying to work towards creating a content schedule for the social media, both Instagram and Facebook. Shameless plug, go follow Pohaku Pelemaka on Instagram and Facebook. Thank you very much. Okay. We prioritize building relationships with our community effectively, and the way that we're gonna do this is to provide a wide range of content, from community events to trivias to fun facts about Niu. We simply just wanted to bring life back into the social media. And so to bring it all together, sorry. Leading into our Mai Ho'ina, community has been the foundation of why we do what we do. We recognize that in order for you to grow within your own community, you must also look outward. So that's exactly what we did, creating spaces where we can connect with one another. And so we are still learning all there is to know about coconut and Niu. So it's not about what you don't know, but about what steps you take to keep learning and deepening your connection with it. And we did this by involving the community through informal talks, poster showings, and developing potential curriculum for future workshops, with the intention for it to grow and expand while remaining as a direct reflection of the varieties you will find not just in Ka'akepa, but eventually Puna and in the future for Hawai'i. This experience has taught me that small steps can lead to big changes over time. And for us, committing to this project from last year all the way up until now and even into the future, I hope to be able to continue these surveys. For future projects in Ka'akepa, this will hopefully re-emphasize why it's so important for researchers and data collectors to develop personal relationships, not only with these places, but also with the community, because this is how we build that trust. And as a haumana at Olala Hawai'i at Kahakaula, I hope to finish off my last year at UH Hilo this coming fall, and returning and bringing, maybe perhaps turning that guide into a catalog. And so with that, I would like to thank my mentors back there, Auntie Layla, Auntie Makani, Uncle Mikey, my ohana for supporting me on this journey. And to the funders, thank you for the money. Thank you.
Amazing job as always, Ho'oli. Do we have any questions for him?
Yes, Auntie Layla. Oh, weeks, okay.
Where are you going to be in a few weeks with this printing project that we have going on within the data collection?
Okay, the question was, where are we going to be in a few weeks with this data collection? Well, hopefully, and Kalekukui will talk a little bit more about it, but in a few weeks we're having Dr. Roland, who I mentioned earlier, the coconut scientist from Tahiti. He will actually come and help us figure out who is in the grove. So he's gonna help us identify the varieties.
Other questions for Ho'oli?
Hi, aloha. That was awesome and educational. I wanted to know, do you know how old the grove is? Is that known?
Through the talks that I had with my mentors, they shared with me that a lot of the trees you see now were planted, I believe, in the 40s or 50s, like the early 1900s, around that time. Yeah. Oh, and they were planted by the Conservation Corps, I believe. Yeah. Paul? Yay.
Can we have one more round of applause for Ho'oli? Ho'omai ka'inui. And up next we have Mr. Peter Kalekukui Watson. Also a returning Mo'o'aina intern with the PIPES program, also working in Pohaku Pelemaka. Take it away, my friend.
Aloha mai kākou. My name is Kalekukui Watson, or Peter, but I go by Kalekukui. And I am a second-year PIPES intern this year. I also did the Mo'o'aina internship. And my title of my presentation this year is called New Opportunities, Seeing New Through a New Lens. Hello. Just like Ho'oli, I'm partnered again with the OG host site, Pohaku Pelemaka. My mentors are Uncle Mikey Kaiser, Auntie Makani Greg, and Auntie Lela Kealoha, all right back there. And as you've heard, they were formed in 2013, and their main mission is to protect, perpetuate, and enhance what makes Puna Puna. So, looking back on my experience last year as a first-year PIPES intern, I would say the Ho'ovehe'i Canoe Camp was my catalyst to keep this surveying going. During that camp, we have a whole bunch of ohana of the area and other nonprofits that we're pili with come down, and we just teach them about the importance and the different uses of niu. There's hundreds and hundreds of uses. But these are just pictures of the ohanas and community coming down, doing things like weaving baskets, going out and collecting niu for apu, and yeah, things like that. Keiki action, baby sukupuna. And this is Injit from Niu Now. Huge, huge help. Without him, we wouldn't be able to do anything with our niu. Yeah, so in preparation for this year, or not in preparation, sorry, the methodologies that we're using is mainly our surveys to identify varieties. So when we do niu surveys, we collect data on 13 different characteristics on the tree. And the reason why we collect data on such a wide range of characteristics is because there's little to nothing known about the niu of Hawaii. And as you've heard from Ho'oli's presentation earlier, we plan on using this database and methodologies to help kickstart the surveying of the ulu niu and niu across Hawaii. We're also going to be using this database to find out where the varieties are within Ka'akepa and to see how much we have, which will be done when Dr. Rowling comes. But besides surveying, doing things like sending the ohanas out to go look for different shapes and sizes of niu during the camps has proven really helpful in finding different varieties. These are pictures from last year. We sent them out with all the keikis to go look for different shapes and sizes, brought them back, sorted them all out into similar shapes and sizes. And then we cut them open to take pictures and catalog them. As you can see, some vary, or they vary a lot in shapes of the shell, husk as well as thickness of the shell and husk. And this picture is from last year. After the ohanas came back and we sorted it out, we found 10 different varieties in Ka'akepa. And if there's 10 in just Ka'akepa, imagine what there is throughout the Painaina. And onto my huaka'i. So exploring the uluniu, or gallivanting around searching for apu, has become one of my favorite things to do. And in doing this, I actually found a rare-ish type of variety, this dark, pointy, shiny one right here. And after finding out that was a variety, that sparked me to go look for more pointy ones, and ended up actually finding a few more trees, which will hopefully ensure this variety's future. Sorry. But besides my gallivanting, surveying is the main thing that we're doing. And as shared earlier, this is about an average or half an average days of work now. That would have taken us about a week last year. So we've been trying to become more efficient at new surveys. And that has really happened mostly during the Mo'aina part of our internship. We were balancing working at school, going to school, and going all the way down to Ka'akepa. We'd go down to Ka'akepa usually every weekend, mostly Saturdays. Sometimes we go both days. So it was a lot of balancing everything, which taught us how to be more efficient. And as I was saying earlier, when we send the groups or the ohanas and groups to go look for different shapes and sizes, we find some pretty cool things. This is actually the new, or the choice new, for a kio'e. I don't know if that's the actual word, so don't quote me. But that's the apu used to serve ava. Finding pretty cool things, you just gotta look up, down, side, side. Because remember, coconuts kill more people a year than sharks. A little fun fact. And as I said earlier, one of the main objectives for this past year was to increase our mulch management efforts. So this is a picture of a giant trench that me and Ho'oli were digging. It took us about like 5 hours to dig this trench. But it ended up being like 5 feet wide and like 3.5, 4 feet deep. But when we do mulch management, we dig trenches like this. This is just a big one. And we grab mulch from the piles all spread throughout Ka'akepa. It's just leaf litter and old coconuts. We chop out the big leaves, throw everything inside, try to maximize the space. And doing this doesn't only clean up the area, it feeds the trees. Because they're growing in almost straight sand. There's a big lack of nutrients. So burying a big block of mulch like this is just giving them a big boost in not only nutrients, but water retention, because sand. And it also helps get rid of potential breeding habitats for larvae like QLB, Queensland longhorn beetle, and CRB, the coconut rhinoceros beetle. But thankfully, we don't have CRB on our side of Big Island yet. So this is more of a precautionary measure. And into my ho'ina. As I was just saying, mulching does clean up the area, and seeing how much it cleans up the area, that actually like made us do a lot of mulching over this past year. It just makes the area look a lot nicer. If you were to come last year, this is what the average grove looks like whenever you look around. And this is what our pico looks like after mulching over here for the past year. Yeah, big difference. So seeing this space so clean and all beautified is just super inspiring, because if we continue mulching like this, the whole grove could look like this. But it's not about doing it once. You have to consistently do it. Also a fun fact about this. This is our Pico area. Last year, me, Kilipohe, and Ho'oli designated this as the Pico area just because we always gather here, and it's like the highest point on the beach. And this year, when we were flagging varieties that we found, 8 of the ones that we wanted to keep ended up surrounding the Pico. #now. And more into a personal reflection, the theme throughout throughout my time in the Mo'aina internship till now has been seeing Niu through a new lens. And what does that mean? Well, if you were to ask me last year when I got the email of what my host organization was, if I was happy, I would have said no. I was like, I'm gonna be looking at coconuts, what do you mean? Like, I wanted to be in the mountain with the ohia, and now, if you were to come to Ka'akepo, you'll see me running around with my bag full of unfinished apus, AKA my bag of opportunities. And I'm even thinking of collecting Niu based on my preference for apu. But yeah, all of these experiences I truly appreciate. Wanted to give a huge thanks to Pipes for letting us rub you guys this hard. And huge shout out to my mentors again, Auntie Leila Kealoha, Auntie Makani Greg, and Uncle Mikey Kaiser. And a huge shout out to the entire team of Niu now, but especially Indrajit Gunasekara. He is from Sri Lanka, and he grew up in a village that's in a coconut grove, and they've been living that way unbroken for over 2,000 years. So having him with us is literally a huge help. He's why we are able to do what we want to do. And he can answer almost any question we have about Niu, which leads us to potential thinning of our ulu Niu. We asked Indrajit if our grove was healthy, and to our surprise, he said no. It's actually kind of struggling. Looking at it, you would think it's healthy just because of how much trees and fruits there are everywhere. But as if we've said earlier, they're growing in almost straight sand, so they're lacking nutrients for one, and it's kind of getting overcrowded now, so they're competing for nutrients in a place that don't really have nutrients. We also plan on collecting seeds from the varieties that we find after Dr. Rowland comes down and helps us confirm these varieties. To start a new nursery in partnership with Omoku'u Kekohua, shout out Kilipohe. And we plan on planting these, and we're probably gonna plant some at Moku'u, but we're gonna be planting them in ways that the variety will stay true. So we'll be planting them in clusters of 3, separate from each other, so they get pollinated by the same variety. Because in this grove, there are lots and lots of hybrids due to the amount of trees and their close proximity. And yeah, I wanted to say mahalo again, mahalo to Pipes, and kipuka, all the funders, Ha'oli Maoloa Foundation. Thank you, thank you.
Great job. Oh, I already see a question in the back. Yeah, Auntie Layla. Thanks for sticking around for a second year. I didn't know that you didn't like your project in the beginning last year. That was news to me.
Now look at me, though.
Awesome job. How do you guys know where the trenches are?
Oh, yeah, we're mapping the trenches on Avenza, because since we can't just do this one time, we have to consistently do it. The next time we trench, we're gonna trench them in different areas. And one other thing is we're all making the trenches face the same way right now. Next time we trench, they're gonna face the other way and in different areas.
Are you able to tell the square footage?
Ooh.
Cubic yards. Yes, you are.
I'm like 2 giant Costco chest freezers.
Oh, that's not a question. Good job, Kalikukui. And yeah, I forgot my second question, but I do have another one. Yeah. I forget. I'm trying to remember what it was.
I have a question about how many opportunities you get.
Ooh. Girl, more than 30. After 30, I kind of lost count.
Do you have a favorite apu that you have finished?
Yeah.
Is there a punahele?
Actually, it's in my car.
It's an apu holo holo.
It's actually a white apu that I made from a young Niu that we found. Yeah, it's super cool to see a white apu. It's probably my favorite one.
Nice. Which of our interns had a question for you?
I had one, but it was basically I was going to ask where your bag was.
In the car. You can look at it after if you want.
How did you figure out that that new variety was desired for the Kioe?
It's just out of, it's not really scientifically backed. It's more of like cultural perspectives, and especially a big help from Indrajit. He said, or not he said, but seeing the shape of everyone's Kioes, the new that we found looks like almost the exact same. It's kind of like a new Hiva, just not as dark, but a lot fatter. So it's fat and elongated, not really round. Perfect for a big scoop. Yeah. One cool thing Auntie Makani is doing is she's trying to put stars on an apu. Ipu Hokel. Yeah. Cool, yeah. Oh yeah, Pohaku Pelemaka, follow them, please. So the question was, how much trees are we collecting data on, just for the online groups? So throughout the Grove, there's about 1,400 trees, which is massive, and we're kind of lacking on inputting our data right now, but I'm gonna ballpark we're around a little over 400 trees done. And to put that into perspective, last year, throughout the entire year, we did 100 trees. Yeah, and then we did 400 from last school year till now. Yeah. Mahalo.
We're running really ahead of schedule, and we get like 9 more minutes. So keep firing at them. Ho'oli, you have to go up there with him, they said. Glory doesn't go on until 2:20. We can't, like, disrupt her flow.
When you're doing the thinning, how many trees do you think you'll have to cut down?
That is a really good question. We'll find that out when we find out what varieties we have and where they're located throughout the Grove. And when we figure out how we want to thin and plant, because we're going to try and plant some in order to keep the varieties there. But when we finish the Grove, I'll let you know.
I also have another question. Was making trenches and burning them, was that like something that they did in the Wakahiko, or would they more just like leave the fronds, or would they make piles, or do you guys not know? Like what were the Hawaiians doing in the Wakahiko? Were they also making trenches and burying the fronds, or?
This I'm not too sure about. What we're doing right now is mostly led by Indrajit, but he's just trying to get us to reconnect because we actually have Hawaiian names of each stage of the fruit, and I think there's 10 stages of fruit, sorry. And it's all Hawaiian names, and Indrajit keeps telling us, like, if there's this much names for all the stages, if there's a new Hiva, if there's a new Lelo, there has to be a lot more out there. So he's just trying to implement things similar to what they do back in Sri Lanka here, and just to try and get us to reconnect to it somehow.
So the bury and burn is from Sri Lanka, or like that's what they do commonly? Oh, cool.
That's what they do when they can dig, and when the Grove is kind of struggling. There's big boost to of nutrients and stuff.
Cool, thank you.
Any other questions? Nino Maila. Just Leonui.
So what will this data collection be contributing to in the next 4 months?
What are we creating? Yeah, it's like going to be a manual to help the other people who start surveying to do the same thing and save the varieties they find in all across the Paiana. Yeah. Okay, pal, mahalo.
Can we get another round of applause for Kalei Kukui and Ho'oli? And up next, last one before we take another little bio break intermission, is Miss Glory Vehilani, who had the opportunity to work with Pilina Aina.
I'm glad we're ahead of schedule. I can yap. Let's go. How's it, everybody? I'm Glory Vehilani Freeman from Kai'ulu Pathway. Join me on my story titled Koa Hia, Koa Dela, Koa Everywhere, from forest to canoe. My hua, which I fondly titled, Hua's this sister? I like to describe myself as that typical Makai kid. Born and raised here in Hilo, I always grew up outdoors. I would go hunting on Saturdays, fishing Sundays, and always seen in the wa'a throughout the week. It taught me how to be self-sufficient and just appreciate Aina for everything it has to offer. The biggest part of my life is being a paddler. I wouldn't say it's a lifestyle, I would say it's my personality. I grew up paddling for years and years and years, and the time on the water really made me appreciate the sport and the kai itself. It pushed me into the direction of transitioning from a kinesiology major into marine science. And the coaches I've had growing up really inspired me to follow in their footsteps and create that next generation of paddlers. So I am a coach for the Kiki program with Kamehameha Canoe Club, my home club, and Waikea High School paddling team, which is my high school alumni. Yes, sir. My passion for coaching really inspired me to create this bigger picture of how it can become a career. And that got into the question of, what if I became an Aina-based educator? Which is how I ended up here in Pipes. Same like Matlin, it was actually Steve Du that pushed me in for 2 years in a row. And I finally gave in. I was like, okay, let's give it a try. Through Pipes, I was lucky enough to be matched with Pilina Aina. Hey, Alicia. Hey, Leila. They're a local nonprofit here in Hilo. They centered their work through the values of Aina as Ohana, Pilina, Kuleana, and Kaia'ulu. They run their program through the Lalono'ea Iola Oe, Iola Mako Ne. My life is dependent on yours, your life is dependent on mine. This really reflects their work as they create educational opportunities that build Pilina to Aina in reciprocity, stewardship, and the strengthening of our community. Going into the haolele. Nice title as well, Shutsuka Zango Haolele. It was a huge transition for me, and I had to think of it as finding the comfort in the uncomfortable. As a kai person, I had to trade in my fins for boots, my kai for the pu'u, and my paddle for a weed whacker. And I will admit, it was really rough in the first couple weeks. I struggled with that transition of not touching water every day and just grass and trees. But I've come to appreciate it more. I've been at the pu'u and listening to everyone's stories and just seeing the kids and how immersed they are. Like I said, I am a marine science major. I have no idea anything about plants. I can answer questions on the currents, on how to catch a wave in a vault. I can tell you all about limu and all the sharks in the area, but plants, all I know is roots, trunk, branches, leaves. So I was lucky enough that everyone I worked with is so knowledgeable. They are basically my walking encyclopedias. Every time I listen to them talk, it brought me back into those younger years in preschool and elementary, where you get to sit in the corner all around everyone and your teacher just telling the children books. And don't laugh. But that was my best memories, just like hanging out and listening and being in that story with them. And I realized that the people I work with aren't just conservationists, they are storytellers and educators. And I realized that storytelling is the key factor in my success throughout this internship. So from that, I realized what my goals are with this. It's 1, listen and learn. 2, build skills in conservation and education. 3, connect with others and grow my kai'ulu. And last but not least, bring in that kai element. Going into the Huaka'ian hostel. I had 3 main work sites. Of course, we gotta stop at the office first. Located at the top of Novello Street in the IPIF building. We are only here to do our paperwork, prep for our field work, clean the truck when we get back, and talk story. Next one is Pu'uhuluhulu Ranch and Farm. This is a time lapse that I took at the ranch with the sunset, and I told the kids, do not touch my phone. And they replied, no worry, Auntie. I'm not gonna touch them, but I like being them. Here it is. The ranch is owned by a Waimea resident and member of Kauai High Canoe Club, Uncle Kalani Kaniho. He invited Kuala Middle School Gero program and Pilina Aina to his ranch with the idea of outplanting native trees, particularly koa and mamane in the slopes of Mauna Kea. Keep in mind that this is a ranch, an active ranch with roaming cattle, horses, and it's on Mauna Kea, which the climate isn't best. So we had to switch up our idea of how to plant these trees to increase their survival rate. We did a method of drilling really deep holes and widening out into a well so water can collect in. We also use water gels that can hold the moisture in there. Weed mats, of course, to keep out the weeds, hence weed mat. And there was already existing fences around the plants, and that's just to keep the cattle out from munching on them. These are the awesome keiki I worked with. You wanna check them out? These are all the plants that we had. We did a total of 30 plants in a day with no breaks. This is Hunter cutting the weed mat, throwing on shaka. Right here, the keiki are prepping for the next batch that will be planted. This is us outplanting. and then the finishing touches of the weed mat. There was never any rest periods. Downtime was used for educational purposes. We went over the Mo'olelo of Mauna Kea, the Ahupua'a where the ranch was located in, which, come to find out, Humuola. And we also did some fun games so the kids don't get too bored. One of them was the native versus non-native species game, seen here. Now, the place where I was at almost every week is Puwawa. This is, hold on, right here. This is the core unit. It's a portion of land that was hanai to us, Pilina Aina, through Kaiulu Puwawa. It was named the core unit because of the existing koa trees that were found there, and because of that, they decided to outplant more koa in there. We also have planted ali'i and iliahi in there. I added this picture because I like it. This is my main work crew right here, Alicia and Kalani, cutting grass all day. We hosted 2 lauhana. The first one was at the Lanakila Learning Center through Hilo High School, and the second was the Kuala Middle School Gear Up program, which I mentioned earlier that I went camping with at Pu'uhuluhulu Ranch. We taught the groups on the Mo'olelo of the area, rot awareness, and our dry forest ecosystems, focusing on the koa itself and the restoration efforts on the pu'u. I was also lucky enough to do classroom visits for both of them, where I got to talk about further education in college and potential career pathways in conservation. I also did mention pipes. I couldn't not add our huakai to Puwawa, so I added Kaiulu Puwawa. Shout out to the Nehe gang. I hope you guys are watching. They're under the Kaka Foundation, and they cover 84 acres in the Pu'u, and the whole idea is to restore it to its native forests. When we went, we planted hundreds of trees, and that was the first time I worked there and it actually rained. It was pretty nice. So after all that hana, it was time for me to crack open the laptop and get to work. Connecting the uka to kai, I tried my best to create an educational booklet aimed for elementary grade levels. It included phases, puzzles, coloring pages, and then I did a makavalu on Puwawa itself, focusing on wa'a, just to add in my little thing. So we went over the parts of the wa'a. I also added a kilo journal and finished it off with oli ikumama. This is an oli that is near and dear to my heart. As a paddler, it speaks of them going up into the mountains and bringing down a huge koa log down to the kai where it will be shaped into a wa'a. This is a oli that my canoe club always uses whenever we bring out the wa'a from our halau to the kai on race day. So I also just wanted to share it with everyone else that will be able to get their hands on this booklet. In modern times, we hear this oli just to rally up everyone for a common cause. So it's not just about paddling, it can be versatile. My little side quest adventure was Species of the Week post on social media, focusing on the native marine life. I did one on limukohu with a poke recipe. Pretty ono. Mono kihi, I was able to get in touch with Danielle Bartz, who is a UH Manoa PhD student. She is doing research here in Hilo Bay on population of hammerheads. And ko'a, Shout out Kumu Steve, wherever you are, Hilo. Okay, it was time to stop, breathe, and reflect. Hence, enough already. The whole idea of this journey was to give back to the community and connect with them as well. My internship with Pilina Aina challenged me in the most comfortable, uncomfortable ways of being upland. However, through this journey, I was very appreciative to be able to contribute to conservation projects and outreach education. Definitely was a rough and bumpy road, but I did learn a lot. However, the questions that remained was, what did I get out of this? And what did I learn about myself? Here are the following. I can grow my kaiulu. 2, I can take on more than marine science. I always thought I would end up at a local IA, like any other local marine science student here, but I guess the forest is okay. Cutting grass ain't that bad. I'll be honest, I was very spoiled growing up. I never did any yard work. So this was my first time actually doing it, and you know what? It's not that bad. Would I continue doing it? I heard that. Maybe I'm not cut out to create curriculum, but I still love to teach. I also added, in quotes, the more you learn, the more you grow. It's something that I told both of my student groups. And it was also just a way to motivate me to just keep on continuing forward. Last but not least, Malka and Makai is interconnected, and we should treat it as such. I think in the conservation realm, we are still separate, but we need to understand that it works hand in hand, and we should all work hand in hand together. Ha'ina, go ha'ina or go home? I'm actually not going home after this. So we're gonna go ha'ina. Looking ahead, like I said, I learned so much, and it's no use to keep this to myself. So whether I end up teaching or just doing field work, I'm gonna continue to incorporate everything I learned from this past summer. And I also wanted to end off with what Mauri touched on earlier. We started off this internship with a really tough question of the kupuna you want to be. I like to think that my answer still stands, but it expanded in a way that I didn't think it would. I still want to be strong. I mentioned that strong in a sense that I was still able to paddle the Kaiwi Channel when I'm 70, but strong enough to continue cutting grass. I want to be open-minded, and the whole idea with that is just listen, learn, and then act. I want to be welcoming, keeping my door open, and it's not just for the ones that are already established in my life, but the new ones that will come into my life later on. Last but not least, I want to be humble but not quiet. The whole idea for this one is speak when needed, but not just to be heard. Okay, one mahalo, then we pow. Huge shout out to the pipe staff and my fellow pipers. My mentors, Leila, Alicia, the rest of the Pilina Aina staff, AFTF, all the schools and programs I worked with this summer, Puwawa itself, Mauna Kea itself, and last but not least, oh, wait, go back. Is there an easier way to do this? There. My pali ohana, who motivated me to getting to this point today. And my sponsors. Mahalo.
Round of applause for Glory Vehilani. Questions.
Awesome job, Vehi. Proud of you.
Thank you.
You are more than a weed whacker, my girl, for sure. And ahoy va'a, you're all the things. Can you share a favorite Aina moment that you had? I know that you've shared some feelings you've had in a va'a. What would be your favorite Aina moment you had?
Okay, yeah, I'm a paddler at heart, so we're gonna go with the va'a story. Crossing the Ka'iwi Channel.
Okay, now I'm gonna push you. What about in the koa unit or at the pu'u, in this new ahufa'a, in this new space, different?
In all honesty, it was just our weekly maintenance day with our crew. My most memorable moment is when me and Kalani were just lying on the grass and you were telling your children's book story to us. I wonder if I have a picture of that in
Don't
here.
you dare.
I don't
No, I'm
know.
kidding.
I wish oh, this one.
Yeah.
This one, okay. Right here on the top, that's Kalani, that's me, and we're just like full on laying down, just being immersed in Alicia's words.
You're welcome. Thank you for sharing that. And then can you share a favorite moment you had taking on that kuleana of being a kāko'o as a kumu in some of the huaka'i we went on with these students?
That would be at the ranch. That's when I spent the most time with the kids. And that's the most time I ever heard Auntie Vehi, Auntie Vehi, Auntie Vehi. I think I understand Anake Mauri'i now. But just being able to like talk story with them and them being so comfortable to come up to me and ask questions, it was definitely worthwhile. Thank you.
Any other questions for Vehi? If not, can we get one last round of applause?
Thank you.
We are on break for the next little bit, 20-ish minutes. Interns, if we can get you guys back in here by 2:55. 2:55. Aloha ho mai kako.
If you can hear me, put your hands on your head. If you can hear me, clap your hands twice. Thank you for participating in what I used to get my kids' attention. That was really awesome.
And with that, we will get ready to start the last section of PIPES Symposium presentations for this afternoon. And starting us off in this last block will be Miss Kahinu Hayashida, who interned with the Sierra Club of Hawaii.
How do I get it on the screen? No, you're good. Hello, hello.
Oh, she's on. Okay, awesome. Okay, aloha pumehana kako. My name is Kahinu Hayashida, and this summer I was a part of the Kaiulu Pathway working with Sierra Club. This summer, my project focused on decolonizing decision-making through kapa'akai analysis, honoring Native Hawaiian rights in land use decisions. Okay, so who am I? I was born and raised a Hilo girl from Waiakea. Growing up in Hilo was truly a dream. I was constantly surrounded by my family and completely engulfed by the aloha of my community. Being surrounded by that feeling is a privilege, and that's something I try to bring with me into any space. I grew up surrounded by aloha, so I try to bring that with me. Fast forward elementary, middle school, and half of high school, and in July of 2019, I found myself as a junior in high school, sitting on the Mauna Kea access road alongside my sisters, kupuna, and strangers with a common goal, protecting our mana. Prior to this, I had never envisioned myself in paths that had to do with advocacy or grassroots activism, but that day it all changed. So 6 years later, here I am. I'm a new graduate from the University of Hawaii at Hilo with a degree in sociology and a minor in political science, eager to jump into the field but don't know where to start. Thankfully, through PIPES, I was able to work with the Sierra Club this summer under my mentor, Sharday Freitas. The club focuses on promoting and educating communities on how to protect the natural environment in Hawaii. Prior to my internship, they do some pretty cool things. Something that they were involved with was the Palila versus Hawaii DLNR. This initiated restoration of Mauna Kea's forest, and this was the first time a non-human status was granted in a court of law. They're also huge advocates for Red Hill. The club has been a strong voice of advocacy to address the contamination of the drinking water on Oahu due to Red Hill. And they continue to push for decommissioning and the cleanup of the facility. They were also kind to let me choose a passion project this summer to hone in on the issues that are important to me and strengthen skills in the field of advocacy. My interest in kaiāulu and legality in Hawaii led me to the kapa'akai analysis. It's a legal framework that is rooted in the protection of Native Hawaiian rights, established in the 2000 Kapa'akai Okaina versus Land Use Commission Hawaii Supreme Court case. This analysis aims to protect constitutionally protective Native Hawaiian traditional and customary rights in government decisions affecting Aina. My goal this summer was to take a deep dive into the analysis as a pathway to get Ike Hawaii and Ike Kupuna into legal spaces in Hawaii. So here's a little history on the case. In December 13th of 1993, Ku'upulehu Developments filed a petition for a boundary amendment to reclassify a little over 1,000 acres of land that was land that's classified as conservation to urban. Ku'upulehu Resort Expansion is what the project was called. They planned to build luxury development consisting of 530 single-family homes, 500 low-rise multifamily units, a 36-hole golf course, an 11-acre commercial center, a 3-acre recreation club, a golf clubhouse, and other amenities for the developments in that area. So this is a little map. As you can see, this map showcases the ahupua of Ku'upulehu, and right in this little crescent-shaped land is where their project location was. January 13th of 1994, the Land Use Commission required an EIS, or Environmental Impact Statement, to be submitted by KD for their proposed project area. On September 22nd of that same year, the Land Use Commission accepted KD's final EIS. The EIS had nothing to do with cultural significance in the area. It focused on mitigation of the physical environment, socio-economical conditions, existing infrastructure, and land use, but then again, nothing about the cultural impacts or cultural significance of that place. From October 26th to November 30th, multiple organizations petitioned this project. Some of the organizations included Kalahui Hawai'i, Kono Hawaiian Civic Club, and the Protect Kohana Ohana. They became known as the Kapa'akai Oka'aina Coalition, And then fast forward 6 years, September 11, 2000, the Hawaii Supreme Court overruled the reclassification of the land. And now we have legal precautions that protect our culture. So out of this case came the Kapa'akai framework. It's a 3-step legal process that developers and state agencies have to conduct. The first step is to identify the specific valued cultural history or natural resources in the project area. So what you got, what's there. The second step is to evaluate the extent to which they will all be affected by the action proposed, what you're doing, and mitigate and determine the feasible action to be taken by the agency to protect those resources if they're found, what you're gonna do. Those little parentheses are not my own words, that's actually what is on the public trust doctrine of the Land Use Commission training. I promise I didn't just make that up. Okay, so kind of a deep dive into how I conducted my research. A lot of it was based on qualitative data, so various legal documents. So here's some of the things that I used. This picture right here is the Katie's proposed action. This is their final environmental impact statement. It was like almost 800 pages, so no, I did not read the whole thing. This is the actual case of Kapaa Oka'aina versus the Land Use Commission. That was super interesting. And then these two little pictures down here are part of the mandatory training course that the land use commissioners must take, among other things. It touches on the Kapa'akai and kind of just anything in the public trust doctrine. And this is a document that the DLNR shares that shows their procedures followed that puts policy into practice. Okay, I also conducted interviews to kind of figure out how this all works. This is a chart that showcases what my mentor and I kind of thought up of people who would be important to interview. So we have people ranging from community members with Auntie Leigh Leitner to the executive officer of the Land Use Commission and all kind of everyone in between. I wanted to focus on answering the questions, how can the Kapa'akai framework be applied to decision making and what improvements can be made to it. Again, I wanted to talk to community members as well because they hold a lot of Ike Hawai'i on Native Hawaiian traditional and customary practices. So out of these 7, I got 4 responses, which is more than half, and I was really happy. Okay, the first person that I talked to was Daniel Orndenker. He is the executive director of the Land Use Commission. I spoke with Auntie Leigh Leitner, who's a kupuna of Kalaimano. She also sits on the Kupulahu advisory board. She's one of their community association directors. Ashley Obrey, who is a senior staff at NHLC or Native Hawaiian Legal Corp, and David Kimo-Franco, who used to work there as well. Some of my goals for the project were understanding the process of change. If we want to make change, we got to know how to make it happen. So my mentor, Wayne Tanaka, told me that there's 3 questions that I should focus on. What does the community want, who can get it, and how do we influence them? I also just wanted to understand the intersectionality between conservation and law. We've touched on this before, how we see these things as very separate entities, where it's kind of in the middle where real change happens. So going on to my interviews, this lovely man right here is Daniel Orndenker. He is the executive director of the Land Use Commission. He helped me understand exactly what the Land Use Commission does. He said that there's a common misunderstanding of what the LUC does. I also had that common understanding. He informed me that they use a quasi-judicial process that has the power to hear evidence, make decisions, and impose penalties. They're similar to like a judge in a court, but instead of creating new laws or policies, they just apply pre-existing rules to the regulations that we already have in place. He spoke about how the EIS is kind of where cultural practitioners and community members can be heard. By the time cases or any proposed projects get to the Land Use Commission, these things have already happened. So they're really there, again, to act as judges and take in all the evidence and listen to any testimony if there is any. He also spoke about how the Land Use Commission truly at its core is there to protect Native Hawaiian traditional and customary rights. Without it, there's no really government agency that does so. So there are some pros and cons, but the true root of the LUC is there to help. In that same day, I spoke with Auntie Leigh Leitner. That switch from having a super legal background to a community member was a little crazy for me. But she spoke about how the Kapaka'i framework was helpful in the success of the tri-weight proposal, which Li'ua spoke about earlier, but it's a 10-year rest period to stop gathering on Kalaimano's coast. One thing that she kept pushing was malama not management, and by that, I took it as she thinks that the people of that aina know how to listen to it best, therefore, they're the ones that know how to take care of it. And lastly, she spoke about changing the mindset. We need to look at aina not as a resource, but as a kupuna and something that we need to be aware of so that we can take care of it. Next, we have Ashley Ober. She's super cool if you have the chance to talk to her. She spoke about how the Kapaka'i framework is a good legal argument because it lets Ike Kupuna and Ike Hawai'i be the experts in extremely Western spaces. Some of the strengths that she said is there are written records that the state requires. So there's gonna be a record of testimony or anything that's found. Some limitations is she said that there's a lack of the second step, which is assessing the impacts. The second step speaks on how there are impacts, but no one really knows what they are, so it's kind of skipped. Yeah. Okay. The last person I spoke to was David Kimo Frankel. He is like a walking encyclopedia of all things legality. He spoke about how the people who conduct these EIS and that conduct the analysis are super, super crucial to the change that we want to see. He said the law is good, but the implementation of it is the issue. To this, he was speaking on the fact that a lot of legal jargon is extremely ambiguous. What means one thing to someone is not going to mean the same to someone else. So one thing that I pulled from the Land Use Commission case is it says the state and its agencies are obligated to protect the reasonable exercise of customary and traditionally exercised rights of Native Hawaiians. But reasonable means something different to everyone else. So that's why, kind of going back to my first point, the people who sit on these commissions is super duper duper important. And lastly, he talked about how the analysis needs to focus more on substance rather than the process of it. A lot of my research I found was that the analysis is something that people kind of just check off, like, oh, it's just like something that needs to get done, but it's not, it's really there to protect us. So after all of these interviews, here are some common themes I found. More community outreach and engagement from the state, equitable and open communication between Kaia'ulu and state agencies. David had shared with me that a lot of the times that action is being proposed, the people in the offices of the commissioners are usually developers because community members simply don't have the time to be in those spaces. So by equitable, I mean that everyone, Kaia'ulu members and developers, are given the same opportunity to be there and have their voices heard. And the last theme that I found was the need for translators. By this I mean community members and the state often talk in different languages. Legal jargon is extremely hard to understand if you don't have a background in it, and I would say most of us don't unless you dedicate time to learning it. So something that I compare this to is it's like trying to translate Hawaiian to English. Trying to do that, you're losing meaning. So by trying to communicate what community members want to the state, you're losing meaning of those practices that could be found. So something that I worked on this summer after finding that is a question that I posed to all of you and a question that I asked myself is how do we become translators in our own Kaia'ulu? And I found that my way is to write. So I wrote this lovely article that you all should read. It's great, but I think I'm obligated to say that because I wrote it. It's taking the mana'o that I gathered and making it into kind of an easier thing to understand. Like I said, legal jargon, kind of terrible. But if you have people who take the time to kind of break it down for community members to understand, then it's really useful. Because what's all this legal jargon if no one can understand it? How are we supposed to make change if we're not speaking their language? So I tried to take my mana'o and, you know, make it into something that's easy to read. It's 5 minutes. Read it if you have time. So through connecting with my aina and Kaia'ulu has given me a greater sense of compassion and fuels my fire to make effective change happen. This connection built through my internship has kept me rooted, and it's the foundation I build upon through endeavors I take both personally and professionally. After 10-ish weeks of being fully immersed in a different space, community, and a new set of responsibilities, I return with new understandings. New understandings of systemic issues that I would like to see change, new understandings of Kaiulu, and new understandings of myself. Through this, I learned that protecting our culture truly is protecting Aina. Our culture is so rooted in the need to malama Aina that if we can't protect our culture, we can't protect Aina. And if we can protect Aina, we can strive for environmental justice. This summer, shout out to my hui. Those are the ice shave baddies. You're lucky to know them. This summer has given me the opportunity and the privilege to dip my toes into spaces that I never saw myself in. The Sierra Club of Hawaii and PIPES has allowed me to merge my passion for advocacy, Kaiulu, and Aina. You might be thinking, what the heck is a social science girl doing in conservation? I don't know either, but I'm honored to offer a new perspective into a space that's so focused on Western science, and I can only hope to do the same for people like me. Although the summer has come to the end, that doesn't mean the work does. We keep doing the work by asking questions, being curious, and occupying space in the places that don't feel like they're built for us. I would like to say a huge thank you to my mentor, Sharda and Wayne. They're watching in there, hey guys. And the staff at Sierra Club, they were super welcoming. I wasn't the only summer intern. And to all the lovely people I spent the last 11 weeks with, it was such a privilege. And to all of our funders for their generosity and support and paying us. Thank you.
Rounds of applause for Kahinu.
Questions?
Questions in the audience. I see a hand up. Miss Ella.
You did amazing, first of all. I wanted to ask what you envision for the kapa'akai analysis and how you think that it can, or how it can be implemented to hold people Pohakuloa and stuff accountable, and in what ways you'd like to see its language change so that people are actually not just checking off boxes, but doing the work and doing the stuff.
Amazing question. So something that I thought of while you were asking that is the fact that there's a lot of ambiguous language. And at HCC, I got the chance to talk to Auntie Hannah Springer, who was their plaintiff for the case. And I spoke to her about the ambiguity of the language. And she said that at the beginning of every meeting or before they conduct an analysis, you need to sit down and you need to decide what these words mean for everyone. So that what reasonable means to me is the same understanding of what reasonable means to you in that case. I think what would be cool is having certain people, and I guess there are consulting firms that do conduct kapa'akai analysis, maybe ones that are more based on Ike Hawaii and Ike Kupuna, and maybe more cultural sensitivity training in these state agencies. I think a lot of times it's easy to not understand the weight of the job they have, so maybe a better understanding for those that sit on those kinds of boards.
Great answer. Do we have more questions for Kahinu? I'm curious, after the experience you had of these 11 weeks, you did just graduate. What do you see yourself doing next after PIPES is done?
Similar to my experience on Mauna Kea, I never thought I would be doing advocacy. PIPES really opened my eyes to conservation. I have this newfound, I don't know, tickle to explore it. I'm in the process of applying for Kupu Aina Corps, which I think would be a great way for me to broaden my skills, especially in Aina. I didn't get to spend too much time in Aina this summer, and that's something I would really like to do. So taking it upon myself and instilling that kuleana in myself for me to be in Aina and help.
Lovely. Excited to see you on the journey that you continue to be on. One last round of applause.
Thank you.
Oh, my Kahinu. And up next, we have Miss Eliana Filiva'a, who had the opportunity to work with Kahalu'ukua Heva this summer.
see a mouse? Oh, is it this one or this one?
They should both work, but you should use this one.
Okay. Talofa lava, everyone. My name is Eliana Filiva'a. I'm aloha mai kako. I'm super excited to share with you guys my project, and I just feel like so overwhelmed of, my heart is like spewing of all this support and just maybe I need to let off some air. Anyway. Yeah, I'm very excited to share my project, Nā Pilina Kalai Kapa Inoa I Kahalu Kuaheva, or relationships in taxonomy, specifically for male eye plants on Kahalu Kuaheva. Okay. This is also the first time my mentors are seeing my story map, so this is gonna be good, I swear. Okay, starting off, just a little bit about me. I'm from South Kona. Kealakekua and Ka'avaloa are very dear to me. And when I went into college, I didn't really know what I was doing, but I know that I cannot do anything that I don't like. I pretty much, I have to fully believe in something for me to, I guess, like stick to something. So I tried out a few majors, but my sophomore year of college, also, by the way, I went to some school in Missouri, very total polar opposite of Hawaii. And yeah, the summer of 2023, I did kupu, and that was like a huge eye-opening for me in terms of like conservation. I had took some environmental classes at school, but nothing like conservation, and specifically like in Hawaii. So that was definitely, if you knew me back then, it was a rough summer, but overall it was amazing because I found something that I think that I could like, regardless, the pay wasn't good, it was hard, but it was good work. And at the end of the day, I could like always count on that. And also during kupu, I met my current mentors, or actually just one of them, Jessie Ko'one and Kim. And then this summer I ended up with them through PIPES again. So that was awesome. And yeah, to go back to Missouri, after kupu, I took some more environmental classes where I did like transects. Again, total polar opposites. That's what my aina looked like over there. And now that I've graduated, here I am at Kahalu Kuaheva through PIPES. Kahalu Kuaheva is a very special place. It's a nonprofit organization led by, I just mentioned them, Jessie and Kim. They're dedicated to revitalizing one of Kona's largest remaining intact traditional agricultural field systems. And this is all through uplifting aina, culture, and community. But I swear there's like so much more to it. There's so much mana of the place. If any of you have been, I think you already know. It's a very forgiving aina, and I'm very thankful for that this summer. Our typical like daily activities were just maintenance. We did some clearing. We built a lot of things, and yeah. For my project though, so this is something that they have been talking about doing for a while, and then me just graduating, coming back to Kona, I really had no idea what I was doing in terms of like conservation. I did my like kupu internship, but I didn't really know all the plants, specifically for Hawaii. So it was kind of a perfect opportunity for me to talk about, and I guess my personal goal was just to explore and learn about Hawaiian taxonomical relationships. And here the tasks were to research varieties and compile a list, collect and plant varieties, creating resources for Kaia'ulu, aka the story map, another story map which will show up later, and then there's also some signs we made. And then expected outcomes, I of course wanted to deepen my relationship with aina, learn more Olelo Hawaii in the process, and improve stewardship abilities, which, spoiler alert, they all happened. And some of you are probably like, well, what is taxonomy? I didn't know what it was either. It's a branch of science concerned with the classification and categorization of living things into shared characteristics. It's usually those like Latin words. There's some in the earlier presentations. And that's mostly based on like a hierarchical, that's not how you say it, system. But in terms of, for Hawaiian taxonomy, there's way more respect to the inoa, and not just that, but like I guess how their appearance and any other connections to aina, akua, so many different things. Yeah, and then the Hawaiian word for taxonomy I found online was kalai kapa inoa, and there's just a deeper dive into it. Kalai being a scientific study, kapa to call term, and then inoa name. And then so these are the groupings that we went through with this summer. Because this was a project that they had already been thinking about, the groupings were already given to me, which was great because I didn't know anything. And yeah, we have ele, that's the dark varieties. Manini, the variegated, of course it's like after the fish. Hapai, relating to pregnancy. Maoli, the native varieties. La'a, the sacred ones. And Lena, the yellow varieties. There was more, but with timing, that was our final. And then for the male kanu, we will only focus on Mai'a, Kalo, Ko, Ava, and Uala. And then with all of this, I kind of dove in with the question, what are relationships in Hawaiian taxonomy, and how can this inform more meaningful environmental stewardship? So now into the preparation, our ha'alele. So for data collection, I just dove in. They gave me plenty of books, those were some of the titles on the last slide. It was a lot of information at first, so I had various docs, various notebooks with a bunch of stuff. That's why there's no pictures of it, because it's a mess and only I can understand it. And yeah, there is a range of topics I took notes on, planning techniques, traditional uses, mo'olelo, inoa, and any connections to akua. And then there's also some on-site preparation. We were prepping the malas to plant the specific taxonomy. So choosing those locations, I was also done with assistance from them, my mentors. There was also soil sampling, which we use as a reference, and clearing, compost, fertilizer, all the good stuff. We brainstormed like what will go where, as well as just like prepping the huaka'i. I'm so thankful they set up all different huaka'is across the island, which we will talk about right now. Yeah. So learning from the mokupuni, there was 3 sites we got to go to this summer. Our first one was, first one was Huimau in Pa'awilo. Sorry, how do I? There we go. So here, this was our Mai'a exploration. It was so awesome. We got to like try different Mai'a. We walked around the site and got to talk story, got to clean. Pretty much was very hands-on. I think later on there's even like pictures of us like removing the stumps. And then our second one was at Laupa'i Keola with Kamu and Kiahi to learn about kalo. And again, super insightful. Not only were we like gaining information, talking story, but we were also like helping out on their site as well. And again, I just have so much thanks for my mentors. They like provided lunch all the time, lays, like I'm like showered in just so much support, and thank you guys. And then last is our Ava exploration with Ed Johnston. He's like a huge renowned Ava researcher. So very awesome. Here we learned about somatic mutation and how like one plant, one little offspring or like stem can be a totally different variety, which is pretty cool. And then this is called Lukoheva, just for reference. And yeah, these huaka'i were awesome and aided immensely in my understanding and my ability to be able to name things. Whereas just reading about it was kind of tricky with like the descriptions. Oh yeah, and then here's just some more pictures. That's at Huimau. More exploration. Again, like you cannot do this just from reading a book. Yeah, before going into this project, I had no idea there was even such variety around me. Ava. And I think that's it. Okay, now the returning, the ho'ina. So with all the information we gathered, I compiled a final list, and the green is what we have. Almost all of it is already planted on site. I'll get into which ones are already in the 6 different areas on site. Yeah, oh, and then we made signs. And then the goal is to have this like little rock border around. We still gotta furnish it though. And then here's our planting. If you wanna scan this QR code, it goes more in depth for each of the sites. It's not fully fleshed out yet, but at least you can see like what the varieties look like up close. So I'll just click on all of them actually. So our hapai, again, we have the varieties. There's apuvai, the hapai banana. Okay, ele ele. I'll just show you guys what they look like. If you click on the QR code though, it'll be more in depth. There's even little descriptions of what mala, lena. So yeah, these were totally transformed from the beginning. Manini. What am I missing? Maoli. A super cool thing too, doing this project, I learned a lot about like listening to Aina, and that's something that was pretty tricky at first, but like Jesse, Kim, they all just trusted me to just try it out, and I didn't really know what I was doing, so it was kind of tricky at first, but it got easier as I kept going. So like this one, I wish I had a better picture. There's kind of like a little puka in the middle, and then there's my in there as well. And that'll be used for like compost and stuff. All right. And then for the ha'ina, just overall lessons. I struggled a lot in the beginning with the amount of information, and I think that was because like coming from a more like Western perspective where I haven't really dived into like native conservation, there's so much beauty and complexity and diversity to everything. I mean, how we categorize the varieties, there's so many other ways you could do it. And for me, that was really complicated at first. But it's like the more you learn, the more questions you have, but at the same time, the more like respect, I guess, is given. And you just appreciate more of like, when you think back in the day, just how well they knew their Aina. It's pretty like, just breathtaking, honestly, for me to like be immersed in this summer. And then of course, just being present, that was the first step for me. And every day it got like a little easier. And then the final reflection I have, just recognizing these taxonomic groupings serve as a reminder again of like who we are as Pacific people, how we got here, as well as that strength and resilience to keep pushing into the future. These varieties have lasted. I read about a lot that were gone, but regardless, there's a lot of mana, not just on site, but at all the other sites we went to. And I think just through knowing the names, speaking the names, like continuing to breathe life into these mea kanu is just another step for resilience, not just for community culture, but for yourself. It's way more grounding, and I think, again, just getting back to that idea of just building resilience, and especially right now in the current climate. And there's so many people, again, I want to thank PIPES staff and friends, of course. Thank you for accepting me into this program. So many eye-opening experiences as well that I never would have had the chance to experience if it wasn't for PIPES. Changing like my perception on not just Aina itself, but like how deep that relationship can go. Of course, Kim and Jesse, Kealohi Law, everyone for just their endless support. Wailea, my sidekick. And then this project wouldn't be even possible without all the huaka'i and all the people and communities that welcomed us and allowed us they were just so happy and like excited to share their information. So much love, so much thankfulness for everything, my family. I could keep rambling and rambling just of how like awesome this summer was, and thank you to the funders and the sponsors again. And that is all I have. Mahalo.
Amazing, amazing job, Eliana. I do think it's funny that at the end of almost every single intern's presentation today, they said, and that's all I have, as if what you've shown us today is not an amazing amount of work that was done in 11 weeks. And I think your presentation today really showed like the depth of Pilina that you got to build, so that is what you did. And with that, do we have any audience questions? Any Nina or wonderings for Miss Eliana?
Hi, Malia.
Hello, Eliana. Very proud of you, first of all. You talked a lot about how taxonomy has helped you to develop a relationship with Aina. I'm kind of curious if you had any specific examples or any specific times you can remember where you felt like you had a deeper connection to Kona, especially as someone who's from there and has spent a lot of time there.
I mean, just like when I come home and I look in my backyard and I see like not just all the things I want to do with it, but there's one day I was like picking tea leaf, and it was definitely like an invasive plant. I'll just point that out first. But it had like these white spots on it, and it kind of looked like tea leaf. I don't know what it was, but I was like, Elepaio hauli. Like it just things hit right away in my head now, and it's like, I don't know. It's just so much more just special and grounding to like continue working and living in this space. It's stuff that I wish I knew, that I wish everyone knew, like from Keiki days, but unfortunately that's not the case. So hopefully I'm gonna keep building on my story map to the other one, keep expanding it, and hopefully it becomes a pretty smooth resource for anyone to just hop in and kind of get to learn the names.
Like, I want to pay her the respect. Aloha, Eliana.
Aloha.
It is so nice to see you come through fruition of your education. So proud of you. It seems like you've earned or learned a lot of respect and relationship with the Aina. Do you have any suggestions on how we can get our youth to acquire this as well?
The first step I learned this summer, even if it's hard and you don't want to do it, just be present, go outside, whatever environment you can. I don't even have that big of a backyard. Or what's that, what did that one comedian say where it's all like trucks parked in your front yard in the grass? That's pretty much what it is. But still, there's like, we have beds, like, I haven't done too much yet. It's been a busy summer, but all these plants in my head, I think it all just starts with, just going outside, seeing what you can, finding creativity in that as well. Make it fun. Growing up, it was like a chore to like pull weeds and stuff. But seeing my papa like transform like a whole area, planning all these different things. It's also like more to it. It's not just like good to get your hands dirty, but you're building resilience for yourself. When I first came over here, we were renting on this like acre land, and we never had to go to the store at first while my papa was there, because he just planted all the vegetables we could ever want and need. Again, it's going back to that resilience. There's so much meaning and so much that can come from just trying it out, just planning something. Yeah, it can start small. Just go outside. Go outside and maybe don't have a distraction, just like reflect.
I saw a question in the front earlier. Ooh, from our other Kahalu'u girlie.
Ho'omaika ia'oi. On the first day of our internship together, our mentors had asked us, what was your first ever memory of being on Aina? And I just am so in awe of what you've put together and also the hana that we've shared together this summer. So I wanted to ask you to reflect even further and like, what is your most recent meaningful memory on Aina? And I better be in it.
Oh my gosh. Well, this whole entire summer, like when me and Vailea have to weed eat, she gets hers like first go, and I have to like, at first in the first month I was like always like asking someone, which was just like embarrassing. So sometimes, I don't know, Vailea was like my little role model this whole entire summer. I never even told this to her yet, but like, so easy to just follow. She would go start and be like, okay, yep, follow her, I know what to do now. And then I think too, Viola would pick me up and take me to work almost every single day, pretty much. I paid for her gas, don't worry. But yeah, I think spending so much time with you and seeing like having a hard day at work and then having our little like calm down jam session in the car just made the summer so enjoyable. There's so much, Viola, I could say. Love you.
Just out of curiosity, what do you think would be the next step to this project? Would you be like expanding into other crops or finding more like similarities in taxonomic terminology?
For sure. There's many ways it could go. We could add more varieties. One of the varieties that actually would be a super awesome resource for our Kaia'ulu was to have a Mawaiwai category. It was just really hard for me to find information on that. So I didn't get to do it this summer, but yeah. I think engaging Kaia'ulu in this process as well, and then finally when things are like sprouting and stuff, actually like being able to have community use it as resources. That's the end, like not end goal, but like the sooner goal, I guess, of, like before it. I don't know. I was talking about it with Jesse. I was like, imagine this in like 10 years. Like how crazy could it be? How big could this go? Yeah. Thank you.
My question was just, with all the varieties that you worked with this summer, what was like your favorite? If you got to like sample any, or yeah.
Ooh, that's a good one. I think my favorite to like explore was the manini, specifically the kalo. I had so much videos, but none of them were good enough for the story map. Like they weren't oriented right, and it was just, but it was like, that's another thing I guess I wanted to say earlier, like not being afraid to get your hands dirty. I was just like pulling apart the manini owali kalo and just revealing like layers and layers of the stripes. It's like, maybe because my favorite animal was like a zebra as a kid or something. But it was, I don't know, just having like such a comforting aina to explore in this summer has been awesome. And yeah, I think manini was probably like the craziest for my mind to be like, this is even real? Like I had no idea before this summer, which is crazy again. Like I wish we all knew this sooner, or maybe it's just me.
Can we get one more round of applause for Miss Eliana Filiva'a? And we're gonna stick around in Kahalu'u with our next presenter, Miss Wailea Tupo. Mekana pahana ola aloha vai ike kahi.
Velina mai e nā poe iwa o kona kai o pua I ka makani mā akua lapu e haile ia nei ka lele aka ho'opulu ia ai ka lau o lono e ho'ailona I ke ea o ka lāhui mai kahi kihi a kahi kihi mai luna a lalo mai kahi kina ai ke komohana ke aloha aina ia o kou pākahi a pau. Aloha nui. O wau iho no o Wailea Tupou, he kama no wau no o ahu a lua, ke ho'i ia nei I keia mau aina kupuna o hilo, kona a me kohala. I keia kauwela, he haumana hui a o wau ma ke ala hana o aina ma pipes. Okay. Eia au ke holomua nei I ko'u makahiki hopelua ma ke kula nui o Hawai'i ma manoa no ka laipua. Ona ke kele ʻelua a'u e ho'oko nei, oia ho'i o ka ʻōlelo Hawai'i, lawa o ka Ethnic Studies, a me kekahi palapala ma lalo o Women Gender Sexuality Studies. He waiwai nui ka ho'opili ʻana I nā kuana ʻike like ʻole ma ho'okahi ala nui, a no'u o ke aloha ʻaina no keia ala nui ho'okahi. Ma o ka huli ʻana I ko kākou kino I ka ʻaina I mea e ho'opiha hou ai ko kākou naʻau e holopono ana ka lāhui. No leila, e like me ka Eliana, I keia kauwela, ua pōmaikaʻi no wau no ka hana pū ʻana, no ka hui mālama ʻaina o ka haluʻu Koehewa. Ma ke ahu puaʻa o ka haluʻu, aia he ʻekolu haneli me kanalima kumaha mau eka, a piha pono keia ʻaina me ka momona o nā kupuna. O ka hapa nui o keia mau eka, ma kahi o kanaiwa kumaiwa mau pakeneka, he mau hiʻohiʻona kahiko koloko. E laʻa me nā pāpo haku kahiko, na auai I nalo wale ia, na makalua a pela wale aku. Eia no, kekahi palapala ʻaina me ka hoʻike piha ʻana I nā hiʻohiʻona kahiko I waiho ia, ma keia ʻaoʻao aia nā pōʻai pōlu a leila ma keia palapala ʻaina, kuhi keia mau pōʻai I ka helu pōlu kekahi. Ma muli hoʻi o ka hoʻonohonoho ʻana I ke lame keia ʻāpana ʻaina, ua hele a mua kaka hoʻi nā inoa o ia mau ʻaupana kekahi. No leila, o nā inoa ma ka ʻaoʻao ma ka ʻulaʻula, pēlā no nā inoa ʻili ʻaina a kekahi ʻiʻimi ʻai. Oi kelakela ka ʻaʻapo hou ana I nā inoa mai ka wā I hala. A mau koʻu hana piha ma ia wahi pana, oi aʻe no koʻu mahalo kekahi I keia mau hiʻohiʻona. Ma ka makahiki 25, 15, ua hoʻomaka ia ka hana nui a me ka waile ia ana I nā kumalaʻau haipilikia, pela no kekahi hana nui loa. A I keia mau lā, ua welehia no 5 mau eka, a kāla e hoʻi kau ka ikena o ia ʻaina. Ma ia mau eka 5, aia he 6 mau kihapai, kahi aʻu I oki mauʻu nui ai. A ma leila no wau I hoʻaʻo ai I ka ʻaina ma o ka ʻālele ana o ka lepo I koʻu ʻālelo. No leila, ua hoʻokahua wau I ka wahi pana I pūʻili me hana ai aʻu I keia kauwela, a e holomu ana wau I ka ʻolokeʻa aʻu I kūkulu ai no keia papahana nei. He papahana keia no ke kākoʻo ʻana I ka ʻaina o ka haluʻu ma o ka hoʻowili ʻana I 3 mau kia, keiki, ulupono, a me ahu puaʻa. O ka pahu hopu nui, oia hoʻi ka paipai mau ʻana I ia mau kia 3 no ka hoʻomau ʻana I ke ea o keia ʻaina nei. I nā hoʻohui keia kia 3, loaʻa hoʻi kākou I ka ʻolokeʻa kapa ia o kua. Mahalo wau I keia ʻolokeʻa no ka hoʻomanaʻo ʻana I nā kua ma ko kākou mau ola like ʻole. Ma kona, aia hoʻi nā kua iwi, nā kua o ka ʻaina I palena ai nā mala ma ke ʻano he pāpo haku. No ka lāhui, kuhi ia ka huʻolalo o kua ʻaina I kekahi makaʻainana I aloha nui ia I kona ʻaina. Ike kākou I keia manaʻo o kua, mana wahi like ʻole o ka ʻolalo ma kuahine. No leila, hahai maʻalahi ia ka waiwai o ia huʻolalo a me ka waiwai o koʻu pahu hopu no keia papahana. No leila, no koʻu haʻalele, eia no nā mea pono e hoʻokahua ana koʻu papahana holokoʻa. E wehewehe ana 3 mau ʻāpana o kaʻu ʻolokeʻa. I mea hoʻi e hoʻoko ai nā pahu hopu o kua, eia nā mea aʻu I hoʻohana ai, o ka mua, creating keiki resources, palapala kilo. No nā keiki, he mea maʻamau ke kipa pinepine ʻana I ka ʻaina ulu wehiwehi o ka haluʻu Koehewa. O ka hoʻoikaika ʻana I ke akamai o nā keiki, ka pahu hopu nui o ia mau palapala kilo. Iala ko I kipa I keia wahi pana, mai kai a iuka loa, e aapo koke ana lako I ka makau o ke kilo. A ohe lua e like ai me ke la. O ka lua, e laa ho'i me ka hapa nui o ka pai aina, piha ua nā moku I nā mea kanu o waho. A eia ho'i ke kahila ana me nā kumala au hei pilikia ma keia aina o kahalu'u. O ka mako hana nui, keia kauwela o e ho'i ka waile ana I nā hele a me ka oki ana I nā kumala au. Eia ke kahila ana o keia ki'i mai ka pule mua keia, a leila ma muli ho'i o ke kaia ulu a me ka hana nui o mau keia kauwela, eia no ka la, hiki no ke ike, malae. Mahalo nui wau I ke la. Mapopo lako, aloha nui wau I ke la apana aina. A leila, o ka apana hope loa, o keia papahana, o ia ho'i o ka ahupa'a, like ka maupopo ana, he aha ia mea he ahupa'a. Lua ole ka makau o nā kupuna a me ko lākou I ini no ka aina mamona. O ke ahupa'a, he hopena ia ma muli ho'i o lākou ea. A ma kona, a kaka ho'i, keia pili na aina, a me ka maupopo ana I nā kulana aina e like me kahakai, kula, kalu'ulu, a pa'a, and a ma'u. Nā leila e mōhala ana keia apana o ka'u olokea, mau ka wehe ana I nā hi'o na aina, a me nā momi o loko. Nā leila, eia ka i'o o ko'u pahana, ha'a no wau no ke ka'ana like ana I keia mau momi, a'u I loa mai ai, a me nā mea maoli, a'u I hana ma ka halu'u kuahewa. O ka mua, no nā keiki, he waiwai no ka maupopo ana I ke ahupua'a ma ka holo o ko'a. Nā leila, I mea e hana ai keia, ua kūkulu wau I keia puke, palapala, kilo, mea like. Nā leila, mau ka maka'i ana o nā keiki, e kilo ana lākou, ma e kolu mau apana, a me ka pane ana e hā mau ninau, he aha kau I ike ai, he aha kau I lohe ai, he aha kau I haha ai, a he aha kau I honi ai. Ma kahakai a I uka. And ea ka mea ālua, o keia kekahi mea punahele o u, mamuli ho'i o nā nina o hohonu, like oi aku no ka maalahi a me ka possibility, no ka ho'ohui ana I nā mana o kiko I o nā keiki, I nā aia ke keiki ma ka aina a ole, ma ke kahi lumi papa maoli, lumi papa maamau. Nā leila, kuhi keia palapala nei I he aha ka waiwai o ke kilo, an pehea ke kino ma kou kilo ana, nā mea o waho o ke kahi mana o haole, ea. Oh my god. There we go. And ea ke kahi mea, ke kahi palapala hou, he mea just like ho'o nui a ka mana o ke kilo ana no na keiki, a hiki ke imi I nā iloa'a keia mau mea o nā ao, o nā makani, o ia aina, o ke anila, pehea la ho'i, a me ka wehe ana. Nā leila, eia nā mea no ka ulupono o ka aina o ka halu'u kuahewa. O iai wau he homana no ma ke ala hu'ea o aina, ua mana'o mākou ma ka halu'u kuahewa, pehea e ho'ike aku ai I ke aloha no ka aina o kuahewa, a me ka ho'opili ana I ko'u aloha no ke kaia ulu a me ka mo'omeheu Hawai'i. No leila, mau ka waile ana, e like me ka'u I o lalo ai, o nā kumala au manako, waiwi, a pela aku. Ua ohi mākou I nā la'au kupono, I mea e pena a kukulu ai I mau ho'ailona no ke la me keia ki hapai. Eia no ka'u I hana ai. No leila, o ke ki hapai mua, oia o kalu'ulu. He inoa keia mai ke kahea ana I ia apana aina like mai ka wakahiko. A I keia mau la maupopo kākou I keia aina ma ka inoa o kona coffee belt. A ka ma'ane I ke ho'i nei ka inoa maoli. Manoa noa keia ki hapai pa'apu me nā mea kanu like ole. Ko mea elua, oi ho'i o ulu, ua mālama mua lākou ma ka halu'u kuahewa I keia ki hapai no ka ho'ohano ana I ka aina maoli o kalu'ulu. and aia he mea kanu ma like ole I loko o keia kihapai. O ke kihapai e kolu, o ia hoi o ho ike, kahea lako mai ia inoa mamuli hoi o ka nui poe. O keia kihapai kahi akalahui I malama nui ai, mau ka hana holo o koa ma ka halu ukua hewa. Oh wait, that wasn't ho ike. Ma ane ai, e ano o ki papa kalamai. O ki papa, he mea ia mamuli hoi o ka waile mua loa ana I keia ki hapai a ua loa hoi nā pāpohaku mai ka wakahiko nā pohaku niho kalamai. Pela no ke kumu no ka kapa inoa ana I keia ki hapai no ki papa. Eia no o ho ike no ka nua kanaka. Eia hoi o pue ai ole makaili na inoa o keia ki hapai. E like me ka mala ki papa he ki hapai keia me ka inoa o kahi mea I ho'ohana e ia ai e nā kupuna. Aia he mau pue pohaku o loko I o ia mala, kahi e kanu ai I ka uala no ka ulu hali I ana. Ka wahi mana'o a'ole I kana mai ka waiwai o ka ho'omau ana I nā ho'oilina moa kaka ana kupuna I waiho ai no kākou. Alila. Ke kihapai hope loa o nā mala ma keia aina, o ia hoi o nā helehele. He inoa keia no ka pilina ma waina o ka nāhele a puni o kākou ma ia aina. And a'ole I hiki ke ole I ka mana o uka, ka mea a me nā kaia ola o nā kumula'au. Ua le'a no keia a pana o kou papahana. No leila, no ka hopena o keia oloke'a o kua, I mea e paipai ke kaia ulu no ka ho'ohana ana I keia mau inoa kiko'i ma waho ho'i o ahupua'a wale no. Ua kukulu wau I keia mau palapala na kumu waiwai no ke kaia ulu. No leila, I nā maupapa kahapa nui o ko kākou lāhui, I nā pai like ole o ka aina a me ko lākou mau inoa pono'i e like me ka'u I o lelo wai. No nā kulana aina o kahakai kula kalu ulu a kahi e noho ai. E loli ana ka pilina ma waina o kākou a me keia mau wahi pana. O ka upahu hopu, o ia no ka makala kupono ana I nā mea pono o ke ahupua'a. A ma ka halu ukua hewa e ia nā ahupua'a features hoi. E like mea ke kahi mala, ke kahi okana, ke kahi ko'ele, ke kahi mau, he mau inoa kiko I keia I mea e ho'ohano ai nā inoa maoli. Eia hou nā palapala a ui kukulu ai no kela mau kulana aina, ma kona. No leila, no ka ho'ina, a ohe lua e like ai me ka pilina au I kukulu ai mau ka ho'i ana I ka mole, ka piko, o ke kanaka, o ia ho'i o ke ia hana aloha aina. Nā wai e ole I ka pomaika'i maoli o ke kukulu ana I ke kahi pilina me ke kahi aina. Mau ka mo'olelo, ka hana oki maui, burp, burp. a me ke kilo ana I ia hana o ka halu ukua hewa. Hewa ko'u maumaka a me ko'u kino ke aloha aku. O kona, he aina kupuna ia, no'u a mamuli ho'i o keia kauwela, ua ho'okokoke no ko'u maupapo ana I keia pilina ohana. Like a like ka ohana me kou kaia ulu. Aia he mau po'e a puni au me ho'okahi mea waiwai. No ka halu ukua hewa, lua ole ke kaia ulu. Kahi a kākou e ola ai. A pela no ka ha'awina nui o ko'u papahana ho'i a one. No leila no ka ha'ina. E like me a ui ha'i mua ai. He kamano wau no o ahu. A ma ka ho'omaka o keia kauwela. Ua ha'alulu iki kana au. Ma muli ho'i o nā wahi pana hou a ui hehi maluna ai. A ka ma ka hopena o keia wā ua emo ole lu'u piha I keia mau wahi pana. O iai ke ho'ea nei kākou I ka hopena. Tears, literally. I ka hopena o keia hana hu'e a'o. A ole I pauloa ko'u mahalo no ke la me keia po'e. Ma keia ala nei o ko'u kauwela. O ka'u hana nui I mamake ia ma keia papahana. O ia ho'i ke kukulu ana I nā kumu waiwai kaia ulu a me ke ka'ana like ana I ke kaia ulu. Mau ka lu'u hohonu ana I ka'u hana I keia kauwela. Ua lo'a piha na ha'awina. He wahi leo mahalo ke ia e ku'u mau alaka'i o Kim, lau o Jessie ke koa ka ho'onei. Na kāko'o me na wahine, mana wahine I aloha nui ia ia kua hewa o ke alohi la a me Eliana. Ha'a ho'i ko'u na au, ma muli ho'i o ke ia kauwela a me ka lilo ana I kekahi kanaka nona ke kule ana o ka malama aina. Mahalo.
Ho'omai ka'i nui e ke hoa. Just ha'aheo valeo ya oe. And I'm not gonna cry. So I need someone else in the audience to ask a question.
Aloha e wai lea.
Aloha.
Ha'aheo wau ya oe. E ke hoa. Okay, e nino ana wau I kekahi ninau e pili ana I kou hana ana maka'aina. O ka'u, he aha kekahi o na momi, kekahi o na manawa au I hana I maka'aina he mea I puina ole ia. He kekahi o na manawa, kekahi o na aina au I kipai. Yeah.
Well, ke kauwela holo o ko'a, ua momi. But mo na mea a pau ma ka halu'u kuahewa, palana ole ko'u mahalo no ia aina. Aka o kekahi huaka'i a'u I mamaki nui ai oia ke kipa ana I ka aina o Honoka Hauiki me nā hoa o Kamu. Yeah, I think like oiai aloha nui wau I ka aina me ka hana ana mai ia aina o nā po'e ka mea e aina ai ka aina. And e like me ko'u mau alaka'i a me kola ua mau hoa ma Honoka Hauiki. Ua ike le'a wau I ka eleu o lākou a me ka ike le'a o nā pilina ma waena o nā kalo Hawai'i a waho kekahi. And oia ko'u mahalo nui, yeah, nā po'e, yeah.
Mahalo.
Mahalo.
Aia kekahi ninau ma ka YouTube. Ua ninau ia e Kealo. Mahalo ia oe e Kealo no kou noho ana I ke ia la holo o ko'a. O ka ninau, ma waino o na kiha pai a pau au I hana ai I ke ia kauwela o wai kau kiha pai puna hele.
He pane ko'u ke ia. O ki papa ko'u kiha pai puna hele mamuli ho'i ka ma'ala hi o ka oki mau'u ana. A ole nui loa na pohaku no ka pa'i ana I ke ia alona ni. No, yeah, and ma waho ho'i o ke la, e like me ka'u I o lalo ai, ina ike wau ina hi'ohi o na kupuna, like ma leila no ko'u mahalo a ko'u ka mea puna hele no'u. No ka pilina ana I nā pohaku I waiho ia ai e nā kupuna. Like, hiki au ke haha I ke lama pohaku, and oia ke kuma no ka puna hele, yeah.
Ho'omai kai. Nani vale. Hewa na maka ke nana aku. Can we get one last round of applause for Miss Wailea Tupou? And rounding us off for today, our last presentation, Miss Kamalani Poi Poi. Remind us again what episode just finished, because she's been going off all summer long.
Episode 30. Let's go. Yee. Okay. Okay, we're ready. Aloha mai kako. Mahalo for giving me the opportunity to close us out. Mahalo nalu for putting me last. My name is Kamalani Poi Poi. I am from Waianae Makaha, Oahu. Just like Ho'oli, he is my brother in Aina. And I am an undergrad student
here at UH Hilo, and I am studying geology. Kind of want to focus on volcanology and going to grad school maybe later on in life. And I'm also minoring in astronomy, so I kind of want to intertwine all of that into the future for my career. And for this summer, I will, can't even see it. My project this year is interdisciplinary analysis of kilo whale petrology and seismology. So we're going to be talking about integrating earthquake data and petrology analysis of kilo whales 2020 through present summit eruptions to kind of figure out how and where magma was stored prior to eruptions. And I would like to mahalo my mentors, Kendra, Liz, and Ninfa. I love you guys. Mahalo for, having me this year again. I'm getting emotional. And I want to mahalo Tutu Pele for showing up today for me. I feel like I've gained a lot of pilina with her, and getting emotional. But yeah, mahalo to everybody. So for my hua, the reason why I'm here, I am a returnee. I did PIPES, the internship last year in 2024. And this is the summer that kind of opened all of the opportunities and of research for me. And yep, second round of PIPES, and I'm having a great time. And my host organization this year again is USGS HVO, and some of them are sitting in here too. Mahalo you guys for coming. And yeah, this is a picture of last year of us in Waipi'o. This is one of my favorite pictures because it's kind of showing us just being there in the aina and getting pilina and hukiing kalo, which I also love to do. I am the Ka'au pathway, the transformation process and kind of connection to data articulation. And much like being a volcanologist or aspiring to be a volcanologist, we do a lot of kilo, of observation. So I feel like this pathway was perfect for me and kind of like to kind of grow more as a young scientist. And kind of an overview of last year's project. I have done this before, but I'm still nervous. So I kind of did some research on kilo whale's June 2023 eruption, and I kind of focused on olivine, which is a mineral that we'll be discussing later on, and kind of exploring of what's happening inside of kilo whale's magma reservoir systems. And a lot of research kind of tells us what's happening inside, kind of like looking at the bigger picture. And by doing that, we kind of, we want to do that to better inform local communities because volcanoes are hazardous, and we want to make sure that we have the right information to give you guys for safety reasons. And also, there's a data release that is in review right now of last year's project. So all of my lab work is in right now for review to release. And basically, I'm getting published. Who would have thunk? And what a data release is basically all of the lab findings that can be released to the public so they can use to kind of reference to it if they want to do a project. And yeah, reference to it. So yeah, talking about olivine, it's a mineral that's filled with history. And olivine is a green mineral or crystal that grows inside of magma systems. And it acts as a stopwatch. And basically, if you look at its chemical compositions, it can tell us where it has been or the environment that it has been in prior to erupting. And this is a picture of the pretty olivines under a picking scope. They're like gold to me. And basically, we kind of look for the olivines forsterite. And what forsterite is, if you can imagine a triangle, and you put iron on one side and magnesium on one side, it's basically the forsterite is the ratio of those two elements. And wherever that olivine kind of lands is where we can plot it on top of concept figures. What is happening? Okay. So the number that it gives us, it kind of tells us where it's been prior to eruption. So So like the magnesium and the iron can tell us that number. It can tell us where it's been. So Halima'uma'u Reservoir and Sal Caldera. So this is the deeper magma reservoir. And we also found out that the lava lake up here is a potential reservoir for olivine to grow, which we didn't know. So hooray for new findings. So I want to transition from my last project into this year's project. The reason why I wanted to do pipes again this year is because I had a burning question. And what happened was I was in a volcanology class last fall with Liz Gallant, which is, don't tell the professors, but she's one of my favorite professors ever. This is Liz Gallant. She's amazing. And Nina von Beddington, also one of my mentors this year, she came to talk to us about earthquake data. And this is my wonderful class that we struggle bonded in. And while she was talking to us, I kind of thought about how can is olivine affected by earthquakes? And if so, how? So that's kind of why I wanted to kind of frame the project this way. Kind of do interdisciplinary research so that I don't need to only focus on one thing like olivine. I want to kind of broaden my horizons a little bit. And I think this research is important. It's because it can create a gateway and more opportunities for research to kind of work towards and figuring out why Kilauea is evolving and changing the way she is now. 30 episodes is a lot. Going to the ha'alele, this is also made by Professor Liz Gallant, and this is kind of just a brief view of like a timeline of all of the eruptions. On all of the pictures that you see up here is all of the Kilauea eruptions, and all of the eruptions down here are more on the rift zones. And the colors that you see is a duration of each eruption. All these squiggly lines over here is all of the seismology, so all of the counts and amount of earthquakes. And then all over here is the amount of magma that has been going into the caldera. And just to give you a little bit of context, this is what Kilauea looked like in 2020. This is December 28, 2020. And this is also like a thermal map. And compared to last year in 2024, it's filled up dramatically. And due to recent findings, it's about 40% filled up, so we're almost halfway there. That's crazy. And kind of to like put it out there as well, whenever there is an eruption happening and we like go out to observe it, that is the last time that you're going to see it that way, because the next time you go there, it might not look the same. So you yourself have a mental image of what it looks like, and you're probably one of like the only ones who do, But yeah, kind of a food for thought. This is your guys' favorite part, I think, because you get to see more lava. And so right here is the locations for all of the vents where all of the magma came out of for all of the eruptions. So this is December 2020. So this is the first eruption that happened right after, not right after, but 2 years after the Hulihia of 2018. So that was the one that kind of destroyed the Leilani Estates. Very catastrophic. And this is the one that lasted 5 months. And this eruption kind of produced 16,000 Olympic-sized swimming pools worth of magma. So that is a lot of swimming pools. And mahalo to Mike Zoller. I don't know if you're online peeps, for giving me the data to kind of like plot and give you guys context of where it's been coming from. So the September eruption, filling up a little bit more, 16,000 pool. And this one is the longest one that we've had. It's like 14 months within this time series. And it's filling up more. This is actually lichens research project. Lichen, are you in the crowd? Yeah, you. So she did a lot of work on this project as well. Shout out to my fellow peeps. Yep, 18,000 Olympic-sized swimming pools. And this is my baby, my baby eruption. So filling up the lava, this was only 13 days. And as we kind of go on, it gets a lot shorter. This is what we call a fissure. And what a fissure is kind of like a crack inside of like, basically a crack, and all of the magma kind of seeps through. So all of the magma doesn't come out in one spot. It kind of like seeps out and makes all of these like fountains that you kind of see. So there's not only one, there's like a bunch. And then 2,000 Olympic-sized swimming pools. And then episode 1, the one that started it all. But yeah, and this was 3,720 Olympic-sized swimming pools. And this was the shortest one. This has lasted for 14 hours. I think this is not the one I went out on. I think it was an eruption that I went out on New Year's Eve. I think it was New Year's Eve. Yeah, that's the one. But she is amazing. The Huaka'i. So I've been, this summer, I've been working on navigating through Valve. And Valve is a internal data center that USGS HVO uses to access all of the earthquake data. And basically, you have all of like these settings over here. And you can like put in all kinds of dates and tell you how many earthquakes there were and where they kind of came from. And I use this to extract all of the data to kind of plot later on in my coding. And I basically learned a new language this year. So mahalo to Ninfa for being very patient with me this summer, because I am not a coder. It's one of my weak spots, but this summer has kind of given me the challenge to kind of overcome that. And I've been using MATLAB, which is a coding program used for like data analysis, figure producing, all that kind of stuff. And the best way that I can say of how the coding languages is kind of like coding pigeon. It's because there's C++, Java, Python, and a bunch of others, I think. All of those languages mashed together. So I've been spending weeks kind of trying to understand how to have the code actually work. And yeah, so I spent most of my summer trying to code. It has been very challenging. And this is a fun little treat for you guys. This is my little axolotl lamp. And I've used the rubber duck debugging method, and it's basically a method for programmers to kind of help debug their codes when it doesn't work. And basically, you talk to this little thing, like it's somebody who doesn't know coding, and it helps you kind of articulate your thoughts, kind of slow down and see, you know, where it went wrong. And this method is comma approved because it really works. I have talked to this thing all summer, and just say, hey, why isn't this working? So this line says, but this line says, you know, that kind of thing. People think I'm crazy, but it actually works. But yeah, so he's my buddy for today. His name is Alistair. But yeah, so doing all that coding, and I did each of these these codes for all of the eruptions, so all 6 of them, all the way from 2020 for all the way to December 1st, so episode 1. And each of the code is kind of unique because you have to kind of switch out a bunch of things. It's very technical. And all of the coding kind of blurps this out. So what we see right here is all of the seismicity that's leading up to the December 2020 eruption. And we want to do this is because we want to figure out if there's a relationship between the olivine and the seismicity. So in the first graph, we can see that there's, it's a histogram. So each of these bars represent the amount of eruptions that has happened in that day. And then this one is a cumulative count, so it's kind of like adding all the eruptions. And then this one is a graph of the olivine. I'm still learning about this, so please be gracious with me. And basically, each of these points represents one olivine. And, but yeah. So what we can see here is an intrusion. And basically what an intrusion is when the magma makes its way to the caldera, so it's kind of like filling it up before it erupts. And we can see over here that there has been a spike. So some of the instruments pick that up. And as you can see over here, there's like a little bump right there. And when we try to put the, try to relate that with the olivine, it pretty much matches because all of the olivine that has been sitting, kind of just been sitting, it's been sitting, and then all of a sudden it does a deep dive. And this means that all of the olivine has really been moving, and so it matches up with the intrusion. So this is saying basically that the olivine has been moving with this intrusion. And then the same thing with the September 2020 eruption. There's a lot of eruptions. This one doesn't seem to kind of add up. And it's because if we do expand this all the way, it doesn't show that it's kind of like plummeting at the same time. There is an intrusion over here, but yeah, it doesn't match up. How do I get out of here? Okay. And yeah, if we compare it, it's very different. And then looking at the seismic maps, remember when I was talking about intrusions? These can indicate intrusions from different types of sources. So magma can come from all types of different sources and not just from one source. So over here for the December 2020 eruption, this is some of the magma came from the East Rift Zone, and then most of the magma came from more of like the shallower reservoir. And as you can see, the circles or the sides of the circles indicate the magnitude of the earthquake. And the darker the color, the deeper it is. So there's a lot of like orange and reds and yellows, so it's fairly shallow. For the September 2021 eruption, there's kind of the same. There's a little bit of magma supply coming from the East Rift Zone, but primarily it came from the Salt Caldera, so that's kind of like the deeper and hotter reservoir, all the way on the bottom. And then just a little bit inside of the Halima'uma'u reservoir as well. Ooh, magma. So kind of a recap. The earthquake maps kind of shows us that there are different types of pathways that the magma can come from. And there is evidence of intrusions before eruptions that is pushing its way into the surface. And the olivines tell us that they have been sitting inside of the reservoirs at different time spans. Forgot to mention that because this sat in longer, this is kind of like, this is 700 days and this is 2,000 days. This set of olivines kind of sat in longer in the 2020 eruption rather than the September 2021 eruption. So because this is a longer time span where the olivine is sitting, it doesn't mean that it came out kind of like the same as this. So they kind of differ in a way. And the olivine diffusion tells us that sometimes the olivine data doesn't kind of align with the earthquake data, and that's okay. And that's also telling us something about Kilauea. She's very random. And as I said before, we use multiple types of data in order to kind of tell a story. And last year, I only focused on olivine. And because I wanted to kind of answer my question about the seismology side of it, I wanted to like expand my horizons. And for my ha'ina, I kind of wanted to put forward that I am still a undergrad student, And this is one of our, I was in field methods, one of our classes, and whenever we would be outside kind of practicing like the methods that we learn in class, when there would be an eruption, Steve would be like, let's go see the eruption, and we just like ditch everything and go. So this is one of the eruptions. My geology peeps can attest to that too. This was episode 26. She was very beautiful. And I want to keep, I want to keep sharing my work, and I want to keep on inspiring Kanaka Maoli or more Kanaka Maoli to do like volcanology and geology work, because there's not much. And I feel like we need a lot more of them. But yeah, this is when I hiked Pu'o'o and completely wrecked my ankle. And this is me doing some lab work, and here's some of Pele right there. My first time riding a helicopter, and my class that I love so much. And this is me kind of, doing some measurements on the eruption. And some of my peeps. I love you guys. And yeah, I just want to keep doing the work, and I want to keep, going. And I wanted to give a huge mahalo to the PIPES the PIPES staff, all of my PIPES peeps, Kendra, Liz, and Ninfa. Mahalo for being my mentors again. Everybody at USGS HBO, I love you guys. Mahalo for having me in your space again. Megan, who is also my boss for the co-op position, and the geology department, and the Holy Maaloa Foundation. And I wanted to leave you guys with a Olela no Eao, Ikawa Ma'amua, Kawa Ma'hope, the future is in the past, because in order to understand the future, we also have to look at the past. Mahalo. Are there any questions?
There have to be tons of questions. I feel like every
my
time I
God.
see Kama, I have a new question for her about the Pele. So, Seth, what are we wondering about today?
I might not know the answer, but I'll try.
For your data set, you were saying that it's like over 2,000 days. Because these episodes are happening so often, are you able to look at this at a much smaller scale, like in the past 30 episodes, rather than a whole 2,000-day data set?
So for each data set that we get for each episode, it's not always 2,000 days. Sometimes it's a lot shorter. So diffusion modeling is basically measuring how long the olivine has been sitting in there and how it moves through the magma reservoirs. So yeah, I think it kind of depends on each eruption. It's not always 2,000. Maybe it's like 60 days. Yeah.
Do you how different would that look?
Kendra, help.
Like how different would like the graphs look compared to a long data set compared to like a small?
Yeah, so the September eruption definitely is quite long, and we still don't fully understand why. That's part of what Kama was working on this summer. And I'll say that for the current eruption, like Kama's coming back to work in the co-op position outside of the PIPES program, and we'll be doing that work this fall.
Yeah, sorry, Seth. Well, I also can answer that we don't have all of the olivine data sets yet. So when I do have it, I'll give you a call.
Maybe one or two more questions for Kama.
Hey, Kama, excellent work. I was just curious if you had to choose, what your next burning question would be, if you had one that you came across while you were working here.
I definitely want to dive into tilt meters more, so more on deformation, more on gas, if can. But like I said, I don't want to just focus on one thing anymore. I want to kind of be a master of everything else. Like, kind of tell the story in a more not a very specific way, but a more broadened way. Kind of step back and look at all of these different information and how it relates to each other, kind of. Does that answer your question? I also want to say that I am a aspiring planetary volcanologist, so I want to study volcanoes on different planets. So, yeah.
We have time for one last question. Hannah's gonna make me run across.
Mahalo, Kendra, for answering that.
Does the olivine have to be relatively new, or could you look at the picture of olivine that you see at like a beach, for example?
I'm sorry, say that one more time.
For the olivine, like looking at how the structure indicates where it's coming from, does it have to be like freshly emerged olivine?
No, I don't think it does. No, it doesn't have to be. It can be like magma that came out like, I don't know, 10 years ago, and you can still kind of look at it. That's kind of one of the wonderful things about olivine is that when it comes out and when it forms and comes out, it keeps all of that information with it throughout its lifespan. So yeah, great question.
Can we get one last round of applause for Miss Kama Poi Poi? And the axolotl.
Hola. Hola. So that's going to conclude our first day of the 2025 PIPES Intern Symposium. Can we give another round of applause for... Awesome job, interns. We still have day 2 coming up tomorrow. I'm going to ask our interns, we're going to close out with a mele mahalo to everyone on our internet audience as well as our audience here. So if we can have the interns come in the corner, and we'll do a quick mele mahalo to close out this space. Once again, mahalo nui for coming. Thank you for being here and supporting the next generation of PIPES people coming into the workforce. This is a little mele that our team wrote. Hoi, hoi. Bra. Where this is a little mele mahalo that our team wrote and our staff wrote in a little retreat with one of our the kahuna of PIPES, Manaya Kalani Kalua. He's gonna love that. And it kind of talks about the characteristics of Mokaolele, which is Ili Aina, that is here at UH Hilo. And we believe those characteristics are also within our interns. So without further ado, I think we're going to try and jam this one for you guys. Mahalo. Mahalo. Thank you for coming.